Plants are complex systems. Like the road systems of cities, they have a network of pathways. Instead of cars, trains, and trucks, you have water, food, and minerals. And just like with roads, you have both one-way and two-way streets. Paths to carry water and minerals from the soil, and paths to carry food from the leaves. Today, we're going to talk about a part of the plant that's particularly important for transport, the stem. The stem of a plant is one of two structural parts of a vascular plant, a plant that has tissues for moving water and nutrients, the other being the root. The stem is the part above ground, which provides support for leaves and buds. It's like the major highway of a plant, and it's vital for plant life. Nodes are the places on a stem where leaves and buds are found, the exits or intersections, and internodes are the areas in between nodes. People often confuse stems and shoots, but shoots are just fresh plant growths out of the ground. These growths can include both stems and leaves. Anything that's newly grown and sticks out of the ground is considered to be a shoot. Stems have several jobs. They provide support for the leaves, flowers, and fruits of the plant. They help the plant reach for light. They transport water and minerals. They help store nutrients. And they produce new plant tissue. If you were to look carefully at the cross-section of a stem, you would find several layers inside, each of which has a different job. From the outside to inside, the layers are bark or epidermis, phloem, cambium, xylem, and pith. The epidermis is the outer layer of the stem. It adds stability and protects the plant from wind and rain. It is able to do this because its cells are wax-coated. This is like the foundation of the highway. The phloem is one of the transport tubes of the plant, moving sugars, which are used for energy, around the plant. This is essentially the food of the plant, so the phloem's job is to distribute food. It's like a slow lane containing a supermarket convoy of trucks, all full of groceries. The cambium is an area of high cell growth. It has the phloem on the outside and the xylem on the inside. The cambium provides cells for both layers on either side, increasing the width of the stem. It's similar to an area of road construction where extra lanes are being added. The xylem is a transport tube like the phloem, but instead of transporting food, it transports water and minerals from the roots. It also provides the bulk of the plant's support system and is the more woody part of the plant. This is an especially large feature for trees, which contain large amounts of wood that we use to build homes and furniture. The xylem is like an express lane that only goes in one direction, always up from the roots. The pith is the very center. It becomes a hollow area in some older plants. In trees, it contains solid, hard wood fiber that was originally part of the xylem. The pith is the median strip in the middle of the highway. They can be anything from a small barrier to a grassy area as big as the road itself. There are two main types of stems, woody and herbaceous. Woody stems are tallest, thickest, and firmest. Most trees fall under this category. They tend to have bark on the outside, but there are also many types of shrubs that fall underneath this category. Woody stems include young twigs, older branches, and the trunk which provides the main support. Herbaceous stems are stems that are mostly supported by water. If the plant lacks water, the stems bend and fall toward the ground. Most garden plants and vegetables with soft green stems fall under this category. Other herbaceous stems include bulbs and tubers, and less green vegetable stems, like potatoes and onions. The stem of a plant is one of the main structural parts of a vascular plant, which provides support for leaves and buds. They're vital for plant life and act like the major highway of the plant. They are also made up of nodes, which are the places on a stem where leaves and buds are found, the exits or intersections, and internodes, which are the areas in between nodes. From the outside to inside, the layers of stems are bark or epidermis, phloem, cambium, xylem, and finally, pith. The epidermis gives stability and protection, like the highway's foundation. The phloem transports food, sugars for energy, around the plant, like a slow lane containing trucks full of groceries. The cambium grows new cells for the xylem and phloem on each side, expanding the lanes of the road. The xylem transports water and minerals from the roots, like a one-way express lane. The pith is the median strip. It can be hollow or not, and can contain wood in some plants. There are two main types of stems: woody, which are tallest, thickest, and firmest, and tend to be trees, and herbaceous, which are stems that are mostly supported by water.
Within plants, there are two systems of structures, shoot and root. For now, we will focus on the basic structures of the shoot system, but in the future, we will look at the growth of the shoot system as well as structures and growth of the root system. The shoot system includes the above-ground structures of plants. That means it includes the leaves, buds, stems, flowers, and fruits of plants. Because of all these different parts, the shoot system is rather complex. Even with this complexity, there are a few basic structures of the stem that we will focus on in this lesson, in addition to differences in structures of stems based on the classification of the plant. As we just went over, there are many different components of the shoot system. In this lesson, we will focus on the stem. The plant stem is best identified as the part of the plant above ground that provides support for other structures. Some of these other structures include leaves and flowers. Within the stem, there are several types of tissue. Before we look at the specialized structures of vascular tissue, let's cover a few other forms of tissue. The meristem is the tissue of the stem capable of cell division. The meristem is where the stem growth occurs. We will look at this more in the future. The next tissue is similar to our skin. We have skin to protect us from outside factors and to keep our insides inside. Plants also need something to protect their inner structures from outside elements. On the stem, this is the epidermis, which contains wax-coated cells for protection. The majority of tissue in plant stem is called ground tissue and basically fills the space around the vascular tissue. We will look at vascular tissue after going over the three types of ground tissue: parenchyma, collenchyma, and sclerenchyma. While these words sound very unfamiliar, you may have noticed that they all end with enchyma. This root word means cellular tissue. Parenchyma is the most common form of tissue in plants and provides a variety of functions, including the storage of food and water. Collenchyma tissue helps support young stems and roots. Lastly, sclerenchyma tissue provides rigid support and protection for the plant stem. The Greek term sclera means hard. This can help you remember that sclerenchyma is hard tissue that provides rigid support. Let's now look at some specialized types of vascular tissue found in plant stems that is surrounded by the ground tissue. Previously, the concept of vascular tissue was addressed. Remember that vascular tissue is the tissue used to transport water and nutrients throughout a plant. They act like roads and plumbing, moving around nutrients and water needed by the plant. There are two types of vascular tissue, xylem and phloem. Xylem transports water and dissolved minerals, while phloem transports food. Previously, it was pointed out that an easy way to remember which vascular tissue is which is that phloem and food both start with the same sound. This may help you remember that phloem moves food and that the other tissue, xylem, moves water. Now that we have the basic concepts of xylem and phloem, let's look at their structures a little bit more in depth. We will first look at xylem. Xylem is made of tracheids, which are non-living, elongated cells to allow for the transport of fluids. The xylem can sometimes help in the support of the stem. The movement of fluids in plants is generally from the roots up through the stem to the leaves. While xylem is made of non-living cells, phloem is always made of living cells and transports nutrients from the leaves down through the stem. Phloem is made of sieve elements, including sieve cells, plates, and tubes that are specialized for the movement of food in plants. Botanists use the arrangement of vascular tissue and plant stems in order to help classify plants. We will focus on the differences seen between monocots and dicots. First, let's review these terms. In a previous lesson on plant classification, we talked about the two types of angiosperms, or flowering plants, based on the number of seed leaves. Remember that the term that scientists use for seed leaf is cotyledon. Flowering plants are either considered to be monocots or dicots. Monocot is short for monocotyledon, meaning one seed leaf, and dicot is short for dicotyledon, meaning two seed leaves. Monocots are simple flowering plants such as grasses, corn, and palm trees, while dicots include roses, sunflowers, cacti, and apple trees. We talked about the differences in seed leaves, flower arrangements, and leaf structures previously. However, we will now look at the differences in the arrangement of vascular tissue. In monocots, the vascular tissue is found in paired bundles of both xylem and phloem. These bundles are dispersed throughout the stem as seen in this diagram. The structure of vascular tissue in dicots is probably more familiar to you. In order to figure out the age of a tree, you count the number of rings. For example, a tree that is 30 years old will have 30 different rings. You can even tell when a year had a significant amount of rain versus a year when the tree may have been through a drought based on the spacing of these rings. This ability to determine the age of a tree is due to the vascular tissue, specifically the xylem. In dicots, the xylem and phloem are found in rings, the xylem on the interior and the phloem on the exterior. The xylem must be made every year as the stem, or in the case with trees, the trunk, continues to grow. The rings that you see in a tree trunk are the xylem that is made each year. The phloem are found closer to the exterior of the trunk and can be used to get sap. You may have seen trees being tapped for collection of maple syrup. When people do this, they are basically breaking into the phloem of the tree in order to get the sugars that are transported from the leaves down the stem. The shoot system of a plant consists of the parts that are above ground. We specifically looked at the stem in this lesson as it provides support for other parts of the plant and provides a place for transit, similar to a highway that allows goods to be moved from one place to another. We looked at the core tissues found in the stem, specifically focusing on the ground tissue and the vascular tissue. Remember that there are three types of ground tissue: parenchyma, which is the most common form of tissue and stores water and food; collenchyma, which helps support young stems and roots; and sclerenchyma, which is hard tissue that provides support and protection for the stem. There are two types of vascular tissue: xylem, which moves water, and phloem, which moves food. These structures are arranged differently in monocots and dicots. In monocots, which are flowering plants with only one seed leaf, the xylem and phloem are found in paired bundles throughout the stem. In dicots, which are flowering plants with two seed leaves, the xylem and phloem are arranged in rings with the xylem on the inside and the phloem on the outside. Phloem can be tapped to collect sugar such as maple syrup, and xylem is responsible for tree rings. While the structures of the stem have a variety of names and purposes, all of the structures of the plant stem are vital to the survival of the plant itself. Roots serve many purposes for plants, including the storage of food and the intake of water. Before we look at the structure of roots, let's look at a few examples of roots. While there are numerous classifications of roots, we will only look at a few. The first one is prop roots. These help brace plants against the elements, such as wind. Corn plants have prop roots as seen here. Some plants, such as carrots, beets, and turnips, have roots that are designed for food storage. These are generally the roots that you may eat. The last type of root we will look at is designed for water storage. A classic example of this can be seen in pumpkins. We know that different types of roots have different functions, but different parts of the root also have different functions. Let's now look at a few key components of most roots and relate the structure to the function. While doing this, we will look back at this diagram of a root in order to better identify the layers. Let's start on the outside and work our way in. When at a construction site, people are required to wear hard hats in order to protect their heads. In roots, the root cap serves a similar purpose. This outer area of the bottom of the root protects other root tissues as the root continues to grow into the soil. The cells in the root cap are specialized for several different things. First, they can sense gravity, which is why roots grow down. Second, they secrete a slimy substance that helps roots move through the soil. Directly behind the root cap is the root meristem, which is where cell division occurs. This means that when the root grows, the new cells come from the root meristem. We will look at the structure more another time. Let's go back to our diagram and label the root cap. You can see that it is found at the bottom tip of the root, and forms a protective barrier between the soil and the rest of the root. This small area here is the root meristem where cell division for new growth occurs. We've talked about the epidermis when we looked at the structure of stems and leaves. Roots also have this protective outer layer known as the epidermis. Remember that dermis means skin and epi means outer. This means that the epidermis is literally the outer skin. While the root cap is found at the bottom of the root, the epidermis is found throughout the length of the root. At the bottom where the root cap protects the root, the epidermis is just inside that hard layer. A good way to relate the epidermis to something more familiar is to think of carrots and potatoes. When you peel carrots and potatoes, you are actually removing the epidermis. The epidermis can produce root hairs, which are the main site of water and nutrient absorption. Root hairs allow for greater surface area so that plants can take in more vital substances. While root hairs are very important, they only live for a few days. This means that the epidermis must continually produce new root hairs. In some plants, there are even specific types of fungi and bacteria that help with absorption. Let's go back to our diagram to label these parts. The epidermis is the outer protective layer throughout the length of the root. At the bottom, where we already have the root cap, the epidermis is just inside. Sticking out from the epidermis are a few root hairs. We don't need to label all of them, but do note that there are many root hairs. As previously mentioned, there are several functions of roots. These include anchoring and supporting the plant, absorbing and moving water and minerals, and storing the products of photosynthesis. The primary root tissue is responsible for storing food, which is one of the products of photosynthesis. This layer is sometimes called the cortex. This layer is found after the epidermis and often makes up the bulk of the root. This is especially the case in some plants such as carrots or radishes. After you peel a carrot, there is a great crunchy snack. The majority of what you are eating when snacking on carrots is the cortex. The end of the cortex in the root is distinguished by the endodermis. We already know that dermis means skin, so let's consider the prefix endo. Endo means inner, so the endodermis is the inner skin. In roots, this structure contains fatty substances that block water. This means that water will not pass from the inner layers to the cortex, and that water in the cortex will not move into the inner layers. Let's go back and label these structures on our diagram. We can see that directly behind the epidermis is the cortex. This large food storage part of the root is surrounded on the inner side by the endodermis here. Much like in the plant stem, the vascular cylinder in the root contains xylem and phloem. Xylem moves water and dissolved minerals within a plant, while phloem moves food throughout a plant. The xylem generally moves water up from the roots, while the phloem moves food from the leaves to the roots. In young roots, the vascular cylinder is in the center. We can see this in our root diagram. Both the xylem and the phloem can be found here. However, in more developed roots, the structure of the vascular bundle may differ. You may remember that there are two types of flowering plants, monocots and dicots. Monocots are flowering plants with one seed leaf, while dicots are flowering plants with two seed leaves. We have been over differences in characteristics in the shoot, or above-ground system, but haven't looked at the root. In monocot roots, the xylem and phloem are arranged in a ring, much like in the stem. The phloem is on the outside of the ring and the xylem is on the inside of the ring. The ring surrounds the pith in the center. We can see this ring here. Note the phloem, xylem, and pith. In dicot roots, the xylem and phloem are arranged in a star shape. As it is in monocots, the xylem is on the inside of the star and the phloem is on the outside of the star. We can see this arrangement here. Note the overall star shape as well as the phloem and xylem. Roots serve many important roles in order to help plants survive. The root hairs help absorb water and nutrients from the soil, while the cortex stores the products of photosynthesis such as sugars. The tip of the root is protected by the root cap, and the epidermis provides an outer layer of protection for the rest of the root. In order to move water and food within the root and to the rest of the plant, the vascular bundle contains xylem for the water and phloem for the food. All of these parts must work together regardless of the type of root. As plants grow above the surface, there is also growth that occurs within the soil. Roots need to grow in order to better support the plant and to better absorb both water and nutrients for the growing plant. We will review some structures of the root in addition to looking at how roots grow. You can think of root growth as a construction site. The root is creating new cells in order to expand and help the plant further develop. When at a construction site, people are required to wear hard hats in order to protect their heads. In roots, the root cap serves a similar purpose. This outer area of the bottom of the root protects other root tissues as the root continues to grow into the soil. The cells in the root cap are specialized for several different things. First, they can sense gravity, which is why roots grow down. Second, they secrete a slimy substance that helps roots move through the soil. We can see the root cap here in this diagram. Directly behind the root cap is the root meristem, which is where cell division occurs. This means that when the root grows, the new cells come from the root meristem. Remember that the root cap serves to protect the root and therefore protect this area of new growth. There are three main areas involved in the growth of primary roots. The new cells start in the meristematic region, which is the location of cell division. This is the first area right behind the root cap and kind of looks like an inverted cone. We can see that the meristematic region is here on our diagram. This is a very active region of the root, and the cells in this area divide about every 12 to 36 hours. The cells formed here eventually create the three tissues needed for primary root growth: protoderm, procambium, and ground meristem. The protoderm will eventually become the epidermis or skin of the root. The procambium will eventually become the vascular tissue. Remember that there are two types of vascular tissue, xylem to move water and phloem to move food. The procambium will produce cells that make both types of tissues. The third type of cell created in the meristematic region is the ground meristem, which will become ground tissue and help with storage of water and nutrients for the plant. The next region involved in primary root growth is the elongation region. This is the area of root lengthening. The cells that were produced in the meristematic region grow in the elongation region. No new cells are produced here, but this is the area that actually creates the growth of the root. We can see in this diagram that the cells in this area actually do look longer than those in the meristematic region. The third region involved in primary root growth is the maturation region. As the name implies, this is the area of cell maturation. It is also where the cells that grew in the elongation region fully develop and become adult cells. The three different primary cells that were created in the meristematic region fully develop into the cells they were designed to become. The protoderm cells become the epidermis, which is like the root skin. The epidermis cells move to the outer layers of the root in order to provide protection. The procambium cells either become xylem to carry water or phloem to carry food. Lastly, the ground meristem cells become the ground tissue of the root, which is basically all the other cells, such as those found in the cortex of the root. Much like some plants have primary and secondary growth, roots may also have more than just primary growth. Lateral root growth occurs after primary root growth and allows for increases in surface area to improve absorption and for support of the plant. Lateral roots start to develop in the pericycle, which is the outermost cell layer in the vascular cylinder. Remember that the xylem and phloem make up the vascular cylinder and that it is found in the center of the root. We can see the pericycle in this diagram. Notice that it is the outer layer of the vascular cylinder in the middle of the root. Because lateral roots start at the vascular cylinder, they are able to contain vascular tissue. As the lateral root develops, it must push through the cortex and epidermis in order to reach the outside of the root. Also, the lateral roots all must remain connected to the vascular cylinder. If they're not connected to the vascular cylinder, they cannot move water and nutrients. We can see this connection to the vascular cylinder in this picture. Note that there are several lateral roots growing in this section of the root, but that they are all connected to the xylem and phloem in order to move food and water. Root growth is vital to the survival of plants. Both primary and lateral roots can be produced. In order to protect the growing cells, the root cap is at the tip of the root. Behind the root cap are three regions involved in primary root growth, including areas for cell division, growth, and maturation. The meristematic region is the location of cell division, which means this is where new cells are made. The new cells then move into the elongation region in order to increase in size. They lastly move into the maturation region, where the three different primary cells that were created in the meristematic region fully develop into the cells they were designed to become. The protoderm cells become the epidermis or skin of the root. The procambium cells either become xylem to carry water or phloem to carry food, and the ground meristem cells become the ground tissue of the root, which is basically all the other cells such as those found in the cortex of the root. In lateral root growth, cells on the outer layer of the vascular bundle push through the ground tissue and epidermis in order to reach the outside of the root. These lateral roots stay connected to the xylem and phloem. Both primary and lateral root growth help increase the absorption of water and nutrients for the plant. When you hear the word hormones, you may think of typical human hormones such as testosterone, estrogen, or even adrenaline. These hormones in our bodies regulate different physiological activities, ranging from the tone of our voices, to our height, to our fight or flight response. Plants, which seem rather unresponsive most of the time, also have hormones to control physiological activities. Hormones are chemical signals that coordinate the different parts of an organism. The word hormone comes from a Greek term that means to excite. These chemicals are produced in very small amounts in one area of an organism and are then sent to another part where a response is triggered. Let's use the human hormone adrenaline to illustrate this. Our adrenal glands are located above our kidneys. They produce a hormone called adrenaline that, when needed, is transported through our bodies to cause an increase in heart rate, dilation of pupils, and other responses to make us more alert. Hormones in plants act in similar ways. They're produced by cells in one area of the plant, such as the leaves, stems, or root, and then transported to a different area of the plant in order to produce a response. While the 5 major plant hormones we will look at sometimes have countless functions, we will focus on the main responses they trigger. Auxins are hormones produced in immature parts of plants that stimulate growth. Auxins were the first plant hormones discovered and have been studied extensively. Auxins are most commonly found in seed embryos, apical meristems, and young leaves. The seed embryo has yet to develop, and the cells have not yet become differentiated. That is, the young cells don't know what they'll be when they grow up yet. Similarly, the apical meristem is the location of primary plant growth and contains new cells that have not yet decided what to become. Auxins stimulate cell differentiation, which means this hormone helps decide if a cell will become ground tissue, or vascular tissue, or protective tissue. Auxins also help stimulate stem elongation, which is what primary stem growth is all about. Primary stem growth will occur when a high enough amount of auxins target a given area. Auxins also help regulate fruit development. Without auxins, fruits are often too small. Some produce farmers will spray artificial auxins on plants, such as apples or pears, in order to increase the size of the fruits. Cytokinins are hormones that are produced in the roots, stimulate growth, and have anti-aging effects. Because they're produced in roots, cytokinins must travel up through the plant's xylem in order to reach the target areas, such as the stems and leaves. Cytokinins have several responsibilities, including working with auxins to stimulate growth and cell differentiation in both stems and roots. Cytokinins also specifically promote growth and development of chloroplasts, the cell organelle responsible for photosynthesis. A unique role of cytokinins is to produce anti-aging effects on some plant parts. When you think of anti-aging products, you may think of expensive face creams that advertise giving a younger, brighter look. Cytokinins actually provide a younger, healthier look in plants. Florists commonly use cytokinins to make cut flowers look fresh for longer. By adding this hormone to cut flowers, florists are able to slow down the aging process, providing us with prettier flowers for longer. Gibberellins are hormones produced in meristems of stems and roots that help regulate stem elongation. This group of hormones is actually named after a fungus. The fungus caused some rice plants to grow unbelievably tall, even to the point where the rice plants would fall over because the stem was too tall for the roots to support. We now know that gibberellins help stimulate stem cell elongation in plants. The fungus in the rice plants caused too much production of gibberellins and, therefore, too much stem growth. Some dwarf plant species don't produce enough gibberellins to make the stem elongate, causing the short stature. However, gibberellins can be added to dwarf plants in order to make them grow to normal heights. Ethylene is a hormone produced in fruits, flowers, and aging leaves that promotes fruit ripening. There are a few other effects of ethylene, but these vary depending on the type of plant. Sometimes ethylene will promote the growth and development of roots, leaves, and flowers. However, in other species of plants, ethylene inhibits this growth. No matter the type of plant, ethylene is best known for promoting the ripening of fruit. You may have placed an apple with unripe avocados in a brown paper bag in order to make them ripen faster. The reason this works is because you trap ethylene gas from the already ripe apple in the bag, making the avocados ripen. Ethylene is also commercially used to ripen fruits. For example, many tomatoes are harvested when they're still green rather than the more desirable red. However, before being placed in your grocery store, these unripe tomatoes are exposed to ethylene in order to make them ready for sale. The last hormone we will look at is called abscisic acid, which is a hormone produced in leaves, stems, and roots that has the opposite effects of other hormones. Abscisic acid is also called ABA. It most notably inhibits growth. It also promotes seed dormancy, meaning it keeps seeds dormant rather than encouraging them to grow and develop. This is useful for long-term survival. Another useful purpose of ABA is to close the stomata, which release water from leaves in stressful times such as during droughts. Whether it is in humans or plants, hormones help regulate physiological activities. Hormones are chemical signals that coordinate the different parts of an organism. Remember that only a very small amount of these chemicals is needed in order to cause a response. We looked at the 5 major types of hormones in plants: auxins, cytokinins, gibberellins, ethylene, and abscisic acid. Auxins are hormones that stimulate growth and are produced in immature parts of plants. They were the first group of hormones studied in plants. Cytokinins are chemicals produced in the roots which stimulate growth and have anti-aging effects. Florists often use cytokinins to keep cut flowers alive and beautiful. Gibberellins are hormones produced in meristems of stems and roots that help regulate stem elongation. Dwarf plants often do not contain much of this hormone and are therefore shorter than normal. Ethylene is a chemical produced in fruits, flowers, and aging leaves that promotes fruit ripening. Lastly, we looked at abscisic acid, also called ABA, which is a hormone produced in leaves, stems, and roots that has the opposite effects of the other hormones. All of these chemicals help regulate vital physiological activities in plants.
When you hear a loud noise, you may respond by jumping. External stimuli result in a response. We easily see this in animals, but it also happens in plants. We generally think of plants as passive. They simply, and much of the time, very slowly grow. However, there is much more to plants than we perceive. When animals, including humans, respond to stimulus, which is something that causes a response, we call it a reaction. For example, if someone touches your arm, you generally turn to react. In plants, the response to a stimulus is known as a tropism. This plant movement towards or away from a stimulus can come in many forms. Before we look at a few, let's better understand the word tropism. Like many words in science, tropism comes from a Greek word. Tropos means to turn. Therefore, a tropism is a turn towards or away from a stimulus. When the movement is towards the stimulus, it's called positive tropism. Likewise, when the movement is away from the stimulus, it's called negative tropism. While there are several forms of tropism, we'll just focus on 3 key types: phototropism, geotropism, and thigmotropism. Phototropism is generally the tropism that makes the most sense. We know that plants grow towards the sun, so they can make food through photosynthesis. This movement in response to sunlight is called phototropism. Let's break this word apart. Tropos means to turn, and photo means light. Therefore, phototropism is a turn towards or away from light. While most phototropism involves plants just growing towards the sun, some plants actually follow the sun throughout the day. For example, young sunflowers will orient their flowers to the sun. In the morning, they point east towards the rising sun. They then gradually follow the sun throughout the day, eventually pointing west towards the setting sun. Let's now look at geotropism. We know that tropos means to turn, so we already understand the second half of the word. Geo may sound familiar because of words such as geography and geology. Geo means earth. Therefore, geotropism is movement towards or away from the earth. You may see geotropism used interchangeably with gravitropism, which is the movement towards or away from gravity. Examples of geotropism include the downward growth of roots and the upward growth of shoots. Roots have specialized cells that sense gravity and therefore have a positive tropism by growing towards the pull of gravity. Opposite of this is the growth of the shoot, which is the above-ground portion of the plant. The shoot exhibits negative geotropism by growing away from the earth. You can see these growth patterns with lateral roots and branches. Lateral roots will curve down, while lateral branches will curve up. The last type of tropism we will look at is thigmotropism. Thigma means touch, and again, tropos means to turn. Therefore, thigmotropism is the movement towards or away from touch. Plants can be affected by touch, both positively and negatively. For example, if you repeatedly touch a young growing plant, this can actually cause the plant to grow less. This is useful for plants growing close together so that they don't outgrow the limited space. We can also see a response to touch in vines and other climbing plants. Vines will grow straight along the ground until they touch something to grow on, such as a tree or a wall. This positive thigmotropism allows vines to find a support on which to grow. Another example of positive thigmotropism can be seen in the Venus flytrap. These plants will close leaves around unsuspecting insects, which are then digested for nutrients. An example of negative thigmotropism is seen in a plant found in Central and South America. The touch-me-not immediately folds its leaves after being touched. It's moving away from the stimulus of touch. Regardless of the perception that plants are not the most interesting and animated things out there, they actually have several unique ways to respond to their environments. Geotropism allows plants to correctly orient themselves for growth. The roots exhibit positive geotropism and grow towards the earth. The shoot shows negative geotropism by growing away from the earth. Phototropism occurs when plants move towards or away from sunlight. Lastly, we looked at thigmotropism. This response to touch can be positive, like when vines grow around a tree or wall that they touched. However, it can also be negative, like the folding of leaves seen in the touch-me-not. All of these responses to stimuli allow plants to thrive in their environments.
Leaves are vital to the survival of plants. They help plants in a variety of ways, including producing food and oxygen through photosynthesis, balancing water loss, regulating gas exchange, and transporting the products of photosynthesis. Because of the vast array of jobs that leaves perform, there are many specialized structures. We will look at these parts of the leaf and relate them to their functions. Remember that the shoot system contains the above-ground parts of plants, including the stem, flowers, and leaves. We already talked about the function of the stem, which is to provide support and transport for the plant, as well as the structures involved in these functions. We also looked at how the stem grows. It is important to know that leaves originate at the apical meristem and are a result of primary growth. While leaves do come in a variety of forms depending on the type of plant, we will focus on the structures of dicot plant leaves as these are the most complex. Before we get into the layers and functions of the dicot leaf, let's first take a look at a diagram. We will work our way from the top of the leaf down to the bottom of the leaf and look back at this diagram as we cover each layer. Just like our skin helps protect us, leaves have an outer layer that protects them. This outermost layer is called the cuticle. It is generally waxy to protect the leaf and prevent water loss. When you touch a leaf, you may feel this waxy coating, and on some plants, such as holly, you may actually be able to see the waxy coat shine a bit. Below the cuticle is the epidermis. On the top of the leaf, this is known as the upper epidermis. This is a single layer of cells found directly below the cuticle. It helps protect the leaf by aiding in preventing water loss and providing an extra layer between the outside and inside of the leaf. Now that we've looked at the first two layers of the leaf, let's add these structures on our diagram. The top waxy layer here is the cuticle, and the layer right below that is the upper epidermis. Below the upper epidermis is the palisade layer. This is one or a few layers of cylindrical cells that contain many chloroplasts. Chloroplasts are an important part of plant cells because they are cell structures that allow for photosynthesis. You may remember that photosynthesis is the process by which autotrophs convert light energy into chemical energy. The palisade layer is therefore mainly responsible for producing food and oxygen for the plant through photosynthesis. Let's go back to our leaf diagram and add in the palisade layer. Remember that it is right below the upper epidermis and contains cells that are cylindrical. Below the palisade layer is the spongy layer. This is an area with loosely packed cells with many air pockets. The cells are more ball-shaped than cylindrical shaped like the cells in the palisade layer. The large air pockets allow for gas exchange between different areas of the leaf. The cells in this layer contain few chloroplasts and are therefore not generally responsible for photosynthesis. This makes sense as the layer is further into the leaf and will therefore not get as much sun as the palisade layer, which is closer to the leaf surface. Within the spongy layer are a few other components of the leaf. Along with the air pockets for the exchange of gases are vascular bundles that contain xylem and phloem. These vascular bundles are also called veins in a leaf. You may be able to see the veins in a leaf such as a maple leaf. This is where the movement of water and food occur. Xylem moves water and dissolved minerals, while phloem moves food. Let's go ahead and label the spongy layer and the vascular bundle on our diagram. The spongy layer is the more open space found directly below the palisade layer. Within the spongy layer, you can see the air pockets as well as the vascular bundle containing the xylem and phloem. Collectively, the area that contains the palisade and spongy layers is known as the mesophyll. We can also label this on our leaf diagram. Remember that the top of the leaf has the protective coating of the cuticle and then a layer of cells known as the upper epidermis. On the bottom of the leaf is another protective layer of cells. Because it is at the bottom of the leaf, this layer is known as the lower epidermis. Within this layer are very important structures known as stomata. These are pores in the leaf that allow for gas exchange. A way to remember this is that the root word stoma means mouth in Greek. This mouth can open and close to allow the exchange of gases. On either side of the stomata are guard cells that control the opening and closing of stomata. Gas exchange is needed for both photosynthesis and cellular respiration. We already went over photosynthesis, which converts light energy into chemical energy, so let's quickly go over cellular respiration. Cellular respiration is the process in which oxygen is used to convert organic molecules to carbon dioxide and water and provide energy for the cell. In addition to regulating gases such as oxygen and carbon dioxide that are involved in cellular respiration and photosynthesis, the stomata also control the loss of water. When plants release water, it is known as transpiration. Transpiration in plants is similar to perspiration in people. This release of water vapor in plants is regulated depending on the needs of the plant as well as the conditions of the surrounding environment. The guard cells will open or close the stomata based on these needs. For example, if the weather is hot and the plant is likely to lose water, the guard cells will ensure that the stomata are closed. However, when the air is cooler and more humid, the guard cells will allow the stomata to open. Let's go back and add these last few parts to our diagram. We know that the bottom of the leaf is the lower epidermis. Within this layer, we can see the stomata, which are the openings. These are surrounded by two guard cells that regulate the opening and closing of the stomata. All parts of the leaf help support the life of the plant, which includes producing food and oxygen through photosynthesis, balancing water loss, regulating gas exchange, and transporting the products of photosynthesis. The cuticle and upper epidermis provide protection for the plant. Below that is the palisade layer, which is the location of photosynthesis within the leaf. Below the palisade layer is the spongy layer, which contains cells that are more spread out, allowing for air pockets. Within the spongy layer are vascular bundles that contain xylem and phloem, allowing for the transport of products of photosynthesis. Together, the palisade and spongy layers are known as the mesophyll. The bottom of the leaf is protected by the lower epidermis. Within this are the stomata, which are the pores allowing for gas exchange that are regulated by the guard cells. All parts of the leaf must work together to allow for the plant to go through both photosynthesis and cellular respiration. Angiosperms are flowering vascular plants. They are the most common type of plant and make up over 90% of the plant species on Earth. The name angiosperm comes from two Greek words, angio, which means container, and sperm, which means seed. When we combine the terms, we get seed container, which is a very good description of angiosperms. While angiosperms are best known as flowering plants, they are also known for the protective container, or fruit, which surrounds the seed. Examples of angiosperms range from small wildflowers to large oak trees. Angiosperms can either be unisexual or bisexual, meaning that some flowering plants contain only male or female structures, while others contain both male and female structures. We already know that many plants go through a life cycle that alternates between diploid and haploid, but let's review some basic aspects of this before looking at how angiosperms, or flowering plants, go through this process. Alternation of generations is a life cycle that includes both diploid and haploid multicellular stages. Remember that diploid means two sets of chromosomes and is commonly abbreviated as 2N, where the N stands for chromosomes. In diploid cells, one copy of the chromosome comes from each parent. For example, in humans, you get one copy of chromosomes from your dad and one copy of chromosomes from your mom. The same idea is found in plants. Each diploid cell contains one copy of chromosomes from the male parent and one copy of chromosomes from the female parent. Haploid means one set of chromosomes and is commonly abbreviated as N because there is only one copy of the chromosomes. Let's look at a diagram of the basic idea of alternation of generations. We can see in this diagram that the life cycle is broken into N on the top and 2N on the bottom. Remember that the gametophyte contains haploid cells and that the sporophyte contains diploid cells. Previously, we also used the garb lengths to illustrate the oddity of this. Remember that the diploid or 2N organism looks like this. However, when it moves into the haploid stage, the garb length looks completely different. Two of these haploid organisms will get together and mate in order to produce a new 2N organism that looks similar to our first garb lengths. This garb length will then eventually produce a new haploid organism and so on. Now that we have reviewed the basic aspects of this life cycle, let's look at how angiosperms go through the cycle. We will start with the haploid stage as we did previously and then move into the dominant diploid stage. As we move through the different stages in this alternation of generations, we will refer back to this diagram illustrating how angiosperms reproduce. The first structures in the haploid stage are the microspore and the megaspore. The microspore will develop into the male gametophyte and is much smaller than the female spore. The male gametophyte in angiosperms is the pollen and develops in the anther of the flower. You may remember from learning about the structure of flowers that anthers are the top part of the male stamens. We can see the anther in this diagram. The other potential start for the haploid stage is the megaspore, which will develop into the female gametophyte and is much larger than the male spore. The megaspores will eventually become the multicellular female gametophyte, which is the ovary. Again, you may remember from learning about plant parts that the ovary is part of the female structures in the flower. We can see the ovary here in this diagram. Once the gametophytes have fully developed, they will produce gametes. The male gamete is sperm and the female gamete is an egg. The mature male gametophyte, the pollen grain, develops the sperm while the mature female gametophyte, the ovary, develops the egg. Pollination will occur when the pollen grain lands on the female structures in the flower. However, until fertilization occurs, the union of the egg and sperm, the cells remain in the haploid stage. As with most plant life cycles, the diploid stage starts once an egg has been fertilized by a sperm. However, in angiosperms, there is a twist to this normal fertilization known as double fertilization. Rather than just containing one sperm cell, each pollen grain contains two sperm cells. One of these haploid sperm cells fertilizes the haploid egg, making a diploid embryo. However, the other sperm cell fuses with a part of the female gametophyte, which has two nuclei. This combination of the haploid sperm cell with the double nucleus creates a cell that actually has 3 copies of genetic information. This 3N structure goes through mitosis in order to develop into the endosperm. The endosperm is a great food source for the developing embryo. The developing embryo will use this source of food and eventually develop into a mature sporophyte. The mature sporophyte will grow and eventually produce structures for reproduction, namely flowers. Remember that some angiosperms will contain either male or female parts, while others will contain both. Regardless of this, the dominant diploid stage of the life cycle will produce flowers, which are specialized structures for reproduction. The female parts of the flower will produce megaspores, and the male parts will develop microspores, continuing the cycle. Angiosperms are unique plants because they produce protected seeds. This alternation of generations in flowering plants, such as oak trees and wildflowers, means that there are multicellular stages that are haploid and diploid. We first looked at the haploid stage, which starts as either a male or a female spore that develops into a gametophyte. The male microgametophyte is contained in pollen grains and the female megagametophyte is found in the ovary. Eventually, the pollen grains will land on the female flower structures and fertilize the egg. Remember that in angiosperms, a process known as double fertilization occurs. In this, one sperm cell fertilizes the egg, making a new diploid embryo that will eventually develop into a seed and then a mature sporophyte. However, a second sperm cell fuses with a double nuclei cell, creating a cell that now contains 3 copies of genetic information. This endosperm becomes the food source for the developing embryo. Once mature, the sporophyte will eventually produce flowers in order to continue the process of sexual reproduction via alternation of generations.
I don't know about you, but I love fruit. Sweet, delicious fruit, though, is actually ripened plant ovaries or female reproductive organs. The seeds inside the fruits are designed to spread throughout the environment and grow into new plants in a process called seed germination. So, if you're a fan of ripened plant ovaries too, check out this lesson to learn how they grow from tasty snacks into full-grown plants. Plants reproduce using eggs and sperm just like people. However, they don't have physical sex. Male plants release pollen, which fertilize ovaries. The fertilized ovaries develop into seeds, which can then be distributed to make new plants. As the fertilized ovaries develop, a surplus of carbohydrates and proteins are stored inside the seed. This will serve as food for the plant until it can develop leaves. Next, the fertilized plant ovaries develop the hardened coat we see as the outside of seeds to protect them against harsh environmental conditions. The seeds then can be carried away by wind or animals to create new plants. After the seed has made it to a new location and is covered with dirt, it can begin germination. Germination is the process of seeds developing into new plants. First, environmental conditions must trigger the seed to grow. Usually this is determined by how deep the seed is planted, water availability, and temperature. When water is plentiful, the seed fills with water in a process called imbibition. The water activates special proteins called enzymes that begin the process of seed growth. First, the seed grows a root to access water underground. Next, the shoots or growth above ground begin to appear. The seed sends a shoot towards the surface where it will grow leaves to harvest energy from the sun. The leaves continue to grow towards the light source in a process called photomorphogenesis. Several factors influence if and how seeds germinate. The most important factors are water availability, temperature, and sunlight. Water is crucial to seed germination. The seed must go through imbibition to activate root growth. However, too much water can be a bad thing, as most gardeners know. When a plant is still growing underground during root formation, it cannot use the sun to make food like most grown plants do. It must rely on the stored food inside the seed and oxygen from the environment to make energy. If the soil is too soggy, there will not be enough oxygen and the plant will not thrive. Think about a person being kept underwater. We wouldn't last too long. Temperature is also an important factor. Some seeds germinate when it is cold, such as plants in northern environments. Other seeds only germinate when the weather reaches spring temperatures, which is why we see so much plant growth in the spring and temperate climates. Other seeds only germinate after extreme temperatures, such as after a fire in the grasslands. Sunlight is also needed to stimulate plant growth. Plants go through phototropism or movement depending on light. The shoots of a plant grow towards light. This is why when germinating seeds indoors for a garden, an overhead lamp is necessary to keep the shoots growing straight. Otherwise, the shoots tend to turn towards the windows where the sun is. Seeds are formed and germinate as a natural process of plants in the wild. However, humans can take advantage of this process to produce food, such as our tasty fruits, vegetables, and other needed products like cotton or fresh flowers. Humans can grow crops outside, as in traditional farming, or crops can be started inside from seeds in a greenhouse. Seeds are planted in small containers of soil and given the right conditions based on the type of seed. Check out this indoor grow system. Plants are germinated in small cups of soil and water is provided through a plumbing system. The lights overhead ensure that the seed shoots grow directly upward. In summary, seed germination is the process of a fertilized plant ovary or seed developing into a mature plant. Seed germination starts with imbibition. when the seed takes in water from the soil. This triggers root growth to allow the seed to get more water. Then the shoots, or the above ground part of the plant, develop and grow towards the sun above ground. After the shoots reach the ground, leaves form, allowing the plant to harvest energy from the sun. Several factors influence this process, such as water availability, temperature, and sunlight. Seed germination is important for natural plant growth and growing crops for human use.
You may know that plants produce seeds and pollen in order to create new plants. However, this form of reproduction is not the only way to create new plants. When plants make seeds and pollen, they often use a significant amount of time and energy. Additionally, remember that not all plants make seeds. For plants that do not make seeds, there are many options for making new offspring. We will look at a few of these forms of reproduction, but first, let's understand some general aspects of asexual plant reproduction. Asexual reproduction is when offspring are genetically identical to the parent. Asexual reproduction only requires and only allows for one parent. Most of the time we think of two parents, a male and a female, making an offspring. However, asexual reproduction only uses one parent. In plants, asexual reproduction is called vegetative propagation. Vegetative propagation, like many things, has its advantages and disadvantages. It is beneficial for plants that are well suited for their environment and when the environment is stable. Remember that asexual reproduction results in genetically identical organisms, so these organisms must be well adapted to their environment in order to survive. Because asexual reproduction doesn't allow for evolution and adaptations to occur as frequently as sexual reproduction, vegetative propagation is not advantageous for plants that live in changing environments. In unstable environments, plants that are identical to each other may all die out at once. However, if plants are genetically different, which is a result of sexual reproduction, some plants may survive in an unstable environment. For example, if we have a specific type of plant that grows very well in hot, humid environments, all of the plants may die out if there is a change in the climate. If the temperature or humidity change, the plants that produce asexually are all the same and therefore will all die out because they are not adapted to the new climate. However, a plant that produces sexually will have some diversity in the individual plants, and some may survive this change in temperature or humidity. We also see this in plants that are affected by diseases. Those plants that are genetically identical are more likely to die out than those plants that are genetically diverse. Now that we have explored what vegetative propagation is, as well as some benefits and downsides, let's now look at a few forms of asexual reproduction in plants. We will first look at how bulbs result in vegetative propagation. Generally, you may think of bulbs that you plant in the fall in order to get flowers, such as daffodils or tulips, in the spring. Bulbs are underground buds that have fleshy leaves extending from them. Bulbs are food storage units for future developing plants. Bulbs contain several buds near the node, which is where leaves are produced. These new buds can eventually develop into new plants. If you plant one bulb, you may find that years later you have several plants coming from that one bulb. If you want, you can separate these new bulbs and plant more flowers. Because this one plant produces new offspring that are genetically identical, bulbs are a form of vegetative propagation. Tubers are somewhat similar to bulbs. Tubers, such as potatoes, are enlarged, fleshy, underground stems. While potatoes do not always reproduce by using tubers, if you've ever left a potato for too long, you know that new potato plants can develop from the tuber. These new stems start on the eyes of the potato, and if you plant a potato with new growth from the eyes, a potato plant will develop. We can see the stems starting to develop on the eyes of the potato here. You can even see that leaves have started to develop. Let's now look at two ways stems can be involved in vegetative reproduction. First, rhizomes are horizontal underground stems. These horizontal stems will grow and then develop a new vertical stem at certain critical points. Rhizomes are responsible for the growth of many grasses, sedges, and weeds. The rhizomes grow out from the original plant and invade the nearby soil. They then make new flowering stalks. Even though we think of grasses and weeds most often with this type of vegetative propagation, this method of asexual reproduction can even be seen in irises. Irises can reproduce sexually through pollen and seeds, but sometimes we'll use rhizomes to reproduce as well. This is why you may see a patch of irises growing close together. They're all sharing the same root system. The second type of stem growth we will look at uses stolons, which are horizontal, above-ground stems. These horizontal stems grow along the ground and may develop adventitious roots along with new flowering plant stems. The most common example of stolons can be found in strawberries. Strawberries have runners, which are really stolons, that grow along the ground and continue to develop new flowering stems and roots. Again, this form of vegetative propagation can explain why you will see a large number of strawberry plants in the same area. They're all connected by the stolons. So far, we have looked at naturally occurring forms of vegetative propagation. Let's now consider how humans have used this form of plant reproduction. The most common ways that humans manipulate plants asexually is through grafting and cuttings. Grafting involves the union of different individuals to create a new plant. This can be done at different parts of the plant, but is most often done at the plant stem. One plant is inserted into the stem of another. The point of insertion is called the scion. This method of vegetative propagation is responsible for many hybrid plants, including wine grapes and dwarf fruit trees. Certain grapes grow in specific areas and produce distinctive flavors. In order to create plants that will grow in different areas, humans can graft different varietals together. The same idea comes into play for dwarf fruit trees. These trees produce the same fruits that we love, but are easier to harvest. Cuttings are plant fragments used to grow new plants. Not all plants are capable of developing from a plant cutting. However, certain types of plants, such as herbs and common garden flowers, can be grown from cuttings. Typically, a part of the stem or root is cut and then placed into soil in order to grow. This new cutting will eventually establish roots and a stem. Vegetative propagation in plants allows for the creation of new plants from only one individual. This form of asexual reproduction occurs in plants. Some plants will use both asexual and sexual reproduction. We looked at a few common examples of vegetative propagation. First, we looked at bulbs. Bulbs store energy for plants, but can also be used to create new plants. Sometimes when you plant just one bulb, there will be a few plants growing in that specific location a few years later. This is because new bulbs will develop near the node where the leaves grow. These bulbs can then be separated in order to grow more plants. We also looked at tubers such as potatoes. If you've ever left a potato for too long, you know that new plants may grow out of the eyes of the potato. Next, we looked at rhizomes and stolons. Rhizomes are horizontal stems located underground, while stolons are horizontal stems located above ground. Both are used to spread out from the original plant in order to grow new flowering stems along the stretching root. Many grasses and weeds use rhizomes, while strawberries use stolons. Lastly, we looked at how humans have used vegetative propagation to create new plants. Grafting involves taking parts of two different plants and putting them together to create one new individual. This is how many different types of grapes for growing wine have been made. Cuttings are sections of plants, normally roots or stems, that can be planted and will grow into new plants on their own. Regardless of the method of vegetative propagation, remember that all forms result in a genetically identical new plant and that this method of reproduction is most useful in stable environments.
Do you try to warm up when you're cold, cool down when you're hot, eat when you're hungry, and drink when you're thirsty? Congratulations, you're alive. Assuming you're not a brick, rock, or zombie, you try to keep the conditions you're in relatively comfortable. But you have all sorts of complex mechanisms to do so, like your thumbs, muscles, and brain. Plants have to do the same, but without the tools you have. How do they do it? Homeostasis is a property of systems that regulate themselves to a relatively stable state. Simply put, homeostasis is just balance. Plants must balance the heat they gain with the heat they lose. You may have noticed that white things seem cooler to the touch in the hot sun. This is because white reflects sunlight. Some plants in hot climates have silvery or whitish leaves for just this reason. One example is the desert brittle bush, which is native to the southwestern United States. The desert brittlebush's whitish color comes from the fine hairs on its leaf surface, and scientists have determined that these hairs keep the leaves several degrees cooler than they would be without them. Plants in colder climates also have to protect themselves against the cold. For example, deciduous trees drop their leaves in cold weather to reduce their metabolism. A more unusual example is the Arctic poppy, which grows in the direction of the sun to increase the amount of radiative heat it gains. This is called heliotropism. The bowl-shaped Arctic poppy can increase its temperature up to 14 degrees Fahrenheit, which not only speeds up seed production, but also provides an inviting and cozy place for pollinating insects to visit. If you have ever seen a wilted plant, you know that they have to regulate their water use. Most of the water plants lose is through transpiration, where water evaporates through tiny pores called stomata. Plants need stomata because these openings help them take in carbon dioxide for photosynthesis, and because transpiration helps plants pull water up from their roots. But losing too much water through stomata can be a problem if it is dry outside. So plants have mechanisms by which they can close their stomata when the weather is dry. Some desert plants, like cacti, only open their stomata at night. Some groups of cacti have fairly large leaves, but many don't appear to have leaves at all. Cactus leaves are microscopic. Cactus spines are considered modified leaves. Without leaves, cacti have a reduced surface area, which means that they absorb less heat and lose less water. However, this strategy is still somewhat puzzling. For instance, some plants can drop or curl their leaves to reduce surface area when conditions are especially dry. Cacti with reduced or absent leaves do not have that option. Plants in dry climates often have deeper roots, so they can find water that is further underground. Some desert-adapted plants, like cacti, have thick stems that can store water. Do you regulate the ions in your cells? Sure you do, whether you realize it or not. Plants also have to regulate the ions in their cells, such as sodium and potassium. Plants have intracellular mechanisms that help them take up sodium, even though too much sodium will kill a plant. Some plants, like mangroves, are adapted to saline environments and can secrete sodium through the leaves or protect themselves from too much salt with a thick layer of epidermis. Plants can respond to their environments in subtle ways, just like you. Did you know they can even move? When a plant moves by growing toward or away from a stimulus, this is called tropism. Phototropism, for example, is movement towards or away from light, while chemotropism is movement towards or away from a chemical stimulus. When plant movement is non-directional, this is called a nastism. Nastisms can be caused by growth or by changing water pressure in plant cells. If you've ever noticed that some plants close their leaves or flowers at night, you have seen a nastic movement. When a whole organism moves, this is called a taxis. Most plants can't do this, but some algae can. In addition, some plants like ferns have motile sperm. Homeostasis is the property of living organisms in which internal systems are kept in balance. Plants stay cool in the desert heat through their reflective surfaces, reduced leaves, or leaves that are parallel to the sun. In the cold, plants may drop their leaves, insulate themselves with dead leaves and hairs, or follow the sun. Plants can prevent water loss through transpiration, by closing pores called stomata, or by dropping leaves. Desert plants may also have adaptations that help them store water or very deep roots. Plants can take in sodium when it is needed. However, plants that live in saline environments must also have mechanisms to get rid of excess sodium. Tropism occurs when a plant grows towards or away from a stimulus. Nastic movements occur when a plant exhibits random movement in response to a stimulus. Taxis happens when whole organisms, like some algae and the motile sperm in ferns, move.
Have you ever thought about what it means to be a multicellular organism? Probably not, but maybe that's because it seems like an obvious question. I mean, if an organism has more than one cell, it's multicellular. Case closed, right? Well, let's look at an example to see how easy it really is. If we have a colony of bacteria growing in a petri dish, do we define the colony as one multicellular organism, or billions of organisms living all piled on top of each other? Let's start by defining an organism. An organism is defined as an individual living thing that is capable of responding to stimuli, growing, reproducing, and maintaining homeostasis. Because each cell in the bacterial colony can perform all of these essential functions by itself, we define each cell as its own organism. By contrast, a multicellular organism is made up of many cells that are dependent on each other to perform these essential functions as a group. The cells have divided up the labor, and each cell has become specialized to perform a specific task. This specialization is a key characteristic of multicellular organisms. But why specialize? If a cell cannot perform all essential functions on its own, doesn't that limit what it can do? The answer to this question really depends on the level of organization that you're looking at, and there are several levels of organization within an organism. Starting at the cellular level, each cell has its own set of functions and requirements that it must carry out. Most cells form groups called tissues, which are groups of cells that share a common structure and function within an organism. Tissues are the next level of organization in an organism, and are often very specialized to perform only one or maybe a few very specific tasks. For instance, muscle tissue is specialized to contract, and that's about all that it does. The next level of organization above tissues is organs. An organ is a structure composed of several different tissues that work together to perform a certain task. The tissues within an organ work together to perform more complicated tasks than any one tissue is capable of. An example of an organ would be the heart. The heart is mostly muscle tissue, but is also composed of connective tissue, which provides structure for the heart, an epithelial layer that serves as a barrier to keep the blood contained in the chambers, and nervous tissue, which regulates the heart rate. Together, these tissues perform the more complicated task of pumping blood. But we're not done yet. There's another level of organization above organs, and that is the organ system, which is a group of organs and tissues that work together to perform a major function of the organism. Let's go back to our heart example. The heart is one organ within the circulatory system, which is composed of the heart, blood vessels, and blood. The heart pumps blood, but the whole circulatory system working together circulates blood throughout the body. And finally, the last level of organization is the organism itself, which as a whole is capable of all essential functions. So back to the question of whether or not specialized cells are more limited than single-celled organisms. At the cellular level, the specialized cell is limited to performing its specific task and is more limited than a single-celled organism. However, at the organismal level, specialization and division of labor opens up almost limitless possibilities for variation of structures. And it frees multicellular organisms from the limits of single-celled organisms in terms of size and complexity. In this set of lessons, we're going to talk about the anatomy and functions of animals, with a particular emphasis on humans. Let's start by talking about tissues. In a human being, there are dozens of very specific tissues, but they can be classified into four main tissue types: connective tissue, epithelial tissue, muscle tissue, and nervous tissue. In this lesson, we'll take a look at the characteristics and functions of epithelial tissue. We'll talk about connective, muscle, and nervous tissue when we talk about those systems. Epithelial tissue is tissue that is organized into tightly packed sheets of cells that line organs and body cavities. In epithelial tissues, one or more layers of cells completely covers a basement membrane, which is a well-defined layer of extracellular matrix that serves as a base for epithelial tissues. Epithelial tissues can take many forms. They can be as simple as a single layer of very flat cells, like the epithelial cells right here that line this blood vessel, or they can be a very complex mix of cells of different shapes and composition, like all of these epithelial cells of the human skin in this picture here. Epithelial tissues can also serve several functions. One major function of epithelial tissue, and especially skin, is to serve as a barrier to protect against injury, invading organisms, and loss of fluids. But epithelial tissues aren't just limited to serving as barriers. Some epithelial tissues are specialized to absorb nutrients, like the epithelial tissues of the small intestine. You may notice in this picture of the small intestine that we have a well-defined layer of epithelial cells here that sit on a very thin basement membrane that you can barely see here. And underneath you have connective tissue right about here. The epithelial cells come in contact with the contents of the small intestine and are organized into these finger-like structures here. These structures are called villi and are finger-like projections of epithelial tissue which increase surface area. The increased surface area created by the villi allow a greater number of epithelial cells to have access to nutrients in the small intestine, and therefore increases absorption efficiency. Other epithelial tissues like prostate gland epithelium are specialized for secretion of proteins. In this section through the prostate gland, we can see the round blue nuclei of the prostate epithelial cells that line the ducts of the gland. In addition, one of the secretory products of the prostate has been stained brown in this particular section to show which cells are actively secreting proteins into the ducts. And as you can see based on the brown color, pretty much every cell lining the ducts is actively secreting proteins. And finally, some epithelia, like the lining of our respiratory tract, have some cells that secrete mucus, and other cells that have lots of small cilia on their cell surface that move the mucus and trap particles out of the lungs. Here is a picture which shows a section through a ciliated epithelium. In this picture, you can see a very prominent basement membrane here, with several layers of cells on top. And then, in the very top layer, you can see some cells which secrete mucus here, here, and here, and other cells that have visible cilia located here, here, and here. The mucus cells secrete mucus, which is then moved by the cilia cells out of the lung. So let's review. Multicellular organisms were created when cells started to specialize and divide up the labor among themselves. These groups of specialized cells that share a common structure and function are called tissues. In animals, there are four main tissue types: connective tissue, epithelial tissue, muscle tissue, and nervous tissue. Epithelial tissue is tissue that is organized into tightly packed sheets of cells that line organs and body cavities. Epithelial tissues can serve several purposes, with the three major functions being protection, secretion, and absorption. In epithelial tissues, one or more layers of cells completely covers a basement membrane, which is a well-defined layer of extracellular matrix that serves as a base for epithelial tissues. Epithelial tissues can take many forms. They can be as simple as a single layer of very flat cells like the epithelial tissue that lines blood vessels, or they can be a complex mix of cells of different shapes and composition like the epithelial tissue of the human skin. People have skeletal systems made up of over 200 bones. Actually, the exact number in adults is 206 if you were wondering. In this lesson, we'll talk about how these bones and other connective tissues are organized into a skeletal system to give the body its form and protect vital organs. We'll also talk about the muscular system and how the skeletal and muscular systems work together to allow a wide range of movements and physical capabilities in humans. Humans have an endoskeleton, which is located inside the body. Most of the human skeleton is made of bone, which you may recall is composed of mineralized calcium phosphate that is secreted into the extracellular matrix by bone cells called osteocytes. The human skeleton also contains quite a lot of cartilage too. Bones are hard and inflexible, but cartilage is rubbery and flexible, which makes it very useful in parts of the skeleton where movements require flexibility of the skeleton or padding is needed between bones that would otherwise rub against each other. Ligaments made of collagen make flexible connections between bones that keep the bones in their proper places while allowing a wide range of motion. The most basic function of the skeletal system is to support and provide structure for the body. Without our skeletal systems, we wouldn't be able to hold our shape, and we definitely would not be able to stand upright. A second major function of the skeletal system is to provide protection for vital organs like the brain, heart, and lungs, which are protected by the skull and rib cage. The last basic function of the skeletal system is to facilitate movement. Part of this function is a direct result of providing support for the body, but beyond support, bones also function as levers that the muscles can manipulate to run, jump, lift, grab, or basically move in almost any way you can think of. In addition to acting as levers, the positioning of bones and the way that they are attached to each other and to the muscles define the kinds of movements that the body can make. So now that we've talked about the skeletal system and its functions, let's talk about the muscular system and how it contributes to the form and movement of the human body. Muscle tissue is one of the 4 major tissue types found in the human body, and all muscle cells function in the same basic way. They contract, or get shorter and pull whatever they are attached to closer together. Muscles only do work when they contract. They do not actively push things further apart when they stretch back out. Instead, muscle cells are said to relax when they stretch back out, because it is a passive action that does not result in any force being applied by the cell. In contrast, contraction requires energy and results in a force being exerted by the cell. Some muscles in our bodies are under our conscious control and allow us to control our movements. These muscles are said to be voluntary muscle, or muscle that can be contracted consciously by an animal. In contrast, the body also contains muscle that is not under our conscious control. This involuntary muscle is muscle that cannot be consciously contracted by the animal. There are 3 types of muscle cells, 2 that are involuntary and 1 that is voluntary. These 3 muscle types are cardiac muscle, smooth muscle, and skeletal muscle. Cardiac muscle cells are involuntary muscle cells that make up the heart. Cardiac muscle is only found in the heart, and a key difference between cardiac muscle and other muscle types is that cardiac muscle cells pass electrical signals to each other. In this way, when one cardiac muscle cell gets a signal to contract, it passes that signal along to all its neighbors. The electrical signal is quickly passed to all the muscle cells of the heart chamber, and they contract in unison. Smooth muscle cells are involuntary muscle cells that are found throughout the body except for the heart. Unlike cardiac and skeletal muscle, smooth muscle does not have visible stripes or striations when viewed under a microscope. This is actually the reason why it's called smooth muscle. Smooth muscle is found in many places throughout the body, like blood vessel walls, the stomach, the small intestine, the large intestine, and the bladder. Skeletal muscle cells are voluntary striated muscle cells that control body movements. These are the muscles that people are generally most familiar with, because many of them are visible and we actively control them all the time in our everyday lives. An interesting feature of skeletal muscle is that each skeletal muscle cell contains lots of nuclei. This is because each mature skeletal muscle cell is actually formed from lots of immature muscle cells that fuse together to form one large cell. So I mentioned earlier that bones can function as levers that the muscles manipulate to move in almost any way you can think of. I also mentioned that the positioning of bones and the way that they are attached to each other and to muscles define the kinds of movements that the body can make. For example, let's look at a few movements that your arms can make. Hold your hand out in front of you, palm up. Go ahead, we'll leave the screen blank while you do this. Now turn your palm down. Did you see how easily you flipped your hand over by twisting just your forearm? Now try twisting your upper arm the same way. Maybe you can twist it some, but not nearly to the extent that you can twist your forearm. Okay, we're back to showing visuals again. The reason why you can twist your forearm so much easier than your upper arm is because you have 2 long bones in your forearm, the radius and ulna, which can be twisted around each other and allow you to twist your forearm. In contrast, you have only 1 long bone in your upper arm, so there is no second bone to twist around. This limits the amount that you can twist your upper arm. Now let's look at how the contraction and relaxation of different muscles can control movement of your arm at the elbow. Bending of the arm at the elbow is controlled by two opposing muscles, the biceps muscle and the triceps muscle. The biceps muscle is this muscle here in the arm. One end of the biceps muscle is attached by tendons to the top of the shoulder blade underneath the shoulder muscle here. The other end of the biceps muscle is attached by tendons to the inner side of the radius and ulna here. Opposing the biceps muscle is the triceps muscle, which is this muscle right here. The triceps muscle is attached by tendons on one side to the shoulder blade just below the arm, right about here. And on the other side, tendons attach the triceps to the back side of the ulna, right about here near the elbow. When the triceps muscle is relaxed and the biceps muscle contracts, it pulls the radius and ulna closer to the upper arm, and the arm bends at the elbow. In contrast, when the biceps muscle relaxes and the triceps muscle contracts, the ulna is pulled back away from the upper arm and the arm straightens. If both muscles contract at the same time, the two forces cancel each other out, and the muscles flex without moving the arm. Other pairs of opposing muscles are found all over the human body, and each muscle controls a different type of movement, which we can combine into more complex movements like walking or typing on a computer. So let's review. Humans have an endoskeleton, which is located inside the body. The human skeleton is composed mostly of bone and cartilage. Bones are hard and inflexible, but cartilage is rubbery and flexible, which makes it very useful in parts of the skeleton where movements require flexibility of the skeleton or padding is needed between bones that would otherwise rub against each other. Three of the primary functions of the skeleton are to support and provide structure for the body, to provide protection for vital organs, and to facilitate movement. We have three types of muscle in our bodies: cardiac muscle, which is involuntary muscle of the heart; smooth muscle, which is involuntary muscle in the rest of the body; and skeletal muscle, which is voluntary muscle that controls body movement. All three of these muscle types function in basically the same way. They contract, or get shorter and pull whatever they are attached to closer together. Muscles only do work when they contract. They do not actively push things further apart when they stretch back out and are said to be relaxed. By using tendons to attach to bones in specific places, muscles form opposing pairs all over the human body. These muscles use the bones of the skeleton as lever arms and control various specialized movements, which we can combine into more complex movements like walking or typing on a computer.
The trillions of cells in your body require a continuous supply of oxygen and nutrients to carry out their vital functions. But cells can use these substances only when they are in the immediate vicinity. Like a busy factory that delivers its products to various stores, your body must have a transportation system to carry its gases and nutrients to its various cells. This system is called your circulatory system. And in this lesson, you will learn about the different parts of the circulatory system and how these parts help move needed substances throughout your body. To illustrate, let's take a look at Mr. Smith's widget factory. Mr. Smith's factory is a very busy place. His factory runs 24 hours a day, 365 days a year. Mr. Smith's workers must work hard to keep up with the continual demand for his widgets. If his workers quit working, his business would not survive. Even though Mr. Smith's factory is very busy, it is nothing compared to the non-stop activity going on in your body at this very minute. Every second of every day, trillions of cells in your body pick up nutrients and drop off waste. In the same way that Mr. Smith's employees use roads to get his products to and from stores, your body uses blood vessels, which are hollow tubes for carrying blood. The blood found within your blood vessels is a fluid that transports oxygen, nutrients, and cell wastes throughout your body. Keep in mind that these vital substances must make their way to every cell in your body. As you can imagine, this is a very big job. In fact, if laid end to end, all of the blood vessels in the human body would travel around the Earth 2.5 times. It seems hard to believe, but because the blood vessels have to carry blood to so many cells in your body contains roughly 62,000 miles of blood vessels. Mr. Smith uses trucks that run on gasoline to get his widgets where they need to be. Your body does not run on gasoline. Instead, it relies on a pumping station known as your heart, which is a hollow organ that pumps blood through the blood vessels. Your heart is the ultimate workaholic, and the amount of work it does is almost too hard to believe. In one 24-hour period, your heart pumps your body's complete supply of blood, about 6 quarts, through the blood vessels over 1,000 times. Good thing your heart does this for you without your conscious input, or you wouldn't get much else done. The heart, along with the miles of blood vessels found throughout your body, make up your circulatory system, which is defined as a closed system of blood vessels for the transport of gases and nutrients. Back at Mr. Smith's factory, we see that his truck drivers have returned from making their deliveries. Now is a good time for us to take a closer look inside Mr. Smith's factory to see just how he runs such an efficient operation. Mr. Smith's factory is divided into 2 separate departments. So distinct are the goals of the 2 departments that it's easier for us to think of Mr. Smith's factory as 2 factories in one. The factory on the right is responsible for piecing together the widgets. In the right factory, widgets pick up their needed parts and unneeded parts are discarded. The factory on the left takes the freshly made widgets and transports them all over the country. Just as it is easier for us to think of Mr. Smith's factory as 2 factories in one, it is easier for us to think of the circulatory system as 2 circuits in one. Because we learn about the body in anatomical position, we look at this picture of the heart as if it is inside a person who is facing us. Therefore, the right side of the heart is here, seen in blue, and the left side is here, seen in red. Like the right side of Mr. Smith's factory, the circulatory system must have a way of picking up and discarding substances within the blood. Instead of assembling widgets, the right side of the heart receives deoxygenated blood from the body and sends it through the pulmonary circuit, which carries blood to the lungs to pick up oxygen and release carbon dioxide. It is in the lungs where carbon dioxide from the returning blood is dropped off and fresh oxygen is picked up. The circulation from the right side of the heart to the lungs and back to the heart completes the pulmonary circuit. We remember from Mr. Smith's factory that the widgets that were assembled in the right factory are then taken by the left factory and sent out for distribution. Instead of distributing widgets, the left side of your heart receives freshly oxygenated blood from the lungs and sends it through the systemic circuit, which carries blood to the cells of the body to distribute oxygen and nutrients and pick up cell wastes. The circulation from the left side of the heart to the body cells and back to the heart completes the systemic circuit. Let's review. Your body is a very busy place. Every second of every day, trillions of cells in your body pick up nutrients and drop off wastes. The blood is a fluid that transports oxygen, nutrients, and cell waste throughout the body. Blood travels through blood vessels, which are hollow tubes to carry blood. And blood is propelled by your heart, which is a hollow organ that pumps blood through the blood vessels. The heart, blood vessels, and blood are the parts that make up the circulatory system, which is defined as a closed system of blood vessels for the transport of gases and nutrients. The circulatory system can be thought of as 2 circuits in one. The right side of the heart is the pulmonary circuit, and the left side is the systemic circuit. The job of the pulmonary circuit is to carry blood to the lungs to pick up oxygen and release carbon dioxide. The circulation of blood from the right side of the heart to the lungs and back to the heart completes the pulmonary circuit. Immediately following the pulmonary circuit, blood enters the systemic circuit. The systemic circuit carries blood to the cells of the body to distribute oxygen and nutrients and pick up cell wastes. The circulation from the left side of the heart to the body cells and back to the heart completes the systemic circuit.
Let's talk about the human respiratory system and why we need it. I mean, we're all surrounded by the air, which is about 20% oxygen, so why don't we just absorb it through our skin? The problem is, if we just absorbed oxygen through our skin, it might reach the first 2, maybe 3 layers of cells before it was all used up, and it would never make it all the way to our brain or muscles or most other places in our body. We just don't have enough surface area to supply enough oxygen, and we need an efficient way to get it into the bloodstream so that it can be distributed throughout the body. So, let's start at the beginning. A person breathes air in through their nostrils or their mouth. The mouth and nasal airways come together at a junction called the pharynx, which is located at the back of the mouth and continues down the throat until it reaches the larynx, which is more commonly known as the voice box, and some people call it the Adam's apple. The air then enters the trachea, which is the main airway that runs from the larynx down to the lungs. The trachea is known to most people as the windpipe. Just as the air reaches the lungs, the trachea splits into 2 smaller tubes called bronchi. Each bronchus enters one side of the lungs and then splits repeatedly into ever smaller tubes called bronchioles. And finally, at the ends of the smallest bronchioles are clusters of spherical structures that look a lot like bunches of grapes. These grape-like structures are called alveoli and are basically tiny air sacs with very thin walls which serve as the main site of gas exchange. The average human lung has about 500 million alveoli, with a combined inner surface area that is about the same size as half of a full-size tennis court. That's 40 times the surface area of a person's skin. So how is this even possible? The organization of the alveoli in clusters creates a honeycomb-like internal structure, where the alveolar walls create a massive amount of surface area for the air to come in contact with. In combination with the sheer numbers of the alveoli, they solve our first problem by increasing the surface area available to absorb oxygen through. But how do the lungs take oxygen out of the air and get it into the bloodstream? Well, surrounding the alveoli is a very dense network of capillaries. You may remember that capillaries are very small, flattened blood vessels with very thin walls which allow gases and nutrients to be easily transferred between the blood and surrounding tissues. The blood collects oxygen from the alveoli as it passes over the entire surface area created by the alveolar walls. However, the oxygen can't just magically jump from the air across the alveoli and into the blood. It has to cross each barrier one at a time. First, the oxygen has to be dissolved, so there is a thin film of water coating the inside surface of the alveoli. Because the volume of water is so small, it becomes saturated with oxygen almost instantly, and the oxygen is then free to diffuse across the cell membrane. You may recall that dissolved molecules diffuse quickly in the direction of a lower concentration, which in this case is the oxygen-depleted blood in the surrounding capillaries. So oxygen diffuses from the saturated water coating the alveoli, across the alveolar wall, through the capillary wall, and into the oxygen-depleted blood. Once in the blood, you may also remember that oxygen enters red blood cells and then binds to hemoglobin, taking it out of solution and keeping the dissolved oxygen level low in the blood. This ensures that the oxygen continues to flow from the alveoli and into the blood. As the oxygen is diffusing into the blood, carbon dioxide, which is the primary waste product of animal cells, is being released. Because there is so little carbon dioxide in the air compared to the blood, diffusion of carbon dioxide occurs in the opposite direction to that of oxygen. Carbon dioxide diffuses out of the capillaries, into the alveoli, into the thin layer of water, and then quickly exits into the air. The end result is that oxygen is taken out of the air and enters the bloodstream, while carbon dioxide is removed from the blood and released into the air. It is important that only a very thin layer of water coats the alveoli. If the alveoli begin to fill up with water, 3 problems occur. First, the large volume of water would require much more oxygen in order to reach saturation of the larger volume. Saturating a large volume takes time and slows oxygen transfer. Second, the oxygen and carbon dioxide would have further to travel across the water to reach the alveolar wall. This further slows the transfer of gases to and from the blood. Third, the surface area would be reduced as the surface area of the alveoli become covered by a thick layer of water. The surface area of the water has now become the effective surface area of the lung. For these reasons, excess water in the lungs creates a barrier to oxygen uptake and can cause drowning. So to review, humans need a large surface area for gas exchange and need to easily transfer gases between the blood and the air. Specialized structures in the lungs called alveoli dramatically increase the surface area of the lung, and the dense capillary network around the alveoli create a very short route between the bloodstream and the air. A very thin film of water coating the alveoli allow oxygen and carbon dioxide to instantly transition between dissolved and gaseous forms, thereby facilitating the uptake of oxygen and the release of carbon dioxide. All was calm in the rainforest. A beautiful butterfly lands on a flower in search of nectar, but unbeknownst to the butterfly, a well-concealed predator is only inches away. In a fraction of a second, the chameleon strikes out with its tongue, snatching the butterfly from the flower and drawing it into its mouth. The chameleon begins to chew. Wait, chameleons don't chew, but little boys like Timmy who pretend to be chameleons do. As Timmy chewed the butterfly and physically broke it down into smaller pieces, his salivary glands went to work, releasing saliva containing amylase, which is a digestive enzyme found in saliva that breaks large polysaccharides into smaller polysaccharides. Amylase is capable of breaking every other bond between sugars in a polysaccharide. Between chewing, which physically breaks the butterfly into smaller pieces and creates greater surface area for enzymes to contact, and amylase, which begins breaking complex carbohydrates down into smaller polysaccharides, the digestion of the butterfly has already begun. The tongue then forms the chewed up and saliva-soaked food into a ball and directs it to the back of the mouth, where it enters the pharynx, which you may remember is the junction of the mouth and nasal airways that leads down the throat. However, unlike the air that passes through the larynx on its way to the trachea and lungs, the food must enter the esophagus, which is the tube that serves as a passage for food from the pharynx to the stomach. When Timmy swallows, his larynx, or voice box, moves up, and at the same time a cartilaginous flap called the epiglottis flips downward to cover the larynx, preventing the food from continuing down the airway. Instead, the food is directed into the esophagus, where muscle contractions push the food down the entire length of the esophagus and into the stomach. The stomach is a fairly large organ with an elastic wall that is capable of stretching to make room for over a half a gallon of food and fluid in the average person. The stomach wall is lined by a layer of epithelial cells and has lots of deep pits that lead to the gastric glands. The epithelial cells inside these glands secrete the different components of gastric juice, which are designed to liquefy food and continue the process of chemically breaking it down into smaller components that can be absorbed by the body. One major component of gastric juice is hydrochloric acid, which you may remember is a very strong acid and can be represented by the abbreviation HCl. Because of its acidic properties, hydrochloric acid kills most bacteria found in food. It also does a very good job of dissolving most of the types of tissues that we eat into a liquid form that is much more accessible to digestive enzymes. Unfortunately, hydrochloric acid inactivates amylase, which leaves the polysaccharides only partially digested. Speaking of digestive enzymes, another major component of gastric juice is pepsin, which is a digestive enzyme that breaks proteins down into smaller peptides. You can remember the function of pepsin if you remember that pepsin breaks proteins into peptides. But this function of pepsin raises a fundamental question. How does a cell make an enzyme that breaks down proteins without the enzyme damaging the proteins in the cell itself? The answer is that the cell makes an inactive form of pepsin named pepsinogen, which is several amino acids longer than active pepsin. The extra amino acids block the active site of pepsin, causing it to be inactive inside the cell where it can damage other proteins. However, the hydrochloric acid in the gastric juice removes these amino acids, converting the inactive pepsinogen into active pepsin. once it is out of the cell and in the gastric juice inside the stomach. To keep them from digesting components of the secretory cells, most digestive enzymes are produced in an inactive form known as a zymogen, which is an inactive precursor of an enzyme which requires a change to be activated. In this case, pepsinogen is a zymogen, which is activated by hydrochloric acid and turned into active pepsin. So why doesn't the pepsin and hydrochloric acid digest the epithelial cells that line the stomach? It turns out that most of the epithelial cells of the stomach secrete large amounts of mucus, which forms a barrier to protect the cells against the acid and pepsin. In addition, the stomach isn't always filled with gastric juice. Between meals, the stomach is empty and pretty much inactive. However, when a person sees, smells, or otherwise anticipates eating food, the brain sends a signal to the stomach to start preparing for a meal. As a result of this signal, the epithelial cells secrete a small amount of hydrochloric acid into the stomach, and the smooth muscles of the stomach are also activated and start churning the stomach at a slow rate. This churning of the stomach without food in it is what causes your stomach to growl when it's getting close to mealtime. When food actually enters the stomach, it activates the full response of the stomach. The epithelial cells secrete lots of gastric juice, and the smooth muscle of the stomach is fully activated, causing the stomach to churn and thoroughly mix the food and gastric juice until it is liquefied. The stomach then releases its liquefied contents into the small intestine, a little at a time through the pyloric sphincter, which is the muscular valve at the bottom of the stomach that controls the flow of partially digested food into the small intestine. It takes about 2 to 6 hours for the stomach to completely empty its contents into the small intestine in this manner. During that time, the churning motion and release of gastric juice by the stomach is ramped down until the stomach is empty and essentially powered down. That is, until it receives the next signal from the brain to start powering up again. So let's review. As soon as you put a piece of food in your mouth, your body starts the digestive process. Salivary glands release saliva that contains amylase, which is a digestive enzyme found in saliva that breaks large polysaccharides into smaller polysaccharides. And chewing begins the physical process of breaking large chunks of food into smaller pieces. The food is swallowed and directed by the epiglottis down the esophagus, which is the tube that serves as a passage for food from the pharynx to the stomach. The stomach wall is lined by a layer of epithelial cells and has lots of deep pits that lead to the gastric glands. The epithelial cells of the stomach secrete 3 major products: mucus that protects the epithelial lining of the stomach, hydrochloric acid, and pepsinogen. Pepsinogen is a zymogen, which is an inactive precursor of an enzyme which requires a change to be activated. But hydrochloric acid converts it to the active enzyme pepsin, which is a digestive enzyme that breaks proteins down into smaller peptides. Together, pepsin and hydrochloric acid are the major components of gastric juice, which essentially liquefies food and continues to break it down into smaller components. Between meals, the stomach is empty and pretty much inactive. However, when a person sees, smells, or otherwise anticipates eating food, the brain sends a signal to the stomach to start preparing for a meal. As a result of this signal, the epithelial cells secrete a small amount of hydrochloric acid into the stomach. Smooth muscle is also activated and starts churning the stomach at a low rate. When food actually enters the stomach, the epithelial cells secrete lots of gastric juice, and the smooth muscle of the stomach thoroughly mixes the food and gastric juice until it is liquefied. The stomach then releases its liquefied contents into the small intestine a little at a time through the pyloric sphincter, which is the muscular valve at the bottom of the stomach that controls the flow of partially digested food into the small intestine. The stomach empties and powers down until it receives the next signal to prepare for a meal. When we last left Timmy, He was playing in his backyard, pretending to be his favorite animal, a chameleon. Oh yeah, and he also ate a butterfly. We followed the path that the ingested butterfly took down Timmy's upper gastrointestinal tract, from his mouth to his pharynx, down his esophagus, and through his stomach. Now let's continue on our journey through Timmy's digestive system as we follow the path that the food takes through the lower gastrointestinal tract. We know that liquefied food and gastric juice is released by the pyloric sphincter a little at a time from the stomach into the duodenum, which is the first section of small intestine where digesting food enters from the stomach. A lot of people think that this food is now digested into products that can be absorbed into the bloodstream, but that's not the case at all, because full digestion of the food still requires a lot more work. As soon as the liquefied food and gastric juice enters the duodenum, the acid and partially digested food stimulates the pancreas to secrete bicarbonate, water, and many different digestive enzymes, which flow into the duodenum to mix with the gastric juice. You may remember that bicarbonate is a base, and that bases neutralize acids. So when the bicarbonate secreted by the pancreas mixes with the gastric juice, it neutralizes the acid. Meanwhile, digestive enzymes are secreted from the pancreas as zymogens, which you may remember are inactive precursors of enzymes which require a change to be activated. These zymogens are activated by other enzymes in the duodenum and then start breaking carbohydrates into smaller sugars and proteins into peptides and amino acids. Once the carbohydrate chains have been broken down into monosaccharides, they can be transported across the luminal membrane and into the epithelial cells of the small intestine. Likewise, proteins and peptides can't be absorbed by the small intestine, but once they are broken down into their component amino acids, these are easily transported into the epithelial cells. However, fats are a little more complicated. You see, fats, which are also called lipids, are hydrophobic. They repel water and clump together with other lipids because when they clump together, they have less interaction with the water. But this means that they are insoluble. They won't go into solution, and they aren't available for soluble digestive enzymes to break them down. Fortunately, our liver produces bile salts, which coat the lipids and keep them separated into tiny droplets that don't clump together. These tiny coated droplets give the digestive enzymes enough surface area to gain access to the lipids and break them down. One such digestive enzyme is lipase, which is an enzyme that breaks lipids down into monoglycerides and fatty acids. These monoglycerides and fatty acids can then be absorbed by the small intestine. Although the liver produces bile, it is stored in the gallbladder, which then releases it into the duodenum when digestion is taking place. The small intestine can be broken down into three parts. We've already talked about the first part, the duodenum, which is the very first section of small intestine where the pancreas, liver, and gallbladder add their digestive enzymes, bile, water, and bicarbonate to the digesting food. Following the duodenum are the jejunum and then the ileum. These sections are specialized for the absorption of monosaccharides, amino acids, monoglycerides, fatty acids, and water. The epithelium is structured into large folds that increase surface area and which increases absorption. And sticking out from the folds are finger-like projections called villi that further increase the surface area and absorption. Even the epithelial cells of the villi themselves are covered with tiny finger-like projections called microvilli, which again further increase surface area and absorption. Digesting food is moved through the small intestine by a process called peristalsis, which is the slow rhythmic contraction of the smooth muscle in the intestinal wall. Peristalsis moves digested food through the small intestine, where most of the nutrients and water are absorbed before it enters the large intestine. The large intestine, or colon, functions mainly to reabsorb most of the water that wasn't reabsorbed by the small intestine. Again, peristalsis moves what is left of the food through the large intestine and finally into the rectum, which is the last part of the large intestine where feces are stored before they are eliminated through the anus. So let's review. The duodenum is the first section of small intestine where digesting food enters the stomach. Contrary to popular belief, most digestion occurs in the small intestine and not the stomach. Not only does the duodenum receive digesting food from the stomach, but it also receives water, bicarbonate, and digestive enzymes from the pancreas, and bile from the gallbladder and liver. As the food is moved through the small intestine by peristalsis, carbohydrates are broken down into monosaccharides, proteins are broken down into amino acids, and lipids are broken down into fatty acids and monoglycerides. The monosaccharides, amino acids, fatty acids, and monoglycerides are then transported into the epithelial cells of the jejunum and ileum, which are the absorptive portions of the small intestine. The absorptive surface of the small intestine is arranged into large folds that increase surface area and which increases absorption. Sticking out from the folds are finger-like projections called villi that further increase the surface area and absorption. Following the small intestine is the large intestine, or colon, which functions primarily to reabsorb most of the water that wasn't reabsorbed by the small intestine. Again, peristalsis moves what is left of the food through the large intestine and finally into the rectum, which is the last part of the large intestine where feces are stored before they are eliminated through the anus. Each year, close to 400,000 people in the U.S. undergo dialysis treatment for kidney failure. You may even know someone who has had dialysis treatment to filter their blood in order to remove waste, remove excess fluid, and restore electrolyte balance. These functions of dialysis highlight the major functions of the kidneys, which are fist-sized organs that filter waste products out of the blood and help regulate blood composition and pressure. In this lesson, we'll go through the excretory system and talk about how the liver, kidneys, and bladder work together to remove waste products and toxic substances from the body. All animals have toxins in their body that they must find a way to eliminate. Some toxins are ingested in food, and others are byproducts of metabolism. Some toxins, like carbon dioxide, can be tolerated in small amounts and can be easily disposed of as they are. But some toxins are so dangerous that the body must change them in some way to make them less toxic or find a way to quickly remove them. Detoxifying and modifying dangerous substances so that they can be quickly and easily removed from the body is one of the many jobs of the human liver. Let's look at an example of a toxic substance that the liver modifies to make less toxic. Ammonia is a major byproduct of human metabolism, but it's extremely toxic, so the liver converts it to a less toxic form called urea, which can be created from 2 ammonia molecules and 1 carbon dioxide molecule. 2 ammonia molecules, these NH3s here, combine with 1 CO2 molecule to form 1 urea molecule here, and 1 molecule of water. Of course, then we still have to get rid of the urea, but it's far less toxic than ammonia, so we can concentrate it and store it for several hours without it harming us. So, once the liver has converted the ammonia into much less toxic urea, then what? Well, first the urea is allowed to leave the liver through the bloodstream. The circulating urea will eventually reach the kidneys, where it will be filtered out of the bloodstream and concentrated in the urine. Here's how it works. In the outer layer of the kidneys are structures called glomeruli, which are ball-like structures of the kidney composed of very porous capillaries. You can think of the capillaries in the glomeruli as being like garden hoses with hundreds of holes all over them that let water spray out when the pressure is on. The blood pressure in these capillaries forces large amounts of water and small molecules, including urea, out through the pores. But blood cells and larger molecules that are too big to fit through the pores stay in the bloodstream. Surrounding the glomeruli are the ends of the renal tubules, which are long, looping tubes in the kidney that collect filtrate from the blood and concentrate into urine. The water, nutrients, salts, urea, and other waste products in the filtrate are collected by the surrounding renal tubule and begin a twisted journey. The filtrate travels through the tubule that crosses back and forth between the outer and inner layers of the kidney. As it does, nutrients, water, and sodium chloride are reabsorbed from the renal tubules and returned to the blood. As water is reabsorbed, the urea and other waste products become very concentrated in the tubules and become urine. The whole structure from the glomerulus to the end of the renal tubule is called a nephron and is the basic functional unit of the kidney. By filtering out a large volume of water and small molecules and then selectively allowing most of the water and nutrients to be reabsorbed, the nephrons allow us to get rid of urea and other wastes in high concentrations and conserve water when needed. Sodium chloride and other salts can also be reabsorbed back into the bloodstream as needed, but that function of the kidney is a key component of homeostasis, which is another topic for another day. The urine exits the kidneys through two long tubes called ureters, which carry the urine to the bladder. Repeated contraction and relaxation of muscles in the ureter walls ensure that the urine flows in one direction from the kidneys to the bladder. The bladder itself is a balloon-like muscular organ which is capable of stretching as it fills with urine. As the bladder fills and the walls stretch to create a larger holding capacity, a signal is sent by the nerves of the bladder which gives us the sensation of the need to urinate. The more that the bladder stretches, the more intense this sensation becomes, until the bladder reaches its full capacity and the sensation becomes so overwhelming that it feels like you're gonna burst. Most of the time, people don't let their bladder reach full capacity because it becomes so uncomfortable. Instead, people normally have control over when they empty their bladder by controlling a muscular valve at the exit point of the bladder. When this valve is opened and the bladder muscles contract, the urine enters the urethra, which the urine travels through before it exits the body. So let's review. All animals have toxins in their body that they must find a way to eliminate. Detoxifying and modifying dangerous substances so that they can be quickly and easily removed from the body is accomplished by the human liver. An example of this would be ammonia, which is very toxic, so the liver converts it to urea, which is far less toxic and can be easily removed from the body. The kidneys are the organs that filter waste products out of the blood and help regulate blood composition and pressure. In the outer layer of the kidneys are structures called glomeruli, which are ball-like structures of the kidney composed of very porous capillaries. The blood pressure in these capillaries forces large amounts of water and small molecules, including urea, out through the pores. But blood cells and larger molecules that are too big to fit through the pores stay in the bloodstream. Surrounding the glomeruli are the ends of the renal tubules, which are long, looping tubes in the kidney that collect filtrate from the blood and concentrate into urine. The filtrate travels through the tubules, and as it does, nutrients, water, and sodium chloride are reabsorbed from the renal tubules and returned to the blood. As water and nutrients are reabsorbed, the waste products are concentrated in the tubules and become urine. After the urine is concentrated by the kidneys, it is stored in the bladder. Most people can control when they empty their bladder by controlling a muscular valve at the exit point. When this valve is opened and the bladder muscles contract, the urine enters the urethra, which the urine travels through before it exits the body. What is homeostasis? In biology, the term homeostasis is used a lot, and it can refer to many different biological processes. But at its core, homeostasis is actually a very simple concept. Homeostasis is the maintenance of a stable internal environment within an organism. Pretty simple, right? Just maintain the status quo and you'll be fine. But that's a lot easier said than done. Maintaining a stable internal environment in a person means having to carefully regulate countless parameters which include body temperature, internal pH, internal concentrations of various ions, hydration, blood pressure, and glucose levels in the blood, just to name a few. So how do our bodies do all of this and keep track of everything at the same time? Well, for the most part, our bodies keep track of all these different conditions separately, and then make adjustments 1 or 2 parameters at a time before something gets too far out of the normal range and starts affecting an important function. In this lesson, we'll take a look at one aspect of homeostasis by looking at how the human body maintains a stable body temperature. Humans, along with all other birds and mammals, are endotherms, which means that they actively maintain a stable body temperature by generating heat. Most people call endotherms warm-blooded animals. In contrast, most other animals like reptiles, fish, and insects are ectotherms, which are animals whose body temperature changes with their environment. People usually call ectotherms cold-blooded animals. You can remember the difference between endotherms and ectotherms if you remember that endo means inside and ecto means outside. So endotherms generate their own heat from within, and ectotherms get their heat from the outside. In general, ectotherms allow their body temperature to vary with the environment because it does not need to be maintained at a specific temperature for the animal to live. However, for most endotherms, body temperature must be maintained within a very narrow range at all times in order for the animal to live, with some exceptions for certain animals that lower their body temperature when they hibernate. Therefore, a stable body temperature of 98.6 degrees Fahrenheit, give or take only a few degrees, is one aspect of homeostasis in humans. The main advantage of having a stable body temperature is that it allows optimization of normal body functions, metabolism, and enzymes to a specific set temperature. This ensures that no matter what the environmental conditions, as long as the animal maintains temperature homeostasis, it will still be able to perform all normal body functions at or near optimal levels. However, there is a price to pay for always operating with full functionality, and that price is energy efficiency. Endotherms ensure a stable body temperature mainly by generating their own heat by burning calories. If they didn't have to maintain a high body temperature, they wouldn't burn as many calories, and therefore would be able to survive on less food or would be able to grow at a faster rate on the same amount of food. So now that we know why people maintain a stable body temperature, let's look at thermoregulation, or the regulation of internal body temperature. As stated before, our high metabolic rate is the major source of heat in our bodies. Most of the time, our body temperature exceeds the temperature of the environment, which means that we are continuously losing heat to the environment. The rate at which we lose heat is therefore a key determinant of body temperature, and many of the most effective methods of maintaining body temperature are based on either increasing or decreasing the rate of heat loss. Long ago, humans developed a unique method for controlling the rate of heat loss called clothing. You may laugh, but the most basic purpose of clothing is to provide insulation, which reduces heat loss. How much of a person's skin is covered by the clothing and how thick the clothing is determines how much insulation it provides and how much heat it conserves. Animals in the wild don't have clothing to increase their insulation. However, most endothermic animals do have a layer of insulation in the form of fur, feathers, or a thick layer of fat. Many animals experience changes in the thickness of these insulating layers to respond to seasonal changes in temperature. In addition, when mammals get cold, they immediately respond by raising their fur, which increases the thickness of their insulation. Even though humans generally don't have enough hair to serve as insulation, we still respond to cold by raising the hair on our bodies, which is why we get goosebumps when we are cold. This is a leftover response from our evolutionary ancestors who did have enough hair to serve as insulation. Yet another way to regulate heat loss is through the use of the circulatory system. Although we think of human body temperature as being 98.6 degrees Fahrenheit, the actual temperature varies throughout the body. Temperatures in the very center of our bodies, like around the heart or in our gut, are closer to 100 degrees Fahrenheit. But temperatures at the skin surface, and especially in our extremities, like our hands and feet, are significantly cooler and fluctuate quite a bit with the outside temperature. You have probably noticed that at times your hands or feet will be cold to the touch, and this is why. Blood that is pumped from the heart in our core out to all the tissues of the body and then back to the heart unavoidably transfers heat from the core to wherever it is pumped. Blood that is pumped through capillaries at the surface of the skin will lose a lot more heat than blood that is pumped through capillaries that are located deeper within our tissues. Our bodies take advantage of this and adjust blood flow through various tissues to help the body either conserve or dissipate heat. When the body needs to conserve heat, capillaries at the skin surface constrict and limit the amount of blood that flows through them, reducing heat loss. When the skin is exposed to extremely cold temperatures, the body will even completely shut down the blood flow to the exposed area as a survival mechanism. If blood flow is shut down for too long, cells in the area will die. This phenomenon is commonly called frostbite. Capillaries at the skin surface can also be used in the opposite manner to help cool us down. When our bodies are too warm, capillaries at the skin surface are dilated, increasing blood flow to the surface and greatly increasing the amount of heat lost from the blood. Another method that the body uses to cool itself down is sweating. You may remember that water absorbs heat when it evaporates. Sweating releases water onto the surface of the skin. As it evaporates, it absorbs heat from the body and cools it down. So, how does our body know when it needs to cool down and when it needs to warm up? It turns out that the body has an internal thermostat that acts a lot like the thermostat you might have in your home. The part of the brain that acts as the body's thermostat is the hypothalamus, which is the part of the brain that tells the body what it needs to do to survive and reproduce. The hypothalamus receives signals from all over the body regarding temperature. If the hypothalamus starts receiving too many signals that the body is cold, an area of the hypothalamus called the heating center is activated. When the heating center is activated, we feel the sensation of being cold and respond behaviorally to warm up by putting on additional clothes, seeking a heat source, or seeking shelter. In addition, the heating center also constricts blood vessels close to the skin surface to reduce heat loss, raises the hair on our bodies, and can activate the shivering response, which generates heat from small, rapid muscle movements. Conversely, if the hypothalamus starts receiving too many signals that the body is hot, an area of the hypothalamus called the cooling center is activated. When the cooling center is activated, we feel the sensation of being hot and respond behaviorally to cool down by removing insulating clothing, seeking shade, or seeking another cooling device like a fan or air conditioning. The cooling center also dilates blood vessels near the skin surface to increase heat loss and activates sweating to induce heat loss by evaporation. So to review, homeostasis is the maintenance of a stable internal environment within an organism. Maintaining homeostasis in humans requires regulation of countless parameters which include body temperature, internal pH, internal concentrations of various ions, hydration, blood pressure, and glucose levels in the blood, just to name a few. Humans, along with all other birds and mammals, are endotherms, which means that they actively maintain a stable body temperature by generating heat. Maintaining a stable internal temperature is a key aspect of homeostasis for endotherms and involves several different methods of thermoregulation, or the regulation of internal body temperature. The human body has an internal thermostat located in the hypothalamus, which is the part of the brain that tells the body what it needs to do to survive and reproduce. If the hypothalamus receives too many signals that the body is cold, it activates several heat-generating and conserving mechanisms. However, if the hypothalamus receives too many signals that the body is hot, it activates several cooling mechanisms instead. Animals interact with their environment, so it's only logical that in order to take advantage of any benefits and avoid possible dangers, they need a way to be able to monitor their environment and then respond in an appropriate manner. In a nutshell, this is the job of the nervous system, and it can be broken down into three main functions. First, the nervous system collects sensory input from the body and external environment. Second, the nervous system then processes and interprets the sensory input. And finally, the third main function of the nervous system is to respond appropriately to the sensory input. Let's take our friend Timmy as an example. Timmy likes to play in his backyard, and one of his favorite things to do in the backyard is to catch butterflies. Timmy's eyes scan the backyard. Actually, what his eyes are really doing is taking light input and converting it into electrical signals that are sent to the brain. Timmy's eyes are sensory organs, or organs which are devoted to gathering sensory input and sending it to the brain. The brain then interprets these signals and converts them into a visual image, which is how Timmy sees the butterfly land on a nearby flower. Not only does Timmy's brain give him a visual image, but it also categorizes what Timmy sees. So he immediately thinks, there's a butterfly landing on a flower. The workhorses of the nervous system that do all of these things that allow Timmy to see the butterfly and recognize it are called neurons. Neurons are specialized cells of the nervous system that transmit signals throughout the body. So, now that Timmy's neurons have gathered information from the environment, relayed this information to his brain, processed the information into a visual, and categorized what he sees, Timmy now has a decision to make. What does he do with this information? Does he ignore it and continue scanning the backyard, or does he make use of this information and act on it? Not surprisingly, Timmy decides he wants to catch the butterfly. So his brain sends signals to the muscles to slowly move towards the butterfly. The neurons that transmit this signal to the muscles are called motor neurons. Motor neurons are not just limited to sending signals to muscles, they can also send signals to glands within our body and stimulate or inhibit secretion of various substances that then carry out or regulate many body functions. But back to Timmy, who was just about to snatch the butterfly when the neighbor's dog barked and startled Timmy. Poor Timmy couldn't help it. The unexpected loud noise triggered a reflex reaction, an automatic, involuntary reaction to an unexpected stimulus. And Timmy jumped backwards before he knew what was happening. Timmy was scared. His brain remembered that the dog next door was big and mean, so it categorized the dog as a threat and told Timmy's legs to run away as fast as they could, so he ran, until he tripped and fell down and scraped his knee when he fell. Specialized pain receptor neurons signal Timmy's brain that his knee is hurt, and Timmy feels pain. Fortunately, Timmy's mother is just inside and hears him crying. She brings him inside and cleans the scraped knee. She puts Timmy's favorite lizard band-aid on his knee and puts an ice pack on it. Now another type of neuron is being stimulated in Timmy's knee, thermoreceptors, which are neurons that sense temperature. These thermoreceptors are now signaling to Timmy's brain that his knee is suddenly very cold. But Timmy isn't worried because he knows that the ice pack will make his knee feel better. You may have noticed that Timmy has lots of neurons that send sensory information to his brain, like the neurons in his eyes that send visual information, the pain receptors in his knee that respond to injury, and the thermoreceptors that sense temperature. All of these are different types of sensory neurons, or neurons that collect sensory input and send it to the brain. In addition to the sensory neurons already mentioned here, Timmy also has sensory neurons in his ears that allow him to sense sound, sensory neurons in his tongue that allow him to taste, neurons in his nasal cavity that allow him to smell, and many more types of sensory neurons that are always sensing what is happening inside his body as well. Let's check back in with Timmy and take a look at an example of sensory neurons that monitor internal body conditions. After Timmy was all patched up, his mother made him a grilled cheese sandwich, which was his favorite. Within 5 minutes, the whole sandwich was in Timmy's stomach. Sensory neurons in the stomach wall are stimulated by the presence of food and send a signal to the brain. The brain then relays a signal back to motor neurons in the stomach that stimulate the secretion of gastric juice and contractions of stomach muscles to mix the food and gastric juice. This nervous pathway is one example of a function controlled by the autonomic nervous system, which is the part of the nervous system that involuntarily regulates internal body functions. Other body functions that are also under autonomic control are breathing, heart rate, salivation, pupil dilation, sweating, and blood vessel constriction and dilation, to name a few. In contrast, the somatic nervous system is the part of the nervous system that voluntarily responds to external stimuli. All of the different senses that Timmy experienced earlier in the lesson, like sight, hearing, pain, and cold, and his conscious mental and physical responses to them, are all parts of the somatic nervous system. In many cases, both of these systems work together in a coordinated fashion. When Timmy heard the neighbor dog bark and got scared, his heart rate and breathing rate were immediately increased by his autonomic nervous system so that he could run away, an action controlled by the somatic nervous system. So let's review. The nervous system has three main functions. 1. To collect sensory input from the body and external environment. 2. To process and interpret the sensory input. And 3. To respond appropriately to the sensory input. The workhorses of the nervous system are cells called neurons, which are specialized cells of the nervous system that transmit signals throughout the body. When Timmy's eyes scan the backyard, sensory neurons, or neurons that collect sensory input and send it to the brain, sense light input and convert it into electrical signals that are sent to the brain. Neurons in the brain then process this information and convert it into a visual image, which is how Timmy sees the butterfly land on a nearby flower. Not only does Timmy's brain give him a visual image, but it also categorizes what Timmy sees, so he immediately thinks, there's a butterfly landing on a flower. Timmy decides he wants to catch the butterfly, so his brain sends signals to his muscles to move toward it. The neurons that transmit this signal to the muscles are called motor neurons, which are neurons that transmit signals to responsive tissues. Timmy's muscles respond to the stimuli and he moves towards the butterfly. The functions that I just described are all controlled by the somatic nervous system, which is the part of the nervous system that voluntarily responds to external stimuli. All of the different senses that Timmy experienced earlier in this lesson, like sight, hearing, pain, and cold, and his conscious mental and physical responses to them, are all parts of the somatic nervous system. In contrast, the autonomic nervous system is the part of the nervous system that involuntarily regulates internal body functions. Some of the body functions that are under autonomic control are digestion, breathing, heart rate, salivation, pupil dilation, and sweating. Neurons are specialized cells of the nervous system that transmit signals throughout the body. You may already know that neurons can do many different things, from sensing external and internal stimuli to processing information and also directing muscle actions. But do you know how far a neuron can send a signal, or how neurons actually work at the cellular level? In this lesson, we'll take a look at neurons and how they work. The most unique and important structures of a neuron are the long extensions that extend out from the cell body. There are two types of extensions, dendrites and axons. Dendrites are extensions of neurons that receive signals and conduct them toward the cell body. Axons are extensions of neurons that conduct signals away from the cell body to other cells. Both axons and dendrites can extend far away from the cell body, and some human axons can reach lengths of over 3 feet, but that's nothing compared to giraffes, which have axons as long as 15 feet. Okay, so neurons have dendrites and axons that can extend far from the cell body and relay signals to and from other cells. But how do they actually relay these signals? For this, let's take a closer look at a section of a neuron's dendrite. When the dendrite is not transmitting a signal, it is said to be in its resting state. In this state, the inside of the cell has a net negative charge, and the outside of the cell has a net positive charge. The membrane is said to be polarized, because negative and positive charges exist on opposite sides. The polarized state of the membrane is actively maintained by the neuron through the use of sodium-potassium pumps. These sodium-potassium pumps pump 3 positively charged sodium ions out of the cell for every 2 positively charged potassium ions it pumps into the cell. Each cycle the pump increases the polarization a little more. In addition, potassium ions leak back across the membrane and out of the cell by diffusion, which again creates more negative charge inside the cell and more positive charge outside the cell. When a neuron receives a signal, sodium channels in the membrane are opened and allow a localized influx of positive sodium ions into the cell, which causes depolarization, or a reduction of the difference in charge across the membrane. The localized depolarization also triggers nearby sodium channels to open up and depolarize the membrane nearby, which then causes more sodium channels to open up further away and depolarize the membrane there, and so a chain reaction is started. Depolarization occurs in a wave across the membrane, starting at the dendrite that received the signal, moving toward the cell body, across the cell body, and then away from the cell down the axon. You can think of depolarization as being like people standing up to do the wave in a football stadium. When the wave reaches people who are seated, it triggers them to stand up, which then triggers the people next to them to stand up, and so on, continuing the wave through the stands. And much like the people in the stands who sit back down, the membrane repolarizes by closing the sodium channels and firing up the sodium-potassium pumps to reestablish the difference in charge across the membrane, and the neuron is ready to pass along another signal. So what happens when the signal reaches the end of the axon? How does the neuron pass the signal along to another cell? At the ends of axons, the axon usually splits into several smaller branches. Each of these branches terminates at another cell at a junction called a synapse, which is the site where an axon terminates at a target cell. At the synapse, there is a small gap between the terminal end of the axon and the target cell. When the depolarizing signal reaches the synapse, it triggers the release of signaling molecules called neurotransmitters, which are the signaling molecules used at the synapse to pass a signal from a neuron to its target cell. These neurotransmitters diffuse across the very short gap from the axon to the surface of the target cell. The membrane of the target cell at the synapse has lots of receptors that the neurotransmitter can specifically bind to, and these receptors are coupled to ion channel proteins that are controlled by the receptor. Typically, when the neurotransmitter binds to the receptor, the ion channel is opened and a specific type of ion is allowed into the target cell. If the signaling neuron is an excitatory neuron, the ion channel will allow sodium ions to enter the cell and cause depolarization at the target cell. However, if the signaling neuron is an inhibitory neuron, a different ion channel will be opened that will allow a different type of ion, like negatively charged chloride ions, into the cell that will increase the polarization of the target cell and decrease the chances of depolarization, even if the cell receives an excitatory signal at the same time. So to review, neurons are specialized cells of the nervous system that transmit signals throughout the body. Neurons have long extensions that extend out from the cell body called dendrites and axons. Dendrites are extensions of neurons that receive signals and conduct them toward the cell body. Axons are extensions of neurons that conduct signals away from the cell body to other cells. When a neuron is in its resting state, the membrane is said to be polarized because negative and positive charges exist on opposite sides. When a neuron receives a signal, sodium channels in the membrane are opened and allow a localized influx of positive sodium ions inside the cell, which causes depolarization, or a reduction of the difference in charge across the membrane. The localized depolarization also triggers nearby sodium channels to open up and depolarize the membrane nearby, which then causes more sodium channels to open up further away and depolarize the membrane there, and so a chain reaction is started. Depolarization occurs in a wave across the membrane, starting at the dendrite that received the signal, moving toward the cell body, moving across the cell body, and then away from the cell down the axon. Axons terminate at junctions with their target cells called synapses. At the synapse, there is a small gap between the terminal end of the axon and the target cell. When the depolarizing signal reaches the synapse, it triggers the release of signaling molecules called neurotransmitters. These neurotransmitters diffuse across the very short gap from the axon to the surface of the target cell and bind to receptors that control ion channels, causing the ion channels to open. If the signaling neuron is excitatory, the ion channel will allow sodium ions to enter the cell and cause depolarization at the target cell. However, if the signaling neuron is inhibitory, a different ion channel will be open that will allow inhibitory ions like negatively charged chloride ions into the cell. This will increase polarization of the target cell and decrease the chances of depolarization even if the cell receives an excitatory signal at the same time. The nervous system is quite complex and includes many types of neurons that have many different functions. But it is very well organized, and despite its complexity, the organization is actually quite elegant and reflects the functions of its component neurons. The central nervous system is composed of the brain and spinal cord, and functions mainly to process information and determine the appropriate responses. The peripheral nervous system is composed of all of the sensory and motor neurons of the body, and it functions to gather sensory information and to control the actions of our bodies. The peripheral nervous system includes two basic types of neurons, sensory neurons and motor neurons. You may remember that sensory neurons are neurons that collect sensory input and send it to the brain, and motor neurons are neurons that transmit signals to responsive tissues. When Timmy sees a butterfly in his backyard, his eyes collect a lot of detailed information about the light they receive. This information is turned into neural impulses by specialized sensory neurons in the eye called photoreceptors, which are sensory neurons that sense light. These neural impulses are then carried by the axons of the photoreceptors to neurons in the brain. But photoreceptors aren't the only type of sensory neuron. In fact, humans probably have more types of sensory neurons than you think. For example, people have neurons that sense temperature called thermoreceptors. We also have pain receptors that are sensitive to excess heat, pressure, or specific molecules that are released from injured tissues. Another whole class of sensory neurons are the mechanoreceptors, which are sensory neurons that are stimulated by physical forces. There are many different types of mechanoreceptors. Some mechanoreceptors are located near the skin surface and sense very small changes in pressure that we interpret as touch. Others, located deeper in the skin, respond to stronger pressure and inform us about the strength of the pressure. Other mechanoreceptors are sensitive to stretch. We have lots of these stretch-responsive receptors in our muscles that monitor the lengths of all of our muscles, which is important for the coordination of movement. Even our hearing is dependent on mechanoreceptors. Specialized cells in the ear called hair cells are sensitive to vibrations of membranes in the ear, which allow us to hear sounds. Chemoreceptors are yet another class of sensory neurons, and they respond to specific molecules. In humans, chemoreceptors are responsible for the senses of taste and smell. Chemoreceptors are also used by the body to monitor the concentration of various substances in the blood, which is necessary for maintaining homeostasis. As you can see, humans have quite a variety of sensory neurons, but there are many more types of sensory neurons found in other animals that humans don't have. For example, honeybees and some other animals have photoreceptors that can sense ultraviolet light, which we cannot see. And what appears to be a plain yellow flower to us sometimes turns out to be a yellow and ultraviolet striped yellow to the bee, just like this flower here. The photograph on the left was taken with a standard camera. The photograph on the right is of the same flower, but was taken with an ultraviolet camera, and reveals that the flower is actually yellow with ultraviolet stripes that we cannot detect with our eyes, but the bees can. Hammerhead sharks and platypuses have electroreceptors, that they use to sense the electric fields of animals that they might not be able to see in dark water or buried under mud and sand. And many snakes have infrared sensors that allow them to locate warm-blooded animals based on the infrared radiation that they produce. As you can see, there is a wide variety of sensory neurons that exist in the animal kingdom. The other half of the peripheral nervous system is composed of motor neurons, which have a completely different job from sensory neurons. While sensory neurons monitor the status of the body and the environment around us and send this information to the central nervous system, motor neurons receive signals from the central nervous system, which they then use to tell responsive tissues what to do. Motor neurons aren't normally classified into different groups, and they are most often associated with controlling muscle actions. But some motor neurons also signal to glandular tissues and control secretion of various substances in the body, like saliva and gastric juice. The main functions of the central nervous system are to process sensory information and determine what the appropriate reaction is. Neurons of the central nervous system only interact with other neurons and are called interneurons. Unlike sensory and motor neurons, interneurons both send and receive signals to and from other neurons. For the most part, sensory neurons throughout the body synapse with interneurons in the spinal cord. The spinal cord neurons will then send the signal to the brain for further processing. But sometimes an immediate reaction before thorough processing can be completed is advantageous. In these special cases, the spinal cord neurons send the signal out to the motor neurons to cause an automatic reaction to a very specific stimuli without any involvement of the brain. This type of reaction is called a reflex, which is basically an automatic reaction to the stimulus that is not consciously controlled. A well-known reflex is the knee-jerk reaction. This is a reaction that doctors use to test people's reflexes. The doctor will tap on the tendon just below the kneecap, which causes a reflexive muscle contraction to straighten the leg. Even though we don't have control over it, we're still consciously aware of what's going on. We can still feel the tap on our tendon, and we can feel our leg move, so the signals are being sent to our brain too. So let's do a quick review. The central nervous system is composed of the brain and spinal cord, and functions mainly to process information and determine the appropriate responses. The peripheral nervous system is composed of all of the sensory and motor neurons of the body, and it functions to gather sensory information and to control the actions of our bodies. The peripheral nervous system includes two basic types of neurons, sensory neurons and motor neurons. Sensory neurons are neurons that collect sensory input and send it to the brain. There are lots of different types of sensory neurons that are specifically designed to gather different types of information and send it to the central nervous system. Motor neurons are neurons that transmit signals to responsive tissues. These neurons receive signals from the central nervous system and transmit them to responsive tissues to appropriately respond to the situation. The central nervous system is composed of two major organs, the brain and the spinal cord. The spinal cord is responsible for receiving signals from most of the sensory neurons throughout the body and passing these signals along to the brain. It is also responsible for transmitting signals to motor neurons. Some specialized immediate reactions, called reflexes, are automatic reactions to stimuli that are not consciously controlled. These reflexes don't involve the brain, but are mediated by the spinal cord instead. However, all of the more complex processing of sensory information and decision-making is performed by the brain.
Your psychology friends are going to help you remember the parts of the brain. Let's start with the oldest parts of the brain, which are responsible for basic functions, like breathing and walking. Here's a phrase that can help you remember the 4 oldest parts of the brain. Bandura totally roughhoused clowns. This is especially useful if you're familiar with Albert Bandura's experiment in which children hit a Bobo clown doll with toy hammers after learning aggressive behavior from watching adults do the same thing. But the first letter in each of these words will also help you remember the first letters in the 4 oldest parts of the brain, which are brainstem, thalamus, reticular formation, and cerebellum. Got it? Great! But they're just fancy words, unless we know what they do. Let's explore these parts of the brain from the bottom up. Your brainstem is the stem of your brain. It's located at the top of your spinal cord where it enters your skull. It controls how fast your heart beats and how quickly you breathe. B stands for Bandura and brainstem. Bandura's brainstem allows him to breathe. Your thalamus sits on your brainstem. It receives information from your senses and forwards them on to other parts of your brain. T stands for totally and thalamus. Your thalamus totally tells your brain about touch and taste. Your reticular formation is made up of nerve cells that run from your spinal cord through your brainstem and thalamus. Your reticular formation is at work when you eat, sleep, and become aroused. Its job is to filter stimuli and pass on important signals. R stands for roughhoused and reticular formation. Your reticular formation is responsible for relaying reactions when you roughhouse. Your cerebellum is behind your brainstem. It helps you walk and remember. C stands for clowns and cerebellum. The cerebellum controls a clown's coordination. Bandura totally roughhoused clowns can help you remember brainstem, thalamus, reticular formation, and cerebellum. One more time. Brainstem, thalamus, reticular formation, cerebellum. Got it? Now let's move on to the core of the brain and see how your psychology friends can help you remember the 4 parts of the limbic system. The limbic system is the part of your brain that controls motivations and emotions. You can use the phrase, "Hungry hounds ate Pavlov," to help you remember the 4 parts of the limbic system: hypothalamus, hippocampus, amygdala, pituitary. In a famous experiment about behavior conditioning, Ivan Pavlov trained dogs to drool when they heard a bell ring instead of when they saw food. You can imagine how hungry these dogs might have gotten to the point of taking matters into their own paws. Your hypothalamus helps regulate hunger, so it makes sense that hungry stands for hypothalamus. The hypothalamus also influences your sex drive, thirst, and body temperature. Your hippocampus processes long-term memory. Use your hippocampus to create an association between hounds and hippocampus. This is how we store information for use later on the test. Your amygdala is responsible for your feelings of fear and anger. Hungry hounds acted on their aggression and ate Pavlov when they found out he was conditioning them with a bell. Your pituitary gland is a master gland that helps regulate the release of hormones. In our imaginary scenario, Pavlov may have overlooked the influence of the pituitary glands in the dog's reaction to the stressful experiments. Poor Pavlov. Hungry hounds ate Pavlov. This phrase can help you remember the 4 parts of the limbic system that are responsible for motivations and emotions: hypothalamus, hippocampus, amygdala, pituitary. One more time: hypothalamus, hippocampus, amygdala, pituitary. Finally, let's look at the names of the last part of the brain. Your cerebral cortex is made up of 4 lobes, and the 4 lobes of the brain can be memorized using the phrase, "Freud tore his pants off," which stands for frontal, temporal, parietal, and occipital. It makes sense that Sigmund Freud, a famous psychologist who thought that many of our impulses are driven by sex, would decide to do away with clothing altogether. Okay, let's look at what each of these 4 lobes do, working from front to back. The frontal lobe of your brain helps you speak, coordinate your movements, and is also involved when you plan. The temporal lobe of your brain processes information about what you hear. The parietal lobe of your brain processes information about what you touch. The occipital lobe of your brain processes information about what you see. Freud tore his pants off can help you remember frontal, temporal, parietal, and occipital. One more time: frontal, temporal, parietal, and occipital. You've learned the 4 parts of the brain involved in basic functions: brainstem, thalamus, reticular formation, cerebellum. The trick to remembering these is to think about how Bandura totally roughhoused clowns. You've also learned about the 4 parts of the brain that control motivations and emotion: hypothalamus, hippocampus, amygdala, pituitary. which you remember when you imagine that hungry hounds ate Pavlov. And you won't forget the 4 lobes of the brain: frontal, temporal, parietal, and occipital, because you'll visualize that Freud tore his pants off. Well, maybe you don't want to visualize that. You can just memorize it.
The innate immune system. When people think of the immune system, the first things that come to mind are often antibodies or vaccines or some other aspect of acquired immunity to an infectious agent that the body has already seen. But how does the body protect itself against potentially dangerous bacteria and viruses that it has never seen before? The immune system needs a way to quickly respond to a variety of unknown invaders, whether they've been encountered before or not, and all of the aspects of the immune system that respond to these unknown threats contribute to our body's innate immunity, or the ability of the immune system to respond to unknown threats. The innate immune system is capable of mounting a defense against a variety of different threats, including any potential pathogens or foreign invaders that are capable of causing disease that might enter the body through an open wound or body orifice, viruses that have already infected a cell, malignant or cancerous cells, and even multicellular parasites. So let's take a look at how the innate immune system works. Let's say that our friend Phil is rock climbing when he cuts himself on a sharp edge. It's not a bad cut, so Phil ignores it, but his immune system certainly doesn't. As soon as Phil sustains even the smallest injury, the potential for infection by a foreign invader dramatically increases. In fact, even with the most minor cuts, thousands if not millions of foreign entities, most of them bacteria, will enter the wound. So whenever there is an injury, it is in the body's best interest to mount an immediate immune response to destroy all foreign invaders before they can establish a foothold. The first response of the body to a localized injury or infection is inflammation, or the immediate response of the body to injury or infection that includes an influx of blood plasma and immune cells to a localized area. Injured tissues release a number of signaling molecules that cause blood vessel dilation, increase blood vessel permeability, and cause endothelial cells that line the blood vessels to present adhesion molecules on their surface. These adhesion molecules are recognized by immune cells in the blood, which bind to the adhesion molecules and stick to the blood vessel wall. The immune cells then squeeze themselves between endothelial cells of the now more permeable blood vessels and enter the tissue surrounding the vessel. The signaling molecules are immune cell chemoattractants that cause the immune cells to exit the blood vessel and migrate to the site of the injury. One of the best-known inflammatory signaling molecules is histamine, a signaling molecule that mediates the inflammatory response, which is capable of dilating blood vessels, increasing blood vessel permeability, and causing immune cells to exit the bloodstream and enter the damaged tissue. There are 5 signs that describe an inflammatory response: pain, heat, loss of function, swelling, redness. The first immune cells that arrive at an injured site are mostly neutrophils, which are quick-response immune cells that recognize and destroy bacteria. Neutrophils are the most numerous type of immune cell in the blood, and their job is to immediately respond to inflammation and kill bacteria by phagocytosis, which is the process by which a cell engulfs, destroys, and digests another cell, bacteria, or cellular debris. Neutrophils have several different types of receptors and recognize several different pathogen-associated molecular patterns that are specific to bacteria and not found in human cells. When the receptors bind to the bacteria's specific patterns, the neutrophil captures the bacteria and engulfs it, surrounding it with its plasma membrane. This intracellular compartment is called a phagosome because it is created during the process of phagocytosis. Lysosomes then fuse with the phagosome to add their digestive enzymes, which kill the bacteria and break it down into its component amino acids, nucleic acids, and monosaccharides. These nutrients can then be used by the neutrophil or released into the tissue for use by other cells. Macrophages are another type of immune cell that phagocytizes foreign organisms, dead cells, and cellular debris. Macrophages are a little bigger than neutrophils, live longer than neutrophils, and stay in the tissue even after all of the pathogens are gone. They act like a cleanup crew that phagocytizes leftover debris, bacteria, and dead cells. Complement. So far, most of the components of the innate immune system that we've talked about have been different types of cells, But there are non-cellular components of the innate immune system too. Complement is a group of serum proteins that mark and destroy foreign microorganisms. Complement proteins are capable of forming a membrane attack complex on plasma membrane surfaces through a series of interactions between the different complement proteins. If the complement proteins are allowed to complete the membrane attack complex, it creates holes in the plasma membrane of its target and destroys it. Complement proteins are very nonspecific. They will attempt to form membrane attack complexes on any plasma membrane they can. Now, this might cause a problem if complement proteins formed membrane attack complexes on the body's own cells and destroyed them, but our cells don't let this happen. Complement protein complexes that are forming on cell surfaces are fairly unstable and can be disrupted at various stages of completion by at least 8 known membrane proteins that are expressed on the surface of all of our cells. So while complement proteins try to form membrane attack complexes on any membrane surface, the body's own cells easily clear the proteins from themselves. But most bacteria and parasites don't have these complement disrupting proteins, so the membrane attack complex is formed and the cell is lysed. However, certain types of microorganisms are resistant to lysis. In this case, the complement proteins remain on the membrane and mark the microorganism as foreign. Neutrophils and macrophages have receptors to complement proteins and will phagocytize the marked invaders. So as you can see, complement serves to kill or mark all foreign cells and doesn't rely on recognizing specific organisms or molecules. This allows complement proteins to neutralize a very large number of invaders, and since our own cells disrupt complement complexes, they don't attack the body's own cells. Natural killer cells are a type of immune cell that kills cells not expressing MHC1 molecules on their surface. Like security guards in a restricted area, natural killer cells check the IDs of cells they come into contact with. For NK cells, the ID that they need to see is the MHC1 molecule that normal cells express at all times on their cell surface. But there are 3 types of cells that do not always express MHC1 molecules on their surface: red blood cells, which NK cells ignore for reasons that we won't go into here, cells infected with a virus, and malignant or cancerous cells. But the body doesn't have a quarantine room or a jail where it can put dangerous cells. Instead, the immune system can't take any chances, and the NK cells kill suspicious cells that do not express the correct MHC1 molecule on their surface. Eosinophils are immune cells that attack multicellular parasites. These cells target multicellular parasites like worms by moving into position right up against the parasite and then releasing a host of destructive enzymes from their internal granules directly onto the target. Eosinophils also serve to ramp down the inflammatory response by arriving in the late stages of the response and releasing inhibitors of inflammatory molecules. Let's review. Innate immunity is the ability of the immune system to respond to unknown threats. The innate immune system is capable of mounting a defense against a variety of different threats, including any potential pathogens or foreign invaders that are capable of causing disease. Inflammation is the immediate response of the body to injury or infection that includes an influx of blood plasma and immune cells to a localized area. The first immune cells that respond to inflammation are mostly neutrophils, which are quick response immune cells that recognize and destroy bacteria. Neutrophils immediately respond to inflammation and kill bacteria by phagocytosis, which is the process by which a cell engulfs, destroys, and digests another cell, bacteria, or cellular debris. Macrophages are another type of immune cell that phagocytizes foreign organisms, dead cells, and cellular debris. Macrophages stay in the tissue even after all of the pathogens are gone and act as a cleanup crew that phagocytizes leftover debris, bacteria, and dead cells. Complement is a group of serum proteins that mark and destroy foreign microorganisms. Complement proteins are capable of forming a membrane attack complex on plasma membrane surfaces through a series of interactions between the different complement proteins. If the complement proteins are allowed to complete the membrane attack complex, it creates holes in the plasma membrane of its target and destroys it. The body's own cells have several different membrane proteins that disrupt complement complexes and don't allow the complement proteins to form membrane attack complexes on the body's own cells. Natural killer cells are a type of immune cell that kills cells that do not express MHC1 molecules on their surface. Like security guards in a restricted area, natural killer cells check the IDs of cells they come into contact with. For NK cells, the ID that they need to see is the MHC1 molecule, which normal cells express at all times on their cell surface. NK cells kill suspicious cells that do not express the correct MHC1 molecule on their surface, which is usually a sign that the cell is cancerous or infected with a virus. Eosinophils are immune cells that attack multicellular parasites by releasing a host of destructive enzymes from their internal granules directly onto their target. Eosinophils also serve to ramp down the inflammatory response by arriving in the late stages of the response and releasing inhibitors of inflammatory molecules. Acquired immunity is an immune response to a specific pathogen that can be reactivated if the pathogen is ever encountered again. Most people are already familiar with this concept. For example, everybody knows that if you've had chickenpox, you won't get it again because you've developed immunity to it. But how do we actually develop this immunity? Would you believe that acquired immunity is largely developed by chance and large-scale trial and error? It may seem odd, but that's basically how it's done. The workhorses of the acquired immune system are lymphocytes, which are white blood cells that recognize and respond to a single molecular structure. Each lymphocyte is very specific and can only recognize a single antigen or molecular structure that is recognized by the acquired immune system. The strange part is that the antigen binding site that a lymphocyte uses to recognize an antigen is created in an almost random fashion before the lymphocyte ever comes in contact with a foreign protein or pathogen. So what are the chances of creating a binding site by chance that can recognize a random peptide that is made by a virus? 1 in a million? 1 in 10 million? Maybe 1 in 100 million? It's impossible to know what the odds really are, but if they're close to this, we are not in bad shape, because immunologists estimate that our immune systems typically have about 1 trillion lymphocytes, which can recognize about 100 million different molecular structures. Even though the odds of matching a particular viral peptide with a randomly created receptor may be close to the odds of winning a multi-million-dollar lottery jackpot, our immune systems can buy enough tickets to even up the odds. In addition, all pathogens make multiple different proteins, and most of these can be chopped up into several smaller peptides that can be recognized by lymphocytes. So not only does the immune system buy 100 million tickets, but the lottery it's trying to win probably has, at the very least, a few hundred different jackpot-winning combinations. With so many potential antigens and 100 billion different antigen binding sites to try against each foreign peptide, our immune system stack the odds in our favor so that we win the jackpot almost every time. Now, you may still be wondering how immune cells can create so many unique antigen receptors and antibodies. Think of it in terms of creating a sandwich. If you had a choice of 250 different types of bread, 200 different cheeses, 50 kinds of lunch meat, 8 different vegetables, and 5 different condiments, think about all the different types of sandwich you could make, combining one bread with one vegetable, one meat, one cheese, and one condiment. You could make 100 million different sandwich combinations. That's basically how a lymphocyte determines what the binding site of its antibodies or antigen receptor will be. You see, the binding sites are actually composed of 5 different modular amino acid sequences, and for each module, the cell has several, sometimes even hundreds of options available to it. Creating all of the possible modular sequence combinations and adding a couple of genetic tricks to multiply the combinations a couple more times would create about 100 million unique combinations. But once the combination is determined in a lymphocyte, it's locked into place for that cell and all descendants of that cell. Okay, now let's take a look at the different types of lymphocytes. There are 2 major types of lymphocytes: T lymphocytes, or T cells, and B lymphocytes, or B cells. These 2 types of lymphocytes are named and classified based on where they mature in the body. T cells mature in the thymus. B cells mature in the bone marrow. But where they mature isn't the only difference between T and B cells. As we'll soon see, there are some major functional differences between T cells and B cells. As we talked about earlier, T cells are lymphocytes that mature in the thymus. There are several different types of T cells that serve different functions in the immune system, but we're going to just talk about 2 of them in this lesson. The first type of T cell that we're going to talk about is the cytotoxic T lymphocyte, or CTL for short. The CTL's main function is to destroy infected cells before they release mature parasites. That seems like a good idea, doesn't it? If the immune system can destroy infected cells before they release new parasites, it will be much easier to control the infection. But how does the CTL know which cells are infected with parasites? If the parasites are inside the cell, they're hidden from the CTLs, right? Well, not completely, because cells are always displaying sample peptides of internal proteins on the outside of the plasma membrane. The cells display these peptides just so that CTLs and other immune cells can monitor them and see a sample of what is inside the cell. If the antigen receptor of the CTL binds an antigen presented on the cell surface, the CTL will kill the cell and make sure that DNA inside the cell is also destroyed so that an infected cell doesn't release viable parasites when it is killed. So what happens if a cell is infected with a virus and then the virus instructs the cell not to present antigens on its surface? Wouldn't this allow the virus to hide from the CTLs? The short answer is yes. This allows the virus to hide from the CTLs. However, the molecule that presents antigens on a cell's surface is none other than the MHC1 molecule, and you may remember that natural killer cells kill cells that do not express MHC1 molecules on their plasma membrane. So the virus may evade the CTLs only to be destroyed by natural killer cells. The second type of T cells that we're going to talk about are helper T cells, which are T cells that activate other lymphocytes. Helper T cells are important regulators of immune responses to specific antigens. Some helper T cells, called Th1 cells, can activate cytotoxic T cells, and other helper T cells, called Th2 cells, can activate B cells. For now, we'll talk about Th1 cells and how they activate CTLs. Before a Th1 cell can activate a CTL, it must first be activated itself, meaning that it must encounter its specific antigen somewhere. If a viral infection is underway, then macrophages and other immune cells will come in contact with the virus and display viral antigens on their cell surface with MHC2 molecules. Cells that express foreign antigens with MHC2 molecules are called antigen-presenting cells. If a Th1 cell comes across an antigen-presenting cell that has an antigen the Th1 cell can recognize, it becomes activated and takes the antigen with it to present on its own cell membrane with an MHC2 molecule. The Th1 cell then goes in search of a CTL that can also recognize the same antigen and has already come across that antigen too. If the Th1 cell can find such a CTL, then the viral infection is serious enough to warrant a full response, and the Th1 cell activates the CTL to proliferate. The number of CTLs that can respond to this particular antigen is greatly expanded, and the immune system will now hopefully have enough antigen-specific CTLs to eliminate the virus. B cells are lymphocytes that are capable of producing antigen-specific antibodies. If a B cell has not yet encountered its target antigen, then it only produces antibodies that stay on its surface and act as antigen receptors for the B cell. Like CTLs, in order for B cells to be activated, they need to encounter their antigen on an antigen-presenting cell and be activated by a T helper cell or Th2 cell that has also seen the same antigen. Once the B cell is activated, it proliferates to produce lots of activated B cells, most of which then become antibody factories that secrete up to 2,000 antibodies per second for about 4 to 5 days until they reach the end of their lifespan. The rest of the B cell progeny become inactivated and wait at the ready for the next time that the immune system encounters the antigen. Okay, so we already know that antibodies are produced by B cells, and you may have also figured out that all antibodies produced by a B cell can only recognize the same single antigen that the B cell recognized when it was being activated. Now let's look a little bit at the structure of an antibody. Your basic secreted antibody is shaped kind of like a capital Y. It has two identical antigen binding sites located at the tips of the two branches of the antibody here and here. This part down here is called the Fc domain. It does not bind to antigens, however, it is very important for some key antibody functions. And speaking of functions, we haven't talked about what secreted antibodies do yet. Yes, they recognize and bind their antigen, but then what? It turns out that secreted antibodies serve four main functions. The first function is neutralization. If enough antibodies attach to a pathogen, they can completely coat it and block it from interacting with the body's cells. The next function is to mark pathogens for phagocytosis. When an antibody binds to a pathogen, the antigen binding sites face the pathogen, which leaves the Fc domain sticking out from the side of the pathogen. Not only that, but when an antibody is bound to its antigen, the structure of the Fc domain changes to a structure that receptors on neutrophils and macrophages recognize as a signal to phagocytize whatever the antibody is bound to. In addition to marking pathogens for phagocytosis, antibodies can also cause agglutination, or binding of pathogens together in clumps, due to the fact that antibodies have two binding sites, each of which could bind to separate pathogens and join them together. These clumps of pathogens are easier for cells to phagocytize as a group rather than one at a time. And finally, antibodies bound to a pathogen can stimulate complement formation of membrane attack complexes and kill pathogens that way. So in review, acquired immunity is an immune response to a specific pathogen that can be reactivated if the pathogen is ever encountered again. The workhorses of the acquired immune system are lymphocytes, which are white blood cells that recognize and respond to a single molecular structure or antigen, a molecular structure that is recognized by the acquired immune system. Three of the most important cell types in the acquired immune system are cytotoxic T lymphocytes, or CTLs, which are lymphocytes that destroy infected cells before they release mature parasites, helper T cells, which are T cells that activate other lymphocytes, and B cells, which are lymphocytes that are capable of producing antigen-specific antibodies. In addition, antigen-presenting cells, or cells that express foreign antigens with MHC2 molecules, play a very important role in the activation of T and B cells. In 1900, the average life expectancy in the United States was 48 to 51 years, and the top three leading causes of death were pneumonia, tuberculosis, and diarrhea, all of which were caused by infectious diseases. However, by 1997, the average life expectancy rose to 74 to 80 years, and pneumonia and influenza combined were the sixth leading cause of death and the only infectious diseases to crack the top 10. Many once fearsome and devastating diseases like smallpox, polio, syphilis, tuberculosis, measles, cholera, and the bubonic plague have either been nearly eradicated or are now easily treated with modern medicine. The dramatic increase in the average life expectancy, not only in the US but around the world in the last 160 years, has been accomplished in large part due to the effective prevention and treatment of infectious diseases through the use of sanitation, antibiotics, and vaccines. Experiments performed by Louis Pasteur in the 1860s convinced the Western scientific community that the germ theory of disease was correct. The germ theory of disease is a scientific theory which states that infectious diseases are caused by microscopic organisms that must be transferred from one host to another in order to spread. The acceptance of germ theory led to wide-scale changes in medicine, food preparation, and waste disposal. Prior to the 1860s, medical practices were extremely unsanitary. Washing hands and instruments before and after surgeries was not a common practice, and the more dried blood and gore that a surgeon had on his surgical clothes, the more prestige and experience he was assumed to have. However, after the realization that disease was caused by microorganisms that could be transferred by bodily fluids, food, and waste, wide-scale changes in medicine, food preparation, and waste disposal were implemented in the Western world. Death rates from infection in hospitals fell, and between 1850 and 1900, the average life expectancy in the U.S. jumped up by more than 25%, from 39 years to almost 50 years. Another breakthrough in medicine that has contributed to the rise in life expectancy over the last 160 years is the widespread use of vaccines. Vaccines are non-disease-causing variants or derivatives of pathogens that are used to create immunological memory against the pathogens themselves. Strangely enough, the first successful vaccination to be tested against a challenge with a pathogen was performed in 1796 by Edward Jenner, more than 60 years before the germ theory of disease was widely accepted by the scientific community. It had been observed that milkmaids did not often contract smallpox, which was in sharp contrast to an estimated 60% of the general population that contracted the disease. However, almost all milkmaids did contract a related but non-lethal disease called cowpox from the udders of the cows that they milked. It was postulated that contracting cowpox made a person resistant to smallpox infection, so Jenner took it upon himself to test this hypothesis. He took pus from the cowpox blisters of a milkmaid and inoculated a young boy with it. The inoculation produced a mild fever and some short-lived discomfort in the boy, but no other signs of infection. 2.5 weeks later, Jenner injected the boy with smallpox material, and the boy remained healthy. Jenner repeated the smallpox challenge 20 times, and the boy never showed any signs of smallpox infection. Jenner then successfully repeated his experiment on 23 additional people. Jenner's findings were published experiments spread among physicians in Europe and America, and by 1801, physicians all across Europe and even a few in North America started to use Jenner's vaccination procedure. Increased use of the vaccine and a concerted effort by the World Health Organization more than a century later to rid the world of smallpox through vaccination resulted in complete eradication of smallpox by 1977. The smallpox vaccine is an example of a live vaccine, meaning that a live virus is used as the vaccine. Usually live vaccines are actually just weaker, less dangerous forms of the disease-causing virus. These so-called live attenuated vaccines, or vaccines that consist of live laboratory-created versions of a virus that do not cause disease, are created in a laboratory under conditions that promote less harmful characteristics of a virus. In the case of the smallpox vaccine, nature provided a much less dangerous alternative to smallpox, which could still impart immunity to smallpox, a rather fortunate coincidence for humankind and modern medicine, especially given how deadly and infectious smallpox was. Some vaccines, called inactivated vaccines, are vaccines that are created by killing or inactivating the pathogen and then using it as the vaccine itself. Inactivated vaccines are easier to create than live attenuated vaccines and don't carry the risk of mutating back into a disease-causing form. However, because this type of vaccine isn't active and therefore doesn't infect the body's cells, the immune response that it elicits is not as strong as an immune response to a virus that is actively infecting cells. The resulting immunity is also not as strong and fades more quickly over time because fewer memory cells are produced. Therefore, inactivated vaccines must be administered more than once, sometimes 3 or more times, to create a strong enough immunity to the infectious agent. Each injection of the vaccine after the initial injection is called a booster shot, and with each additional shot, more memory cells are produced and the body's immunity is strengthened. In certain types of bacterial infections, the disease is caused by a toxin produced by the organism. In most of these situations, it has been found that the toxin can be inactivated with formalin and used as a toxoid vaccine, a vaccine created from an inactivated toxin. An immune system primed against a toxin will be able to identify and destroy the toxin before it can do too much harm, especially if booster shots are given. Recombinant subunit vaccines are vaccines that use recombinant DNA technology to produce high quantities of antigens that elicit an immune response. With the rapid advancement in recombinant DNA technology, these types of vaccines have become easy to create and produce. The real challenge with these vaccines is finding the antigens that will produce an immune response strong enough to create immunity. And finally, DNA vaccines hold lots of promise. There aren't any approved DNA vaccines on the market yet, but DNA has the potential to combine the best attributes of other vaccines. DNA vaccines, the future of vaccination? Well, DNA is easy to produce, manipulate, transport, store, and administer. In addition, if the DNA is incorporated into the genomes of some of the body's cells, it could mimic a viral infection without having the danger of introducing a virus that could revert back to a dangerous form. Messenger RNA or mRNA vaccines are a somewhat new development and are currently being given to prevent the spread of COVID-19. Like other more traditional types of vaccines, mRNA vaccines prepare the body's immune system to recognize a virus so that the patient does not get sick from the disease. However, mRNA vaccines do not contain viruses that are inactive or dead. Instead, they contain messenger RNA, or mRNA, which is a specialized protein that carries the genetic instructions for the body to make a spike protein, which is a surface protein on the COVID-19 virus that causes the disease to manifest. When this mRNA enters the body's cells, the manufacture of the spike protein begins, and the body's immune system then responds by making antibodies. The 2 mRNA vaccines that are now available are manufactured by the Pfizer-BioNTech and Moderna pharmaceutical companies. These are the first vaccines of this kind that have been given to the public on a large scale, although researchers have been working with them for many years. Since they are new, many people have reservations about taking them, But these vaccines do not change the body's DNA and have thus far largely been shown to be quite safe. In 1928, bacteriologist Alexander Fleming made one of the most important discoveries in modern medicine while cleaning up some old bacterial cultures. On a plate of Staphylococcus aureus, a contaminating colony of mold was growing on the plate. Fleming was going to clean out the dish with Lysol, but before he did, he noticed something strange about the plate. There was a ring around the mold where no bacteria was growing. Being a keen observer and a bacteriologist who was looking for a human-safe drug that could combat bacteria, Fleming recognized the potential in this contaminating mold for an antibacterial agent. The mold was identified as a penicillin mold, so he named the antibacterial agent that it produced penicillin, even though he wasn't able to isolate it himself. Not to worry though, because other scientists eventually purified penicillin, developed methods to mass produce it, and figured out how to use it to combat many types of bacterial infections, including Staphylococcus aureus, diphtheria, gangrene, pneumonia, syphilis, and tuberculosis. Penicillin was the first discovered antibiotic, or substance with antimicrobial properties that can be tolerated by humans. Antibiotics are fundamentally different from vaccines because they do not stimulate the immune system. Instead, they directly kill or inhibit the growth of microorganisms. The first major application of penicillin was in the mid-1940s by Allied forces to treat life-threatening bacterial infections of Allied soldiers, and is credited with saving hundreds of thousands of lives during the war. Penicillin was extremely effective against many types of bacteria, but there were lots of diseases that it was not effective against as well. Soon other molecules were discovered that had antimicrobial properties as well, each with a different set of diseases that they could combat. Some were more effective than others, and some had undesirable side effects in patients. The earliest antibiotics, like penicillin, were molecules isolated from natural sources like molds and plants. However, as chemical techniques became more advanced, chemists started altering these molecules and formed new forms of antibiotics. Eventually, chemists started making completely synthetic molecules and tested them by the thousands for antimicrobial properties. Today, there are over 100 known antibiotics, each with their own characteristics and microbial targets. And even as we speak, drug companies are still working to develop new types of antibiotics. So in review, there have been some remarkable breakthroughs in medicine in the last 220 years, but none have been quite so dramatic as the breakthroughs in microbiology and immunology. Between 1850 and today, the average life expectancy in the U.S. has nearly doubled, and most of that can be attributed to the control and prevention of infectious diseases through improved sanitation, vaccinations, and antibiotics. Improved sanitation may seem like an obvious way to limit disease. However, before Louis Pasteur essentially proved the scientific theory that infectious diseases are caused by microscopic organisms that must be transferred from one host to another in order to spread, otherwise known as the germ theory of disease, sanitation was often ignored as a factor in the spread of disease. Strangely enough, even before the germ theory of disease was widely accepted by the scientific community, vaccinations had already been discovered and effectively used against the spread of smallpox. The smallpox vaccine has been so successful that the disease has now been completely eradicated. No other infectious disease has been eradicated, but vaccines have been able to prevent large-scale epidemics of many other infectious diseases that were quite common before 1800. And finally, antibiotics were the third key breakthrough that has helped cure millions of various types of infectious diseases. Without antibiotics, diseases like strep throat, syphilis, gangrene, and staph infections would still be deadly killers.
Antibiotic resistance and virulence are two characteristics which make microbes more dangerous to humans. Actually, since virulence is defined as the relative ability to cause disease, it includes several different characteristics, but we'll talk about that later. For now, let's start by talking about antibiotic resistance, which occurs when a microbe acquires a gene which allows it to resist the effects of an antibiotic. Since the 1930s, when the first antibiotics were discovered, modern medicine has grown to rely on these drugs more and more. Antibiotics were used to treat obvious bacterial infections. Then they started being used in cases of suspected bacterial infections. And now, some doctors prescribe antibiotics for diseases that are almost certainly viral. even though antibiotics aren't effective against viruses at all. Some doctors may be covering all their bases in case they're wrong and the disease is caused by a bacteria. Others may be prescribing antibiotics to make the patient feel like they've gotten help from the doctor. And then there are doctors that have become so used to prescribing antibiotics that they do it almost automatically. After all, they're busy people, and it's hard to figure out what the exact cause of many illnesses is without doing a full workup and laboratory testing. It would be a waste of resources to hunt down the exact cause of every disease that they saw. Instead, it's easier to prescribe an antibiotic in case it's bacterial. The problem with using antibiotics so often is that when antibiotics are always present, bacteria evolve resistance to them. Worse still, bacteria can pass along resistant genes not only to their direct descendants, but to unrelated bacteria as well, even bacteria of different species. You see, unlike animal cells, bacterial cells exchange DNA with each other from time to time. In addition, bacteria can pick up DNA from the environment and use it themselves. So when an antibiotic-resistant bacteria dies and its DNA is floating around in the environment, another bacteria can pick it up. If the DNA is useful and gives the bacteria an advantage, then bacteria with that DNA will outcompete bacteria that don't have this competitive advantage. So in an environment where antibiotics are usually present, once one type of bacteria, even a harmless one, evolves resistance to an antibiotic, it's only a matter of time before it passes this resistance gene along to a pathogen. And the more antibiotics that these bacteria encounter, the faster the resistance genes spread. But overprescription of antibiotics isn't the only cause of antibiotic-resistant bacteria. Many farms across America routinely feed antibiotics to their livestock, even when there are no signs of disease. In fact, about 80% of all antibiotic usage in America occurs in agriculture. This means that farms are using more than 4 times the amount of antibiotics used by hospitals, research institutions, and biotechnology and pharmaceutical companies combined, making these farms perfect breeding grounds for antibiotic-resistant bacteria. So far, pharmaceutical companies have been able to stay one step ahead of antibiotic-resistant bacteria and develop new drugs before bacteria develop resistance to the old ones. But it is currently a biological arms race, and if we ever get to a point where we run out of new antibiotics, staph infections, gangrene, syphilis, and many other diseases will again start claiming more lives. Switching gears a little, you may remember that a microbe is considered to be pathogenic if it causes any kind of disease at all, or has the ability to cause disease under the right circumstances. Severity of the disease and our ability to treat it does not change how pathogenic a microbe is, because it's an all-or-nothing term. If it can cause disease, it's a pathogen, and if it can't cause disease, it's not a pathogen. However, there is another way to measure how dangerous a pathogen is, and that is by how virulent it is. Virulence is the relative ability to cause disease. Virulence can be used to compare how dangerous or aggressive different pathogens are. For example, the swine flu strain that caused the Spanish flu pandemic of 1918 was much more virulent than the predominant flu strain of the year before, because it was able to make more people sick and, more importantly, caused far more deaths. Many characteristics can make a pathogen more virulent, including a life cycle that kills host cells, a rapid growth rate, production of toxins, interfering with normal functions of the host, the ability to evade the immune system, and resistance to antibiotics. Some of these characteristics are easier to isolate and define than others. For example, when a pathogen produces a particular toxin that is harmful to the host, or becomes resistant to a particular antibiotic, the genes and proteins responsible can be isolated and characterized. These proteins that make pathogens more virulent are called virulence factors, and the genes that encode for virulence factors are called virulence genes. Now, just because a bacteria or virus carries a virulence gene does not necessarily make it a pathogen. Virulence is a scale, and some virulence factors play a bigger role in pathogenicity than others. In addition, virulence of pathogens is very species-specific. Let's take a look at some real-world examples of species specificity and virulence. SIV, or simian immunodeficiency virus, is very similar to HIV. In certain species of monkeys, it causes a disease that is almost identical to AIDS and is extremely virulent, but it's not virulent at all to humans because it doesn't infect humans. In this case, SIV is a very virulent virus in some species of monkeys, but a harmless virus to us. It can also work the other way too, though. Humans first contracted HIV from chimpanzees, but HIV infects chimpanzees without causing any signs of disease. It is not known exactly why HIV is so virulent in humans, but is apparently not virulent at all in chimpanzees. However, it is fairly common for viruses to be non-pathogenic to their primary host species, which makes sense. If a virus can live in a host without causing disease and killing the host, it will be able to reproduce and spread much more efficiently than if it killed its host within a week or two. This is probably the main reason why Ebola virus doesn't spread very well through the human population. Ebola virus is perhaps the most virulent virus to humans. It kills over 2/3 of the people it infects, and it does it very quickly, usually in less than 2 weeks. However, killing a high percentage of people so quickly doesn't give the virus too many opportunities to spread very far, so outbreaks are usually contained to small areas and quickly die out. Let's review. Antibiotic resistance is when a microbe acquires a gene that allows it to resist the effects of an antibiotic. Overprescription of antibiotics and the routine use of antibiotics for farm animals that don't show signs of disease creates a selective pressure for antibiotic-resistant strains of bacteria. The more that antibiotics are used, the greater the selective pressure, and the faster that resistance develops and spreads. Because bacteria are able to exchange DNA with other bacteria and pick up DNA from their environment, resistance genes can be spread very quickly and even between bacterial species if there is a selective pressure. Pharmaceutical companies are currently in an arms race with pathogens to develop new antibiotics before bacteria can develop resistance to existing antibiotics. Microbes that have the ability to cause disease, no matter how mild or severe, are called pathogens. Virulence is defined as the relative ability to cause disease and can be used to compare how dangerous or aggressive different pathogens are. Many characteristics can make a pathogen more virulent, including a lifestyle that kills host cells, a rapid growth rate, production of toxins, interfering with normal functions of the host, the ability to evade the immune system, and resistance to antibiotics. Proteins that make pathogens more virulent are called virulence factors, and genes that encode for virulence factors are called virulence genes. Just because a bacteria or a virus carries a virulence gene does not necessarily make it a pathogen. Virulence is a scale, and some virulence factors play a bigger role in pathogenicity than others. In addition, virulence of pathogens is very species-specific, and many viruses are pathogenic to some species and harmless to others. In fact, it can be advantageous to the virus to infect its host without causing disease, because the living host can spread the virus faster than a host that dies very soon after being infected. As you might recall, there are two different fundamental methods that organisms use to reproduce: asexual reproduction, which is a form of reproduction from a single parent which makes a clone of itself through mitosis, and sexual reproduction, which is when two parents produce offspring with unique combinations of genes from both parents. Some multicellular animals, like corals, use both asexual and sexual reproduction, while others, like mammals, only reproduce sexually. The sequence of events in sexual reproduction starts the same way in all sexually reproducing animals with gametogenesis, which is the formation of gametes. You may remember that gametes are specialized sex cells produced by each parent for sexual reproduction. There are two types of gametes, sperm and ova. Sperm, which are the male gametes, are created during the process of spermatogenesis. Ova are the female gametes and are created during the process of oogenesis. After gametogenesis, the process of sexual reproduction takes many forms. Somehow, the male and female gametes need to combine to form a zygote, which is basically a fertilized ovum. In some animals, like many fish species, fertilization occurs externally outside the bodies of both parents, while in others, like mammals, fertilization occurs internally inside the female. Likewise, the early development of the offspring can also occur either internally or externally. Some animals, like common garden snails, are hermaphrodites, or organisms that have both male and female sexual organs. Hermaphrodites still reproduce sexually by cross-fertilizing each other's ova. Spermatogenesis is the process by which the male gametes, called sperm, are created. Now you may recall that in order to create sperm that can combine with a female gamete, each sperm must be haploid and contain only one copy of each chromosome. You may also remember that in order to create haploid gametes, a cell must go through the process of meiosis, which involves replicating its genome and then dividing not once, but twice, in order to create 4 haploid gametes from a single diploid cell. In humans, spermatogenesis takes place in the seminiferous tubules, which are an intricate system of tubules in the testes where spermatogenesis takes place. The seminiferous tubules of an adult human male can sometimes produce over 100 million sperm per day. In order to produce such a huge number of sperm, the tubules contain a very large and active population of dividing, self-sustaining germ cells, which are the cells that will become gametes. The germ cells that are found in the testes are called spermatogonia. Every day, somewhere around 25 million spermatogonia enter meiosis and become primary spermatocytes. After the first meiotic division, the cells double in number and are called secondary spermatocytes. Once the second meiotic division is completed, but before the haploid cells have had a chance to differentiate into actual sperm, the cells are called spermatids. These spermatids now have the correct number of chromosomes to be functional gametes, but they don't have the physical characteristics of sperm that allow them to swim to the ovum and fertilize it. A functional sperm looks a lot like a tadpole, but unlike tadpoles, sperm have only 3 basic components. The nucleus contains the haploid genome and not much else. The main job of the sperm is to get the haploid genome into the ovum, which will provide the other half of the genome, along with pretty much all of the cytosol, cell membrane, and cellular organelles. The acrosome is a structure that forms a cap over most of the nucleus of the sperm cell. The main job of the acrosome is to penetrate the outer layers of the ovum so that the sperm can get inside. The flagellum is a long whip-like cellular appendage that is used for locomotion. A sperm cell uses its flagellum in a whip-like fashion, lashing it back and forth to propel the sperm forward. Now you may be wondering where the sperm cell gets the energy to whip a long flagellum back and forth so vigorously. After all, the nucleus contains very little besides DNA, and the acrosome doesn't contain any mitochondria to provide energy. That just leaves the flagellum itself. Sure enough, a portion of the flagellum right here is surrounded by mitochondria that provide the ATP that powers the whipping motion. Okay, now let's see how all of this works in the context of the seminiferous tubules. As you can see from this highly magnified section, the testes are packed with seminiferous tubules, which produce millions of sperm and release them into the lumens of the tubules. The tubules are lined with an epithelial layer of Sertoli cells, which are somatic cells of the seminiferous tubules that support and provide nutrients to the various sperm precursors. The rapidly dividing spermatogonium are located at the base of the Sertoli cells. Often, when a spermatogonium divides by mitosis, one of the daughter cells will take a position between Sertoli cells and become a primary spermatocyte. The primary spermatocyte migrates away from the base of the Sertoli cells and divides to form two secondary spermatocytes. The two secondary spermatocytes continue migrating and then divide to form four haploid spermatids. After meiosis is complete and each spermatid has the correct number of chromosomes, they begin the process of differentiating into fully functional sperm with their chromosomes packed tightly in the nucleus, an acrosome capping the nucleus, and a single flagellum to propel the sperm toward the ovum. Let's review. Spermatogenesis is the process by which the male gametes, called sperm, are created. Spermatogenesis takes place in the seminiferous tubules, which are an intricate system of tubules in the testes where spermatogenesis takes place. The tubules are lined with an epithelial layer of Sertoli cells, which are somatic cells of the seminiferous tubules that support and provide nutrients to the various sperm precursors. The rapidly dividing spermatogonia, or sperm cells that are found in the testes, are located at the base of the Sertoli cells. The spermatogonium becomes a primary spermatocyte and migrates away from the base of the Sertoli cells. Then it divides to form two secondary spermatocytes, which continue migrating and then divide to form four haploid spermatids. After meiosis is complete and each spermatid has the correct number of chromosomes, they differentiate into fully functional sperm. Mature sperm have a nucleus that contains the haploid genome and not much else. They also have an acrosome cap, which will be used to penetrate the outer layers of the ovum so that the sperm can get inside. In addition, they have a single flagellum, which is a long whip-like cellular appendage that is used for locomotion. A sperm cell uses its flagellum, lashing it back and forth to propel the sperm forward. Oogenesis is the process by which the female gametes, or ova, are created. The female gamete is called an ovum. Sometimes people will refer to female gametes as eggs, but the term egg can include more than one stage of development, and the definition of an egg also changes depending on the type of organism. For example, the entire prenatal development of birds occurs inside an egg, but in placental mammals, after the egg is fertilized and starts dividing, nobody calls it an egg anymore. So, we're going to use the more precise term for a female gamete, which is ovum, or ova for the plural form. Now you may recall, each ovum must be haploid and contain only one copy of each chromosome. You may also remember that in order to create haploid gametes, a cell must go through the process of meiosis, which involves replicating its genome and then dividing, not once, but twice. In theory, this would create four haploid gametes from a single diploid cell, like it does during spermatogenesis. However, this isn't the case for human female gametes. Let's go through the process of oogenesis to see how this works and how many haploid gametes are made from a diploid cell. The diploid germ cells that have the potential to develop into ova are called oogonia. In humans, all of a female's oogonia that she will make in her lifetime are created when she's still a fetus and hasn't even been born yet. In fact, about 1 or 2 months before a baby girl is born, most of her approximately 7 million oogonia die, and the remaining surviving oogonia enter meiosis I and become primary oocytes. These primary oocytes press the pause button on their development in prophase I, after they've replicated their genomes, but before they've made the first meiotic division. They stay arrested at this stage of development for over a decade until the girl begins her first menstrual cycle. Then, for about the next 30 to 45 years, on a monthly basis, primary oocytes resume meiosis where they left off and complete the first meiotic division. When the primary oocyte does finally complete its first meiotic division, it divides the chromosomes evenly, just as you would expect. However, it does not divide its cytoplasm equally. Almost all of the cytoplasm remains in one of the two daughter cells, which becomes a secondary oocyte. The other daughter cell, which gets half of the chromosomes but very little cytoplasm, is called a polar body. The polar body is not a functional oocyte. Instead, it degenerates and dies. The formation of a polar body allows the primary oocyte to reduce its genome by half and conserve most of its cytoplasm in the secondary oocyte. The secondary oocyte still has two copies of each chromosome, so if it's going to become a fully functional ovum, it must undergo the second meiotic division. This division is also uneven, like the first one, with half of the chromosomes going to another very small degenerate polar body, and half of the chromosomes being retained by the ovum, along with almost all of the cytoplasm. In this way, the ovum achieves its haploid state while conserving as much cytoplasm as possible. In humans, oocyte development occurs in the ovaries. Each oocyte is surrounded by a number of follicle cells to create a follicle. When the menstrual cycle begins, one or maybe even a few primary oocytes begin to grow larger, and the follicle cells increase in number and cause the follicle to grow larger too. Usually, some of the developing oocytes will degenerate and only leave one follicle that will mature, but every once in a while, two or even more follicles will mature. In these cases, fraternal twins or triplets that are genetically distinct may be born. In any case, as a follicle reaches maturity, the primary oocyte completes its first meiotic division and becomes a secondary oocyte. Very soon after that, the follicle ruptures and the secondary oocyte is released into the fallopian or uterine tube, even though the second meiotic division hasn't occurred yet. The release of a secondary oocyte from the ovaries is called ovulation. Once released from the ovary, the oocyte slowly travels down the fallopian tube toward the uterus. Fertilization usually occurs in the part of the fallopian tube nearest the ovary. This is because the oocyte is only viable for up to 24 hours after ovulation. If it isn't fertilized by a sperm within this time period, then the oocyte never completes the second meiotic division, and so it never technically becomes an ovum. However, if the secondary oocyte is fertilized by a sperm, it immediately finishes its second meiotic division, releases a polar body to leave only a haploid female genome in the ovum, and then becomes a zygote as the nuclei of the sperm and egg fuse together. In summary, oogenesis is the process by which the female gametes, or ova, are created. The female gamete is called an ovum. The diploid germ cells that have the potential to develop into ova are called oogonia. In humans, all of a female's oogonia that she will make in her lifetime are created when she's still a fetus and hasn't even been born yet. By the time a baby girl is born, all of her oogonia have either entered meiosis I and become primary oocytes or degenerated and died. The primary oocytes press the pause button on their development in prophase I after they've replicated their genomes, but before they've made the first meiotic division. In humans, oocyte development occurs in the ovaries. Each oocyte is surrounded by a number of follicle cells to create a follicle. When the menstrual cycle begins, one or maybe even a few primary oocytes begin to grow larger, and the follicle cells increase in number and cause the follicle to grow larger too. Usually, only one follicle reaches maturity, and the primary oocyte in the follicle completes its first meiotic division and becomes a secondary oocyte. Very soon after that, the follicle ruptures and the secondary oocyte is released into the fallopian or uterine tube before the second meiotic division has occurred. The release of a secondary oocyte from the ovaries is called ovulation. Once released from the ovary, the oocyte slowly travels down the fallopian tube toward the uterus. If the secondary oocyte is fertilized by a sperm, it immediately finishes its second meiotic division, releases a polar body to leave only a haploid female genome in the ovum, and then becomes a zygote as the nuclei of the sperm and ovum fuse together.
Did you know you started out as a single cell, but now you have hundreds, maybe even thousands of different types of cells in your body? This is because as the fertilized egg begins to develop, its cells are assigned different roles. Cells inside the embryo become all the different cells in your body, but some cells are located outside the developing embryo and are called extraembryonic cells, or membranes. While this may look like a big word, if you break it down, it's pretty easy to understand. Extra, meaning outside of, and embryonic, referring to the developing embryo, so extraembryonic means outside the embryo. You can think of the extraembryonic membranes as a protective bubble that surrounds the developing fetus, insulating it from the outside environment. This bubble has 4 different parts to it. First, the layer closest to the embryo. This one is called the amnion. The amnion surrounds the embryo and is filled with amniotic fluid. You can think of it like a small tiny baby, about the size of a peanut. Now put that little peanut into a balloon. The balloon is like the amnion surrounding the baby, or in this case the peanut. Then fill the balloon up with water. The water is now surrounding the peanut, protecting it. The water is like the amniotic fluid. Together, the amnion and the amniotic fluid have a few different jobs. 1. Protect the embryo from injury, like if mom's belly gets bumped. 2. Provide a safe space for the embryo to move around, giving him or her plenty of free space to practice kicking mom's belly button. 3. Help the fetus maintain a normal temperature and supports proper lung development. Did you know when in the uterus the fetus is actually breathing in amniotic fluid? The first breath of air doesn't come until after birth. 4. Transfer waste from the fetus to the mom's bloodstream so her body can get rid of it. Did you know a fetus releases urine? Kind of makes you appreciate your mom that much more. Our next structure is called the allantois, which is the precursor to the umbilical cord. This would be like attaching a thread to the peanut and the other end of the thread to the balloon, kind of like an anchor on a boat. This structure actually doesn't stick around the whole time. It is only present during the beginning of fetal development because these cells form the precursor to the umbilical cord blood vessels. Once the umbilical blood vessels are fully formed, the allantois is no longer needed. The yolk sac comes next. You know that yellow substance you see in the center of an egg? Well, in a fertilized egg, that yolk sac helps to nourish the developing embryo or fetus, at least in most mammals. In humans, however, we have something called the placenta to provide nourishment to the fetus, so our yolk sac is small and eventually becomes part of our digestive and respiratory systems. And that brings us to the last layer, the one furthest away from the embryo, the chorion. This is like surrounding our balloon with another balloon, so you have a double layer. The chorion is the outermost layer. It connects the amniotic sac to the placenta. This connection is essential to development because the fetus relies on the placenta for nourishment from the mother. Not only does the chorion provide the connection between mother and fetus, it also secretes a hormone essential in helping the uterus to maintain pregnancy during the first trimester. This hormone, human chorionic gonadotropin, HCG, is what all those at-home pregnancy tests detect to let a woman know whether or not she is pregnant. Once the placenta is fully formed, it takes over the production of hormones. The placenta, along with the umbilical cord blood vessels, is the fetal lifeline, which is the essential connection to its mother. Without these, the fetus would not be able to obtain the nutrients it needs to grow. The placenta is a mixture of both extraembryonic and maternal tissue. Remember the chorion we talked about earlier? Well, it develops finger-like projections, called chorionic villi, which embed themselves into the uterine tissue of the mother. As they do this, they cause the uterine tissue to bleed, and small pools of blood are created. The villi extend into these pools where they gather nutrients from the mother's blood. Essentially, they are creating a surface allowing for the exchange of nutrients, oxygen, and antibodies because inside the villi are tiny blood vessels, or capillaries, that transport these materials to the fetus. The placenta provides an attachment site connecting the mother to the fetus. This is essential for the two main functions of the placenta: the transport of nutrients, oxygen, antibodies, and other materials to the fetus, and the removal of waste from the fetus. Another important exchange route is the umbilical cord, or more importantly, the vessels inside the umbilical cord. The umbilical veins and arteries are larger vessels that also allow the transport of essential nutrients and other materials to and from the fetus. These vessels come from the fetus and go to the placenta. At all times, the maternal and fetal blood vessels are separated, with the placenta being the neutral zone between the two. This is where the fetus can drop off waste and pick up nutrients, antibodies, and oxygen from the mother. So there you have it. All of these layers help protect and nourish the developing fetus. They are his or her temporary housing, complete with room service. The extra embryonic membranes include the amnion, the layer that surrounds the fetus and is filled with amniotic fluid to help protect the fetus, maintain body temperature, support lung development, and provide space for it to move around. The allantois, the precursor to the umbilical blood vessels. The yolk sac, to provide nourishment, and in humans becomes part of the digestive and respiratory systems. And finally, the chorion. This layer is the interface between the fetus and the mother. It gathers nourishment from the mother's blood via small finger-like projections called chorionic villi, and secretes the hormone HCG to maintain pregnancy. The chorion connects the fetus to the placenta. The placenta is made up of maternal tissue and, along with the umbilical arteries, transfers nourishment, oxygen, and antibodies to the fetus.
Now, the principles of genetics have existed for thousands of years. We just never called it genetics. Think about farmers trying to cultivate hardier, more delicious crops, or horse breeders who want to develop faster, better racing horses. And they would do this by only selecting the best horses in each generation for breeding. Without realizing it, early farmers and breeders practiced genetic selection before we really knew what genetics was. Thousands of years later, Gregor Mendel's studies of pea plants formed the foundation upon which modern genetics was built. But rather than read about pea plants, though, let's go visit our modern-day scientist, Adrian, to see these principles in action. Adrian is excited because he's just discovered a brand new species of hamster that can fly. Now, he thinks this is just going to be a scientific gold mine. So he decides to drop everything that he's been working on and study this new species. Let's take a minute to talk about terminology so we can understand Adrian's work. Now, remember that a gene is just a piece of DNA that encodes a gene product. So we can also refer to this DNA code as genotype. But it'll be difficult to track the genetic changes occurring in our flying hamsters. It'd be a lot easier to monitor observable traits. So recall that the gene products are what provides functionality in the cells and for the organism. So in performing these various functions, gene products are producing observable traits. And these observable traits are referred to as phenotype. So what this means then is, if I was to make a change in the genotype, let's say we change this T and let's mutate it into an A. If I was to change the genotype, a change in the genotype will in turn cause a change in the phenotype, which then can be observed by us as scientists. Now, geneticists are interested in heritable traits. A mutation in the DNA can be passed on to offspring, and in that way, this change in the DNA code has become a permanent change in the genome of this organism. However, not all traits are heritable. For instance, if one of our experimental hamsters happens to break a leg, that hamster won't pass on a broken leg to its offspring. This is not a heritable trait.
Because of the link between genotype and phenotype, you can see how farmers could select for genetic improvements in their crops and their livestock by selecting for phenotypic changes occurring over time. The different types of corn that we see in the supermarket today, yellow, blue, white, etc., are good examples of genetic manipulation by cultivation. Okay, so I think we have genotype and phenotype pretty square, but we need a few more terms to be able to really understand the full extent of genotype. Now, genes are located on chromosomes, and this makes sense because we just got through saying how a gene is made up of DNA, right? And a chromosome is just a discrete structure of DNA that's found in the genome. By observing the chromosomes under a microscope, Adrian determines that the flying hamster, like most animals, is a diploid. And furthermore, he determines that flying hamsters have a total of 6 chromosomes. So hopefully all those words sort of ring a bell from our previous lessons, but here's another way of looking at it. A chromosome is defined by a specific sequence of DNA, which in turn means it's defined by a specific set of genes. However, because the organism is diploid, it has 2 versions of each chromosome. Each of these versions is called a homologous chromosome or a homolog. So to summarize, our flying hamsters have 3 different chromosomes. I got chromosome 1 here, chromosome 2, and chromosome 3. And because the flying hamster is a diploid, that totals up to 3 pairs of homologous chromosomes in each hamster cell. Now we refer to a specific location on a chromosome as a locus. Now in general, a locus is going to refer to a specific gene on that chromosome, although it could just be referring to a specific location on the chromosome, not necessarily a gene. So we have a pretty good grasp on traits and chromosomes now, but we need a better way of talking about differences in DNA. Adrian has collected hamsters from a bunch of different geographical areas, and he notices that hamsters from one area are brown and those from this other area are white. He hypothesizes that the same gene encodes coat color in each animal, but that different versions of the gene account for the different colors. So we call each version of a gene an allele. So in this case, we would have 2 different alleles. We'd have a brown allele and we'd have a white allele. Then the differences in these alleles is what encodes the different coat color in the 2 different animals. So if Adrian's hypothesis is correct, a single gene determines the coat color, and one allele encodes a brown coat, and the second allele encodes a white coat. So to summarize, we've learned that genotype describes the DNA sequence associated with a locus. A locus is a specific location on a chromosome, and it's often used in reference to the location of a gene, although it also can refer to a specific physical location on the chromosome that's not associated with a gene. The genotype of an organism determines its phenotype, or an observable characteristic of the organism. Different traits, such as a difference in coat color in our flying hamsters, for example, are determined by different versions of the same gene, which are called alleles. So stay tuned to see if Adrian's hypothesis about flying hamster coat color is correct. Let's check back with our intrepid scientist, Adrian. When we last saw Adrian, he was trying to determine the genetic basis for flying hamster coat color. To understand his experiment, though, we'll need to learn a little bit more about how the genetic makeup of these hamsters impacts the coat color. Now, in a diploid organism, we know we have 2 copies of every gene. We also know that different versions or alleles of the gene may exist. Let's say we represent the coat color gene with the letter B. That means I could represent 2 different alleles with either the uppercase or lowercase b. Because the flying hamster is diploid, it also means that it could have 2 copies of the same allele. I could have 2 big B's, or I could have 2 little b's in the same organism. I could also have a scenario where I have one of each kind of allele in a given hamster. We refer to a locus that has two different alleles as heterozygous and one that has two copies of the same allele, homozygous. Now, let's remember that Adrian hypothesized that the coat color of his hamsters was determined by a single locus. So let's apply our new knowledge of alleles so that we can refine this hypothesis a little bit. Now let's say that the big B allele represents brown, and we'll have the little b represent white. So let's see then what's going to happen in the case of the three different genotypes that a hamster can have. So it should be fairly obvious that if I have two brown alleles, I should produce a brown hamster. If I have two white alleles, I should produce a white hamster. But what happens when I have one of each kind of allele? The heterozygous state is resolved by considering the relationship between alleles. Adrian decides to breed a brown hamster with a white one. He finds that the brown and white hamster mating only produces brown hamsters. Adrian has encountered one of the central principles of genetics. While there may be more than one allele associated with a given gene, one trait is sometimes dominant over the other one. Since the brown and white populations of hamsters always produce the same color, so for instance, if I have a brown hamster mating with a brown hamster, we always see brown hamsters. And by the same token, if I have white hamsters mating, they always are producing white hamsters in these specific populations that he's isolated from the wild. So we know that these parent hamsters came from populations that are referred to as true breeding. What that means is, in terms of the trait we're talking about, brown hamsters that are mated with brown hamsters always produce more brown hamsters. White hamsters mated with white hamsters always produce more white hamsters. Since that's the case, we can infer that the genotype of these animals are big B for the brown one, and little b for the white one. Therefore, all of the progeny from this cross should have a big B, little b genotype. The fact that all of these hamsters in the progeny generation are brown indicates that the brown allele is dominant over the white allele. So a dominant trait is referred to as a trait which is expressed in the heterozygous state. That means that a recessive trait is a trait which is not expressed in the heterozygous state. Geneticists usually represent the dominant allele with a capital letter and the recessive allele with a lowercase letter. So this experiment has provided Adrian with some data that supports his hypothesis that flying hamster coat color is determined by a single locus. And we'll see how further experimentation can provide more support for this hypothesis. In this lesson, we've learned that an organism homozygous at a given locus possesses two copies of the same allele. For instance, if we're talking about gene B, big B, and little b are both homozygous genotypes. An organism heterozygous at a given locus possesses two different alleles. For instance, big B, little b would be an example of a heterozygous genotype. A dominant trait is the trait which is expressed in a heterozygous state. A recessive trait is the trait which is not expressed in a heterozygous state.
Long ago, people's understanding of heredity, or how traits are passed down from parents to offspring, was a little fuzzy. At the time, they believed the traits were blended together, so if you had a giant nose and your partner had a small nose, your child would have a medium nose. Of course, this isn't exactly right, but it was a reasonable explanation for the time period. Fortunately for us, the brilliant Austrian monk and scientist named Gregor Mendel came along. Gregor Mendel was fascinated by heredity. He conducted experiments in the mid-1800s using pea plants. Pea plants, as it turns out, come in many different forms. You've got pea plants with purple flowers, some with white flowers, some with green seeds, some with yellow seeds. Well, you get the idea. Mendel used all of this diversity within the pea plant to figure out some pretty neat genetic stuff, including the law of segregation, which is also referred to as Mendel's first law. Now, I know you're eager to hear all about this law, but you're going to need some background information first, so we'll have to come back to it. Mendel conducted numerous experiments, but let's hone in on one in particular so we can get some vocabulary cleared up. Mendel crossed a purple-flowered pea plant with a white-flowered pea plant, and all of the offspring were purple. That certainly didn't fit the blending ideas of the time, did it? But here's where it gets weird. When he crossed the purple-flowered offspring together, the next generation had purple and white-flowered plants. What? How in the world did purple-flowered pea plants produce white-flowered pea plants? A mystery. Today we know that traits like purple and white flowers are the result of different alleles, or different forms of a gene. Within your cells there is a bunch of DNA coiled up. This DNA has all of the written instructions on how to make you, well, you. If you were to check out your DNA, you'd see sections that have genes, and these genes tell your body to make a particular protein. Pea plants are the same. For example, pea plants have genes that tell the plant what color to make the flowers. Now, some pea plants have genes to say, make the flower purple, and some have genes that say, make the flower white. The different forms of these genes are called alleles. Back to our mystery. So how did those purple-flowered pea plants make white-flowered pea plants? Each pea plant has 2 alleles for flower color, 1 allele from each parent. In addition, there are 2 possibilities, or 2 alleles, for flower color in pea plants, purple and white. Scientists use letters to represent these alleles. Let's use an uppercase P for purple and a lowercase p for white. Here are the different combinations and consequent flower colors you'd get if those alleles came together. You can have uppercase P, which results in a purple flower. You can have uppercase P, which results in a purple flower. And you can have lowercase p, which results in a white flower. You may have noticed that whenever you have an uppercase P, the flower is purple. That is because the purple allele is dominant over the white allele. Scientists represent dominant alleles with a capital letter, hence the uppercase P. This means that it covers up or masks the white allele. The white flowers are recessive because they are covered up by the dominant allele. Recessive alleles are represented by lowercase letters, hence the lowercase p. So even though uppercase P, lowercase p is purple, it's carrying the allele for white flowers, and this is why 2 purple plants can produce white-flowered plants. Still confused? Don't worry, we'll explain further. Finally, we're here. Eggs and sperm are referred to as gametes, and gametes are produced during meiosis. Although we won't get into the details, know that meiosis is a process that halves the genetic information. Why would that need to happen, you ask? Well, if each egg and each sperm have half of the genetic information, then put them together and you have all the genetic information you need. The law of segregation means that during meiosis, each gamete will only receive 1 copy of the genetic material. Remember, the cells that aren't gametes have 2 copies, 1 from each parent. Let's say we had the pea plant with the alleles uppercase P, lowercase p. According to the law of segregation, 1 gamete would receive the uppercase P, which codes for purple flowers, and 1 gamete would receive the lowercase p, which codes for white flowers. Are you starting to see how 2 purple plants could produce a white-flowered plant? Let's finish up this lesson with how to apply the law of segregation and to show you exactly how 2 purple plants can produce a white-flowered plant. In that second generation, Mendel crossed 2 purple-flowered pea plants. Those pea plants carried the allele for white flowers, so each was uppercase P, lowercase p. Let's use a Punnett square, which is a tool to help you predict the genetic makeup of offspring. Remember, during meiosis, each allele goes into a different gamete, so you can separate them out and use the Punnett square to see what possible offspring you'd get. Step 1. Draw a box with 4 squares. Step 2. Segregate, or split up the alleles, just like in the law of segregation, and place them next to each box. Drop the alleles into the boxes, both vertically and horizontally. Now you can see you are left with several possibilities. There's a 25% chance your offspring will be purple with uppercase P, a 50% chance your offspring will be purple but carry the allele for white flowers or uppercase p, and a 25% chance your offspring will be white or lowercase p. So there you go. You just witnessed how purple flowered pea plants can magically produce a white flowered pea plant. Gregor Mendel reshaped the way scientists understood heredity, or how traits are passed down from generation to generation. He did so by studying pea plants and observing dominant and recessive traits. Although it appeared that some traits magically reappeared, it was just that some pea plants carried the recessive allele, or form of a gene. Mendel's first law, or the law of segregation, states that each allele for a trait will segregate, or separate, from its partner, so each gamete, or sex cell, will only have one copy of the genetic material. This works out nicely because we don't want offspring to have too many copies of anything. The law of segregation makes predicting offspring for pea plants pretty easy using Punnett squares.
Given the burgeoning success of flying hamster research and our fascination with the little rodents, you and I decide we need to do our senior thesis in Adrian's lab. Now, a lot of the other members of Adrian's lab are focused on studying the genetics and molecular biological function of specific genes and proteins in the hamsters. I think it would be interesting to study the genetics of an entire hamster population rather than just studying a single gene. The study of the genetic variation within a population is called population genetics. Now, since we seem to be working together so well, let's go ahead and collaborate on our population genetics projects. Okay, well, let's think about the mechanism of evolution before we get started. And that sort of begs the question, what is evolution? Well, for now, let's think about evolution as a change in the genetics within a population over time. Now, as we learn more about population genetics, we'll be able to refine this definition a little bit more. While having lunch with some other student researchers in our building, we learn about their theory regarding the evolution of traits. They tell us they believe the evolution of new anatomical structures is driven by necessity. Similarly, if an organism doesn't need a structure, that structure will become smaller and less developed because of disuse. The idea that characteristics acquired by an organism during its lifetime can be passed on to its offspring is attributed to the scientist Jean-Baptiste Lamarck. This theory is referred to as heritability of acquired characteristics, or also known as Lamarckian inheritance. Under this theory, a giraffe that stretches its neck to reach leaves and higher branches of trees would pass on stronger neck muscles to its offspring, which would enable longer reach and eventually would result in the evolution of giraffes with longer necks. Similarly, the theory proposes that the disuse of the small toe in the feet of some mammals, such as pigs, caused it to shrink over time. We might say that the theory of acquired characteristics is akin to the adage that necessity is the mother of invention. The need for a trait drives the evolution of that trait. Now, our classmates' theory seems pretty logical, but it's always a good idea to consider alternative possibilities. From our previous studies, we know that variation exists in a population of organisms. Mutations can introduce new alleles, which may or may not provide improved functionality over the previously existing ones. Random assortment and crossing over create new combinations of alleles of different genes. And for a given situation, new genetic combinations could theoretically provide a competitive advantage to that individual. Let's consider an example. Suppose there is variation in the size and strength of flying hamster wings in an island population. Hamsters with stronger, larger wings can fly farther than the ones with smaller wings. However, since food and mates are plentiful on the island, that's not really that important. Now, suppose the volcano in the middle of the island suddenly erupts, and the flying hamster habitat is completely destroyed. The hamsters with larger wings are strong enough to fly to a new island, while the weaker ones are stuck on the ruined island or simply can't make it all the way to a new piece of land. In the end, the significant alteration to the environment selected for a new population of hamsters with large, stronger wings. These are the basic principles of the theory of evolution that was proposed by Charles Darwin. He reasoned that populations, rather than individuals, evolve. This evolution is possible because genetic variation exists within a population. Individuals with different genotypes in the population survive and reproduce at different rates. The agent which determines a differential rate of reproduction is called natural selection. Note the difference between Darwin's and Lamarck's theories. Lamarck believed that the need for a new trait would promote the evolution of that trait, whereas Darwin proposed that evolution occurred through the selective pressure for a pre-existing trait in a population. Both theories seem kind of reasonable. How can we determine which theory most accurately describes what's actually happening in nature? What if we tie the wings of some flying hamsters so they can't fly? The theory of acquired characteristics predicts that since the wings aren't being used, they will progressively become smaller and possibly even disappear over several generations. As it turns out, a scientist named August Weismann conducted a similar experiment to disprove Lamarck's theory. Weismann repeatedly cut off the tails of mice over several generations. He found that the tails of mice in subsequent generations neither disappeared nor grew appreciably shorter. Today, Darwin's theory of evolution is widely accepted as the principle which governs population genetics. As we begin our study of flying hamster population genetics, we will see how the principles of Darwin's theory can be used to predict the allelic, genotypic, and phenotypic frequency within a population. In summary, the study of the genetic variation within a population is called population genetics. Evolution is a change in a characteristic within a population over time. The theory of acquired characteristics states that characteristics acquired by an organism during its lifetime can be passed on to its offspring. It is also sometimes called Lamarckism. The principles of Darwinian evolution include the ideas that populations, rather than individuals, evolve. Evolution is possible because genetic variation exists within a population, and individuals with different genotypes in the population reproduce at different rates. Natural selection is the agent which determines a differential rate of reproduction.
In another lesson, you learned about evolution, which explains that species living today are descendants of species from long ago. Charles Darwin observed this phenomenon and described it as descent with modification. Evolution occurs on a large scale over a very long time, but it is not unlike you and your parents. You are a descendant of your parents, but in a modified version. You are not exactly like them, even though you came from a combination of their genes. Evolution is a hot topic these days, but despite the controversy, evolution is in fact the core theme of biology and has been studied extensively enough to be called a theory. In science, a theory is much more than a simple explanatory idea. A scientific theory explains natural phenomena and has been repeatedly confirmed through experiments and observations. Evolution is a scientific theory because not only has it been studied and tested, but we also have several different sources of evidence to support it. The field of paleontology is important to the support and understanding of evolution. This is the study of prehistoric life, including fossils, footprints, and past climatic events. As organisms die, they become part of the ground. Often they leave behind bones and imprints, which allows us to see what they looked like millions of years later. Because new layers of ground and fossil form on top of old layers, this fossil record forms a sort of biological timeline. The fossil record shows us the sequence of historical changes in organisms. We can visually see how organisms evolved over time, but we can also use radiometric dating to determine the age of rock and fossils. Of course, prehistoric organisms that did not form fossils are hard to study, so we can't know everything about the history of life on Earth. But we do know some really interesting things, like that whales and dolphins likely evolved from four-legged land animals. How do we know this? One piece of evidence that supports this is the body structure of both animals. They have flippers in the front that may have evolved from front legs, but they also have small internal back limbs that likely evolved from legs once used on land. Other evidence in support of evolution comes from biogeography, which is how species are distributed across Earth. This is what first suggested to Charles Darwin that species evolved from a common ancestor. Darwin observed the animals of the Galapagos Islands and noticed that they were very similar to the animals on the South American mainland, but very dissimilar to animals on other islands that had similar environments. From this, he concluded that the animals on the Galapagos had migrated from South America and after a long period of time became new species as the populations adapted to their new environment. This also helps explain why there are no polar bears in the Antarctic and no penguins in the Arctic, despite both places being very icy and cold. It makes sense that organisms that are related to each other will have similar features. You may have your mother's hair or your father's eyes, but you also have 2 arms, 2 legs, a mouth, and a nose, just like other primates such as monkeys and apes. This similarity in characteristics from a common ancestor is called homology. You can easily see this type of similarity if you compare the forelimbs of mammals. Humans, dogs, whales, and bats all have the same skeletal elements in this part of their body. The difference comes in the function of that limb. A human arm serves a much different function than a bat's wing, but the similarity suggests that at one time we shared a common ancestor that had similar structure, and each organism's forelimb evolved to meet different environmental demands. Because these structures come from a common ancestor but have different functions, we call them homologous structures. Embryology, the study of embryos, is another way we can compare evolutionary relationships in anatomy. Many organisms share similar structures that are only present during development, and these similarities suggest a shared common ancestor long ago. For example, all vertebrate embryos have a tail at some point during their development, though I bet you'd be hard-pressed to find a tail on an adult human. Vertebrate embryos also have throat pouches that have different functions in adulthood. For humans, they become part of the throat and ears, but for fish, they become gills. Comparative anatomy can be very useful, but only to a certain point. The relationship between organisms that are very distantly related can be difficult to link with anatomy, so we use molecular biology instead. This field uses biological change at the molecular level to describe the evolution of organisms. So instead of looking at anatomical homology, biologists look at DNA homology. Just like you and your siblings will have very similar DNA because you are related, so will organisms that inherited their DNA from a long ago common ancestor. The degree of difference tells us how different the ancestor is. For example, your DNA is very similar to that of your parents because you inherited it from them. Your children will also have DNA very similar to yours, but less similar to your parents because they are not as closely related. Likewise, their children will have DNA that is even less similar to your parents because now there are multiple steps in the genetic lineage. The same happens over evolutionary time periods, so comparing DNA can tell us how closely related organisms are and maybe even how long ago they shared the ancestor from which they are descended. The support for the theory of evolution is overwhelming. Many scientific fields contribute to the study and understanding of how populations change over time to meet the demands of their environments. Paleontology tells us how species change through time by studying the fossil record. Biogeography tells us how species are distributed geographically, which helps us understand why similar environments do not always support the same species. Comparative anatomy allows us to visually compare the homology of organisms so that we can see how different environmental demands may have led to similar structures with different functions. And when organisms are very distantly related, we can use molecular biology to understand evolutionary change and homology on the molecular level. Generally, evolution is not something that happens quickly. On average, it takes about 6.5 million years for a new species to develop. It takes such a long time for populations to evolve because the changes occur slowly over many generations. But that doesn't mean that all evolutionary changes happen at the same rate. In fact, relatively speaking, evolution can happen quickly, 4,000 years, slowly, 40 million years, and everything in between. Scientists are able to study how quickly or slowly populations evolve through different means, but most of the information we have comes from the fossil record. The fossil record tells us a lot about the history of life on Earth because as organisms die, they become part of the ground. They may leave behind bones, imprints, or even footprint fossils that we find millions of years later. And since each layer of ground builds on top of the one below it, the fossils create a timeline through the Earth, essentially telling us a story of how things changed over time. Often, new species come about slowly in the fossil record as they evolve over time. Take these butterflies, for example. Two new species come about from a common ancestor and then slowly change over time. You can see the gradual change as each species continues to diverge from its original ancestor on the left. Sometimes, though, fossil species appear quite suddenly and then remain unchanged through many layers of the fossil record. These species may even disappear just as suddenly as they came about, leaving scientists with many questions. These types of events in the fossil record are called punctuated equilibria. They describe long periods of equilibrium, little change, with abrupt episodes of speciation. Let's look at the butterflies again. This time, the common ancestor on the left suddenly gave rise to two new species that appear to have little change once they appear in the fossil record, reaching a sort of evolutionary equilibrium. It may be tempting to make assumptions about the species that came about suddenly and changed very little, but we need to be careful. The new species may not have originated as suddenly as the fossil record suggests because shorter periods of time cannot be easily distinguished between the layers. For example, if the butterflies in the punctuated equilibrium model had most of their changes occur within the first 50,000 years of existence, a relatively short time period in evolutionary standards, these would not show up well in the fossil record. Additionally, if the change that led to the new species occurred in a remote location or from a small isolated population, the chances of those fossils being discovered is quite low. So, while the fossil record does help us understand where some new species came from, it can be misleading because we do not always have all of the details about the changes that occurred. The fossil record is very useful for understanding rates of evolution, but we can also use molecular data to track evolutionary time. Some parts of an organism's genome, or all of its genetic information, accumulate changes at constant rates. This allows us to compare genomes of different organisms to determine evolutionary relationships. For example, just by looking at them, you might think that sharks and tuna are more closely related than bats and dolphins. Actually, the reverse is true. By comparing their DNA, we know that bats and dolphins are more closely related because their homologous genes, the ones that are similar in location, structure, or function, are more alike than the homologous genes of sharks and tuna. When we know the rate of change that occurs for a gene, we can calibrate a molecular clock. This technique compares the number of nucleotide differences against the dates of evolutionary branch points that we get from the fossil record. What this allows us to do is estimate relative dates of evolutionary events that do not appear in the fossil record itself, because your DNA is like a set of instructions and it changes very little over time. That one small change can make for huge differences, since it's the order of the nucleotides themselves that makes each DNA strand unique. This is not a foolproof system, though. The rate of change may vary in different genes at different times and among different organisms. Sometimes this can be accounted for by calibrating the molecular clock to multiple genes instead of just a few. Evolution is a long process. On average, it takes about 6.5 million years for a new species to develop from its ancestor. However, this is only an average, and evolution can occur rapidly, as little as 4,000 years, or very slowly, over 40 million years. The fossil record provides us with a lot of information about how life has evolved on Earth. By looking at the order that fossils occur in the ground, we can create a timeline of evolutionary events. The fossil record is not complete, though, and sometimes we see species evolving gradually, while other times they seem to appear out of nowhere. These sudden appearances in the fossil record, followed by little change, are called punctuated equilibria because the fossil record is punctuated by the new species, but then it appears to remain in evolutionary equilibrium after showing up. One explanation for this phenomenon is that the fossil record is somewhat incomplete. Changes that occur over relatively short periods of time or that happen in small, isolated populations may simply go unrecorded in geologic history. In addition to the fossil record, we can create molecular clocks for organisms. This technique compares the number of nucleotide differences against dates of evolutionary events. This is one way that we can estimate and fill in events that are missing from the fossil record, gaining a better understanding of the rate of evolutionary change for different species.
We've learned that population genetics is the study of genetic variation within a population. Now, if you recall, genetic variation arises from differences in the genetic code and can be measured as observable differences in the characteristics of an organism. For instance, a mutation can alter the genotype of a gene and create a new allele. That allele, in turn, can result in the creation of a new characteristic or phenotype. That means that, at its core, population genetics is a study of the allelic, genotypic, and phenotypic variation within a population. This information can then be used to determine whether or not a population is evolving and make a prediction regarding the cause of the evolution. For our research project, let's see how the traits we've been studying, like coat color and fire breath, affect a wild flying hamster population. Instead of analyzing the progeny of a single cross, like we did earlier with Punnett squares, this time we're going to try to analyze the genotype and phenotype of all of the progeny of an entire population of flying hamsters. Adrian suggests that we use the Hardy-Weinberg equation for our studies. The Hardy-Weinberg equilibrium equation describes genotypic frequency in a population. When a population is in Hardy-Weinberg equilibrium, allelic and genotypic frequency can be predicted by the equation. However, for a population to be in Hardy-Weinberg equilibrium, several criteria must be met. First, mating must be random. The genotype of individuals at the gene being studied can't affect mate choice. Second, the population size must be very large, virtually infinite. If you're considering a population of 4 individuals, random death of a single individual would significantly alter the genetic makeup of that population. In contrast, a random death in a population of, say, 10,000 individuals would not. The third criteria for Hardy-Weinberg equilibrium is that there can be no migration between populations. If individuals enter or leave the population being studied, the genetic makeup of the population is altered. Fourth, there can be no mutations. Mutations could introduce new alleles or alter the effect of alleles currently being studied. Finally, natural selection cannot affect the alleles under consideration. If individuals with a specific genotype are more likely to survive and reproduce, the genetic makeup of the population can be significantly altered. If it seems like an awful lot of restrictions for this equation to work, you're right. Would all of these conditions ever be met in a population that we'd find in the wild? The answer is almost surely no. If that's the case, then really what's the point in learning this fancy equation? The answer is that using this equation can tell us whether or not an evolutionary agent is affecting a population we're studying. That is, allele frequency within a population should not change from generation to generation unless an evolutionary agent changes it. Therefore, if we can establish that a population in nature has deviated from Hardy-Weinberg equilibrium, we can conclude that the genetic makeup of the population is changing. And maybe we can even conduct further studies to infer the major evolutionary agent or agents at work. In summary, population genetics is the study of the allelic, genotypic, and phenotypic variation within a population. The Hardy-Weinberg equilibrium equation describes genotypic frequency in a population. The prerequisites for a population in Hardy-Weinberg equilibrium are random mating, large population size, no migration between populations, no mutations, and no natural selection. The Hardy-Weinberg equilibrium equation can be used to determine whether or not the genetic makeup of a population is changing. So we've seen how the Hardy-Weinberg equilibrium equation can be used to test the evolutionary status of a population. Now, an evolutionary agent is just any force which alters the genetic structure of a population. If no evolutionary agents are affecting a population, the population is in equilibrium, because allelic and genotypic frequency are not changing. By using the Hardy-Weinberg equilibrium equation to analyze the allelic and genotypic frequencies within a population of flying hamsters, we were able to determine that the coat color trait is in equilibrium. However, the real power of the Hardy-Weinberg equation is identifying populations that are not in equilibrium. When a population fails to comply with the Hardy-Weinberg equilibrium predictions, we can try to infer what evolutionary agent or agents is affecting the population. By generating an educated guess or hypothesis, we can provide the basis for further experimentation to explore the evolution of that population. Well, let's go out into the wild and start examining flying hamster populations. Let's start with the population where we found the different tail colors. You know, I'm thinking it'd probably be a good idea to check our lab notebook to refresh our memory about this trait. Alright, according to our notes, the tail color trait was an example of incomplete dominance, meaning the heterozygote exhibits a phenotype that is partway between the two homozygotes. We used big B to represent the blue tail allele and big Y to represent the yellow-tailed allele. Hamsters with a big B genotype have blue tails, those with a big B, big Y genotype have green ones, and those with a big Y genotype have yellow ones. We could make a hypothesis that the population is in Hardy-Weinberg equilibrium. That will allow us to use the Hardy-Weinberg equation to determine whether that is true. If the population satisfies all of the requirements that we've discussed previously, our observed data should match the numbers predicted by the Hardy-Weinberg equilibrium equation. Now, after walking around the area counting hamsters, we determined that the population contains 526 blue-tailed, 42 green-tailed, and 432 yellow-tailed hamsters. Since each blue-tailed hamster contributes 2 big B alleles, and each green-tailed hamster contributes 1 big B allele, there are 1,094 big B alleles out of a gene pool of 2,000 total alleles. That means that P equals 0.547. Now, since we determined earlier that P plus Q equals 1, we can therefore say that Q equals 0.453. So let's insert these values into our handy-dandy Hardy-Weinberg equilibrium equation. We want to know the frequency at which each phenotypic class should occur in the population. Each value in this equation represents one of those classes. Therefore, if we solve for p squared 2pq, and q squared, we can determine those frequencies. And since we're dealing with a population of 1,000 hamsters, we can predict how many of each type to expect if the population is in equilibrium. The equation predicts we should see 299 blue-tailed, 496 green-tailed, and 205 yellow-tailed hamsters. Since the number of hamsters in each category that we observed is different than what we expected, an evolutionary agent may be affecting the population. But some variation beyond the predicted values has to be expected. For instance, just because I don't get exactly a 50-50 split when flipping a coin, that doesn't mean that there isn't a 1 in 2 chance of getting heads or tails. The question is, how can I tell the difference between random variation between data sets and data sets which are significantly different? To answer this question, we need to use statistics. The chi-square test is a statistical test commonly used to determine if observed values are significantly different from expected values. If we evaluate our data with this test, we find that the difference is significant. That means we can reject our hypothesis that the population is in Hardy-Weinberg equilibrium and predict that one or more evolutionary agents are altering the population. By conducting further studies, we may be able to make a hypothesis to identify the major evolutionary agents affecting the population. For instance, while we were doing our counts, I noticed that the blue-tailed hamsters seem to prefer to mate with other blue-tailed hamsters, and yellow-tailed hamsters tend to mate with other yellow-tailed ones. This observation may be an indication that hamsters in the population do not mate randomly. Non-random mating alters genotypic frequency, which in turn alters the phenotypic composition of a population. Okay, great. So that could make for a really interesting research project. Although we can't exclude other possibilities simply based on this observation, we can perform some controlled experiments to provide more evidence to support our hypothesis. But let's do that later. For now, let's continue our field research. Let's shift our studies to the fire-breathing trait. Now, if we refer to our lab notebook again, we can see that the fire-breathing trait is a recessive autosomal trait. We represented the non-fire-breathing allele with big F and the fire-breathing allele with little f. So let's count the hamsters in the population. However, this time, we'll need to perform a genotyping test to distinguish big F from big F little f individuals. Those efforts yield the following results: 119 big F, 265 big F little f, and 616 fire-breathing hamsters. As before, we can use this data to calculate the allelic frequency. 119 big F hamsters translates to 238 big F alleles. And 265 big F little f hamsters contribute 265 big F alleles to the gene pool. That means that out of a 2,000 allele gene pool, 503 are big F. We can simplify this information as P equals 0.252. And plugging that P value into our allelic equation, P plus Q equals 1, we find that Q equals 0.748. Now, if we plug those values into the Hardy-Weinberg equilibrium equation, we predict we should see 63 big F, 377 big F little f, and 560 fire-breathing hamsters. A statistical analysis of the expected versus observed data tells us that we can reject the hypothesis that the population is in equilibrium with respect to the fire-breathing gene. I think our observations while counting and typing the hamsters in the population again provide a strong indication regarding at least one evolutionary agent which may be affecting the population. We saw more than one fire-breathing hamster use its breath to defend itself from predators, while their poor non-fire-breathing counterparts were eaten. What evolutionary agent do you think could be working here? Well, since it seems like one phenotype has a survival advantage over another, it seems like natural selection is providing fire-breathing hamsters, in this population at least, with the opportunity to contribute more offspring to the next generation. Again, further studies could provide evidence for this hypothesis. Well, so far, I think we've collected a lot of really interesting data. Certainly enough for each of us to perform our own independent research. Just as we're about to go back to the lab, though, we find an amazing discovery. There in front of us is a flying hamster that somehow appears to have wings that move more like a helicopter than the flying hamster wings we're used to seeing. However, just as we're about to capture it, we watch in horror as a giant boulder falls off a hill and crushes it. Okay, no problem. We'll just find another one, right? I mean, it's not like we haven't been counting thousands of hamsters during our field study or anything. As we count the hamsters in this population, we find that the population is actually really small, and we can't find another helicopter hamster. Whereas we've been counting 1,000 hamsters in the other populations, we can only find 5 others in this population. Because the population was so small, a random event that killed the helicopter hamster has drastically altered the genetic makeup of the population, because it happened to be the only hamster with that phenotype. The random loss of individuals and the alleles they possess is called genetic drift. Now you can see why Hardy-Weinberg equilibrium requires a large population. The larger the population, the easier it is for the population to absorb random loss of alleles without any significant effect on the gene pool. When population size decreases significantly, as seen in this example, the population is said to experience a population bottleneck. We also learned about another form of genetic drift earlier, when we considered the example of stronger winged hamsters escaping the devastation of a volcanic explosion on an island. Only a subset of the population was able to survive by moving to a new piece of land. The strong-winged hamster may have been the minority phenotype in the old population, but they are the majority now. This special form of population bottleneck, in which a small group leaves the main population to form a new population, is called the founder effect. The disproportionate representation of the alleles in the population causes a drastic change in the gene pool and disrupts equilibrium. In summary, an evolutionary agent is any force which alters the genetic structure of a population. The Hardy-Weinberg equation can be used to identify populations that are not in equilibrium. A hypothesis is an educated guess proposed to explain a phenomenon. Non-random mating is an evolutionary agent which alters genotypic frequency. Natural selection is an evolutionary agent which allows certain individuals in a population to contribute more offspring to the next generation relative to the others. Genetic drift is the random loss of individuals and the alleles they possess. A population bottleneck occurs when a population decreases significantly in size. The founder effect is a special form of population bottleneck in which a small group leaves the main population to form a new population. Whew, counting all those hamsters was hard work, but I think we have a lot of interesting data to start a project. One interesting research project might be to try to determine how a flying hamster could have evolved. But we probably should do a little research in the library first before we try to develop a hypothesis. We've seen that natural selection plays an important role in the evolution of a population of organisms. For instance, in our example of a volcano erupting on a remote island, natural selection favored the flying hamsters with the largest, strongest wings. Those hamsters were able to escape the island and continue to pass on their genetic information to their offspring in a new location. We can say that those hamsters were more fit than the ones that could not make it to another piece of land. However, this is not fitness in the common sense. When we speak of evolutionary fitness, we're really talking about a measurement of the ability of a trait to increase or decrease the relative contribution of offspring by an individual to the next generation. Now, that's really just a fancy way of saying that a phenotype which improves the fitness of an individual will improve its viability or reproductive success relative to the other individuals in the population. For instance, the large, strong wings of the island flying hamsters conveyed a higher fitness level to those hamsters compared to the rest of the population after the volcano erupted. Note that before the volcanic eruption, those big wings didn't allow those hamsters to contribute a higher proportion of progeny to the next generation by surviving better or reproducing more successfully. The trait neither improved nor detracted from the fitness of the individual. Only after the volcanic eruption was stronger wings beneficial, because those hamsters could escape the devastation. Recall that this is one of the major principles of Darwin's theory of evolution. Variation within a population provides the genetic variability for improvements to the population. That variation provides the basis for the population to adapt to environmental changes, such as the volcanic eruption on the island. We can say that the larger, stronger wings are adaptations that better equip the hamsters to deal with an unstable habitat. The ability to breathe fire may be another example of an adaptation. We could hypothesize that maybe a mutation in a gene first allowed a hamster to breathe smoke to confuse predators while it made its escape. Through a series of additional mutations, that trait could have evolved into the more direct form of fire-based protection. Now let's consider how hamsters might have evolved the ability to fly. Let's assume that a single mutation didn't just suddenly enable the ability to fly. What if an ancient hamster developed an extra limb? And with the other 4 legs in place for walking, this limb could have been free to evolve into a wing-like structure. As the new limb became more and more wing-like, the benefit to the individual's fitness increased. Maybe an early wing-like structure increased the fitness of the hamster by bestowing the ability to parachute or glide away from danger. Slight increases in fitness over time could slowly select for the winged hamster we are studying today. If we look at the wing more closely, our hypothesis is supported by the fact that the wing bones appear to be very similar to the other hamster limb bones. In fact, if we compare the limb bones of the hamster to a human arm, those bones are similar as well. Any structures which are shared by 2 or more different organisms and are inherited from a common ancestral structure are called homologous. Homologous structures develop from a common ancestral origin, but may have diverged in appearance and function over time. For instance, different selective pressures altered the appearance and primary function of the human arm and a bird wing, but both limbs possess similar bones, as depicted by the colors in the diagram. Contrast this homologous relationship to the relationship between the wing of a bird and the wing of an insect. Both structures allow the organism to fly. So you might think that they are homologous as well. However, if you consider the anatomy of the structures, an insect wing is clearly different than that of a bird wing. For instance, the bird wing possesses bones, while the insect wing doesn't. Traits or structures which are similar in appearance or function, but which evolve separately, are called analogous. The ability of insects and birds to fly is an example of convergent evolution, because selective pressure resulted in the independent evolution of flight in each of these organisms. In summary, fitness is a measurement of the ability of a trait to increase or decrease the relative contribution of offspring by an individual to the next generation. If a trait increased the fitness of an individual, that organism will contribute a higher proportion of the offspring in the next generation relative to individuals without that trait. An adaptation is a trait that enhances the survival or reproductive success of an organism. A homologous structure is a structure shared between different organisms which evolved from a common ancestor. An analogous trait is a trait which may be similar in appearance or purpose, but which evolved independently in the two organisms in question. Convergent evolution occurs when selective pressures results in the independent evolution of similar traits in two or more different organisms. Well, we've come up with an interesting theory on how flying hamsters could have evolved. Our data and research has prompted us to propose that an extra limb could have slowly evolved into a wing. Hamsters that could fly were more fit than those that couldn't, and thus the flight adaptation evolved. To write up our senior thesis, though, I think we need a more complete understanding of evolution. We've proposed that over time, a rudimentary wing may have increased the fitness of an ancestral hamster enough to set off a directional evolutionary change. Suppose there were many different rudimentary wing sizes. If we were to graph the size of the rudimentary wing, we might get a distribution like this, where most of the hamsters have a rudimentary wing that is average in size. The rudimentary wings that were the largest worked the best for gliding or slowing falls. If the increase in fitness for larger rudimentary wings is significant, at each generation, hamsters with larger rudimentary wings would survive better and end up contributing a greater proportion of the progeny in the next generation, due to the lower survival rate of their counterparts in the population. Eventually, this could result in the evolution of a true wing that allowed the hamsters to fly. When characteristics which fall at one end of the phenotypic spectrum are favored over the others, the selection is directional. Directional selection is a common force in an evolutionary trend like we are proposing for the hamster wing adaptation. However, directional selection is not the only type of selection. A second type of selection is known as disruptive selection. Although we think we've identified the type of natural selection that might have led to the evolution of a flying hamster, let's just consider these alternatives. Consider the example of peppered moths living outside London. Oddly, the moths living near the urban areas were dark gray, while the moths in rural areas were light gray. Scientists hypothesize that the dark moths were more fit in the urban areas and the light moths were more fit in the rural areas. But why? An examination of the habitat provided the answer. In the rural areas, tree bark was covered with lichen that was a light gray peppered color. Therefore, darker colored moths would be easier for birds and other predators to identify and eat. However, coal-burning factories covered the trees near urban areas with black soot. Now the lighter colored moths are at a disadvantage, while the dark colored moths are camouflaged. If we consider the mortality of different shades of moths in the whole area, dark colored moths could survive well in the urban area, and lightly colored moths fared well in the rural area. But moths of intermediate color were easy prey in both areas. Thus, disruptive selection favors individuals with traits on either end of the phenotypic spectrum. A third type of selection is known as stabilizing selection. Whereas directional selection favored a trait on one end of the trait spectrum, and disruptive selection favored either end of the spectrum, stabilizing selection favors the middle of the spectrum, or those individuals with an average measurement of the phenotype. Human birth weight is an example of a trait controlled by stabilizing selection, particularly births before the advent of modern medicine. Babies with an average weight tend to survive better than babies that are particularly small or large. The smallest babies are often small because of a premature birth. Not only could they suffer from maladies because of incomplete development, but they also have trouble maintaining body heat and are more susceptible to infection. In contrast, larger babies often meant a difficult birth. Particularly before the advent of modern medicine, difficult births often increase the mortality rate. As a result, stabilizing selection reduces phenotypic variation, in this case favoring average-sized babies. Directional, disruptive, and stabilizing selection are terms usually reserved for more complex quantitative traits. However, we can also describe the selection of simple traits. For instance, let's revisit the disease sickle cell anemia. We've learned that people who are homozygous for the sickle cell anemia allele develop the disease. Obviously, people afflicted with the disease are at a disadvantage because of the sickle-shaped red blood cells. However, evidence suggests that the sickle-shaped cells confer resistance to malaria. The sickle cell alleles actually have an incompletely dominant relationship. So let's reassign plus to the allele that produces normal red blood cells and minus to the allele that produces sickle-shaped red blood cells. That means that the heterozygote possesses both normal and sickle-shaped red blood cells. These sickle-shaped red blood cells help to disrupt the malarial pathogen life cycle. Although minus individuals are more resistant to malaria, their fitness is still adversely affected by the sickle cell disease. However, since the phenotype of this gene is incompletely dominant, a person heterozygous at this gene has enough normal red blood cells to avoid suffering from sickle cell anemia, but just enough sickle-shaped cells to exhibit an increased resistance to malaria. Since plus individuals are susceptible to malaria and minus individuals are susceptible to sickle cell anemia, the heterozygotes have an advantage over both. When there is a heterozygote advantage, the population can be said to be carrying a balanced polymorphism. A polymorphism is simply a fancy name for alternate versions of a trait, such as a round red blood cell versus a sickle-shaped one. Therefore, a balanced polymorphism is simply a situation in which two alleles of a gene are maintained in a population because heterozygotes are more fit than their homozygous counterparts. In summary, directional selection is a type of natural selection in which individuals with a trait at one end of the phenotypic spectrum are most fit. Disruptive selection is a type of natural selection in which individuals at either end of the phenotypic spectrum are most fit. Stabilizing selection is a type of selection in which individuals in the middle of a phenotypic spectrum are most fit. Heterozygote advantage is a case in which the heterozygous genotype has a higher relative fitness than either homozygous genotype. A balanced polymorphism is a situation in which two alleles of a gene are maintained in a population because heterozygotes are more fit than their homozygous counterparts. So we've come up with some interesting ideas about how a flying hamster could have evolved from a non-winged one. And we've hypothesized that long ago, a hamster may have developed an extra limb, which then developed into a rudimentary wing. Now, if this rudimentary wing gave the hamster a better chance of surviving in its environment, the increased fitness would have selected for individuals in the population that could glide or float with these rudimentary wings. A question we haven't addressed yet, though, is whether or not flying hamsters are a completely different species than regular hamsters. Now, to answer this question, first we need to ask, well, what is a species? We refer to different kinds of organisms as species. But what does that mean, really? Historically, scientists have used the morphology of organisms, that is, the size and shape of body parts, to classify those organisms. For instance, a cat and dog clearly look different, and we call them different species. However, we could also say that a Chihuahua looks significantly different than, say, a German Shepherd. Yet scientists categorize both as dogs. It's more accurate to think of a species as an independent evolutionary unit of organisms. The gene pool of dogs does not overlap with the gene pool of cats, because genetic information can't flow between those two populations. Members of the same species either do or could mate with each other given the opportunity. For instance, dogs can mate and produce what we call mutts. In contrast, members of different species, like say cats and dogs, do not mate, or the matings are unsuccessful in some way. We will see later how the process of classifying organisms into species works. For now, let's consider how species are formed. Speciation is the process by which an ancestral species splits into two or more new species. It's sometimes difficult to determine when speciation has occurred, because it is often a gradual process, and not all evolutionary changes result in a new species. For speciation to occur, members of an ancestral species must become isolated from each other. Dividing these organisms into two populations results in two separate gene pools. These gene pools can be separated through geographical or biological mechanisms. In addition, altered reproductive mechanisms can further isolate two populations of organisms. Let's first consider how gene pools can become isolated. Usually, speciation involves division of a population into two new populations by a physical barrier. This is called allopatric speciation. Allo means different, and patris means country. So allopatric literally means different country. It is also referred to as geographic speciation. Allopatric speciation is considered the dominant mode of speciation among most groups of organisms. It's easy to see how things like the appearance of a mountain range, a canyon, or a body of water could isolate populations of organisms. Once two separate populations are established, different selective forces could alter these populations enough to establish new species. Since we haven't seen any normal hamsters in the area during our field studies, allopatric speciation may have played a role in the speciation of flying hamsters. However, sometimes speciation can occur without geographical isolation. This is known as sympatric speciation. Not surprisingly, the root sym means together, since this form of speciation occurs without any physical separation of the new species. A new species evolves while living in the same location as the original one. The most common means of sympatric speciation is polyploidy. Polyploidy is a characteristic of a cell or organism with more than two complete sets of chromosomes. Consider the following example. We've said that most organisms are diploid. What if an error in cell division created an organism that is tetraploid rather than diploid? Rather than two complete sets of chromosomes, a tetraploid has 4 sets. Mating between a diploid and a tetraploid would introduce all kinds of problems. The diploid parent would produce haploid gametes, whereas the tetraploid parent would produce diploid gametes. If a haploid and a diploid gamete fused, the resulting cell would be triploid. Even if the resulting triploid organism survived to reproductive age, having 3 sets of chromosomes introduces an impossible problem during meiosis. How do you divide an odd number of homologous chromosomes evenly? Well, the answer is you can't. So triploid organisms are usually sterile. Since our flying hamsters are diploid, sympatric speciation probably didn't play a role in the evolution of flying hamsters. However, sympatric speciation has played a major role in the evolution of many plant species. In summary, a species is an independent evolutionary unit of organisms. Members of a species either do or could mate with each other given the opportunity. In contrast, members of different species do not mate, or matings are unsuccessful in some way. Speciation is the process by which an ancestral species splits into two or more new species. For speciation to occur, a population's gene pool must become divided. Typically, this isolation is caused by either a physical or biological barrier. Allopatric speciation is speciation that results when a population is separated by a physical barrier. It is also referred to as geographic speciation. Sympatric speciation is speciation which occurs without physical separation of members of the population. Polyploidy is a characteristic of a cell or organism with more than two complete sets of chromosomes. So let's delve into this question of whether or not flying hamsters constitute a different species from your run-of-the-mill hamster. We learned that speciation occurs when the gene pool of a population becomes split. The barriers to gene flow could be geographic or biological in nature. However, speciation is often caused by multiple factors, so let's break down the possible mechanisms for speciation a little differently. Another way to think about how the gene pool could become split is the timing of the barrier to reproduction relative to egg fertilization. Recall that the cell which forms when a sperm cell fertilizes an egg cell is called a zygote. Therefore, scientists can also divide the mechanisms of population isolation into pre- and post-zygotic reproductive barriers. That is, the barrier could prevent fertilization itself and thus occur before a zygote can be formed. There are two other ways populations of organisms could become different species. Some barriers affect the offspring that result from a mating between members of the two populations. Recall that hybrid refers to something derived from a mixed origin. Since these offspring are a mixture of the two types of organisms, they are called hybrid offspring. Mechanisms which reduce the viability or reproductive capacity of hybrid offspring are called postzygotic reproductive barriers. If hybrid offspring have a lower viability, speciation can occur. Viability is basically the ability to avoid dying. For instance, if an organism is sickly due to genetic defects, it has a lower viability. An organism with a super awesome mutation, like say fire breath, has a relative higher viability than its non-fire breathing counterparts, because predators are probably going to eat the things that don't spit fire first. If the reproductive capacity of hybrid offspring is lower, hybrid organisms would have trouble passing on their genetic material to future generations. In this way, even if the hybrid organism itself is perfectly fine, if it makes little or no contribution to future gene pools, speciation can still occur. Well, it looks like we need to get ourselves some regular hamsters to see if they can mate with our flying ones. So let's head off to the pet shop. Okay, so it looks like the local pet shop is out of hamsters. Well, while we wait for the next shipment of hamsters to arrive, let's do more research to see what kinds of prezygotic barriers could have split the two types of hamsters into separate species. It looks like there are 5 major types of prezygotic barriers to reproduction: spatial isolation, temporal isolation, mechanical isolation, gametic isolation, and behavioral isolation. Probably the most obvious way that two species may not be able to mate anymore is that they live in different areas. This is known as spatial isolation. For instance, right now we're having trouble finding regular hamsters, and we have to wait to get them flown into the area. In the wild, geographical separation of different populations of organisms could result in speciation. However, speciation does not always occur when populations become separated. For instance, if our flying hamsters are able to mate with the regular hamsters when given the opportunity, then spatial isolation is not a prezygotic barrier to the hamsters mating. The next way the hamsters could be blocked from mating is temporal isolation. If two populations mate at different times of the year, reproduction would be blocked temporally. In our example, the hamsters might normally live in the same habitat, but if they mate at different times of the year, obviously that'd be a problem. If this is the case, this is where using a controlled laboratory environment becomes a huge advantage to solve a pretty difficult problem. By controlling lab conditions, like the ratio of light and dark the experimental hamsters experience during the day, we might be able to fool both types of hamsters into mating at the same time. That would allow us to determine if the two possibly different species of hamsters are isolated from each other temporally. Another prezygotic barrier that could result in speciation is mechanical isolation. This means that the size or shape of the reproductive organs prevents successful matings. A fourth way reproduction could be blocked prezygotically is gametic isolation. If the sperm of one type of hamster cannot find, attach, or fuse with the egg of the other hamster, a zygote would never be produced. Finally, different species often simply reject or don't even recognize individuals of other species as possible mates. If one type of hamster fails to recognize or simply rejects the other type of hamster, behavioral isolation could have separated these two populations of hamsters into different species. Spatial, temporal, and behavioral considerations of other organisms can also affect speciation. For example, two populations of plants could diverge because of the preference of their respective pollinators to choose one species of plant over the other, preventing cross-pollination. Once we can conduct some mating experiments with some non-flying hamsters, we'll be able to determine if any of these prezygotic barriers to reproduction have caused flying hamsters to diverge from non-flying ones. In summary, a zygote is the cell that forms when a sperm cell fertilizes an egg cell. A prezygotic reproductive barrier is a mechanism which prevents fertilization from occurring. A postzygotic reproductive barrier is a mechanism that reduces the viability or reproductive capacity of hybrid offspring. Spatial isolation is a type of prezygotic barrier in which different species are reproductively isolated by location. Temporal isolation is a type of prezygotic barrier in which different species are reproductively isolated by different mating seasons. Mechanical isolation is a type of prezygotic barrier in which different species are reproductively isolated due to incompatible reproductive organs. Gametic isolation is a type of prezygotic barrier in which different species are reproductively isolated because the sperm of one species cannot find, attach, or fuse with the egg of the other species. Behavioral isolation is a type of prezygotic barrier in which species are reproductively isolated because individuals in one species reject individuals of another species as possible mates. Well, while we were waiting for our run-of-the-mill hamsters to show up, we learned a lot about the types of barriers that can cause reproductive isolation at a point before fertilization. Matings between our flying and non-flying hamsters successfully result in fertilization and zygote formation. Therefore, it doesn't seem like the gene pools of the two types of hamsters are isolated prezygotically. However, even if the two types of hamsters were able to mate and produce a zygote, that zygote still has to successfully mature to a reproductive age and produce progeny for the two hamsters to be simply variants of the same species. Although prezygotic barriers may not have caused speciation, a mechanism that occurs after zygote formation could still be isolating the gene pools of the two populations of hamsters. Postzygotic reproductive barriers occur after the zygote has formed, meaning they either reduce the viability, which basically means the ability to avoid dying, or the reproductive capacity of the hybrid offspring. Hybrid zygote abnormality, hybrid infertility, and low hybrid viability are postzygotic mechanisms which could still define the flying hamsters as a different species than non-flying hamsters. Let's examine these possibilities more closely to see if our flying hamster is a new species. Although fertilization may succeed, the hybrid zygote still has to develop and mature into an adult organism. Sometimes hybrid zygotes fail to mature normally. This is referred to as hybrid zygote abnormality. Normally, this means that the hybrid dies during development, but sometimes the hybrid survives birth, yet suffers from such severe abnormalities that it can't successfully mate as an adult. Well, it looks like early development of our hybrid hamsters progressed normally. The hybrid survived birth and appear to be growing normally, albeit a little bit small. So it looks like we can cross off hybrid zygote abnormality as a possible mechanism of speciation, dividing the flying and non-flying hamsters. Another mechanism that can separate two gene pools is hybrid infertility. If all hybrids are sterile, genes can't flow between the flying hamster and non-flying hamster populations. Since we defined a species as an independent evolutionary unit of organisms, we would call these two populations different species, since no genetic information can be shared between them. A common example of hybrid infertility is a mule. Although a horse and a donkey normally ignore each other in the wild, humans have engineered mules through artificial means. Mules themselves are obviously quite healthy, serving as pack animal and mode of transportation for man throughout history. The problem with a mule isn't apparent until it tries to pass its genetic information onto a new generation of mules. Since a mule has an odd number of chromosomes, 63 to be exact, meiosis is, well, a mess. If you recall, the goal of meiosis is to reduce the genome by half to create gametes. Do you see the problem? It's impossible to divide an odd number of chromosomes evenly into two cells. Therefore, virtually all potential mule offspring are doomed to suffer from some kind of aneuploidy. Since we learned that aneuploidy is generally a lethal trait, it's not surprising that mules rarely are able to produce viable offspring. Since mules can't pass along their genetic content, donkeys and horses are considered separate species. Well, the hybrid hamsters aren't old enough to assess their reproductive capacity yet, but let's go ahead and check on the little guys before we go home. Uh-oh. I think we know that they aren't the same species now. It looks like the hamster hybrids suffer from low viability. Low hybrid viability is a third form of postzygotic isolation. The fact that the young hybrid hamsters appear to be dying at an alarming rate indicates that hybrids probably suffer from lowered viability compared to comparably aged flying hamster or non-flying hamster offspring. So, that's kind of a bummer, but this experiment indicates we've discovered a brand new species. Hopefully, future experimentation with flying hamsters will lead to discoveries which will improve human life. In actuality, often more than one mechanism for reproductive isolation affects the separation of a population into two separate species. I think we have a pretty good basis for some really stellar research projects. It's been fun collecting field data and brainstorming new theories with you. So let's get writing and see if we can make a name for ourselves in the exciting new field of flying hamsters. In summary, a postzygotic reproductive barrier is a mechanism that reduces the viability or reproductive capacity of hybrid offspring. Hybrid zygote abnormality is a type of postzygotic barrier in which hybrid zygotes fail to mature normally. Hybrid infertility is a type of postzygotic barrier in which hybrids are incapable of producing offspring. Low hybrid viability is a type of postzygotic barrier in which the viability of hybrid offspring is lower than offspring in either population.
The theory of evolution explains the phenomena that species living today are descendants of species from long ago. A common misconception about this process is that individuals adapt to their environment in order to survive, but this is not the case. For example, a giraffe may want a longer neck so that it can reach leaves on higher branches, but it certainly can't make its neck grow longer to do so. However, other giraffes within the local population may naturally have longer necks, and if leaves on lower branches are in short supply, the shorter necked giraffes within that population will not survive as well as the ones with longer necks. In this way, the population changes over time so that longer necked giraffes are more common, and this reflects the demands of the environment, the higher branches. So it's in this way that evolution is more of an editing process than a creative process. There's no such thing as a perfect organism. Evolution simply selects for individuals with traits that are best suited to the current environment. If low branches become more available, the short-necked giraffes would thrive since they are the ones within the population best suited for this type of environment. Charles Darwin saw much evidence for evolution because he observed variation among individuals within populations like the short and long-necked giraffes. These differences are known as genetic variations, which are the naturally occurring genetic differences among individuals. Variation is easy to see. For example, if your friend is looking for you in a crowd, they can easily spot you because you look different than everyone else. This comes from your unique DNA, which helps you look just a little bit different than all other humans. Even within your immediate family, you share many common traits, but each sibling will have their own variation on the genes inherited from your parents. We can measure genetic variation, but what may be more important to understand is the potential for a characteristic to vary within a population, or the genetic variability. This is different from genetic variation because instead of measuring the actual variation within a population, it measures how much the trait will vary. Genetic variability is directly related to biodiversity and evolution because a population needs enough variability to be able to adapt and evolve to environmental changes. Let's look at our giraffe population as an example. The genetic variation in the population is the different length necks, short, long, and everything in between. The variability is how much that neck length tends to vary within the population. A high variability will allow the population to adapt to environmental changes, like the branch height, whereas a low variability means that population will not be able to adapt to new branch heights and will risk extinction. Some variations are hereditary, like neck length in giraffes. Long-necked giraffes will tend to have long-necked giraffe babies, while short-necked giraffes will tend to have short-necked babies. Sometimes, though, variations occur within a population because of random mutations, which are changes in the nucleotide sequence of DNA. Your DNA is like a set of instructions, and a mutation occurs when those instructions are changed. Random mutations are a critical component of evolution because mutations create genetic variation within a population. If a mutation is harmful, it will likely be weeded out very quickly because any change in the population that makes an organism less fit will ultimately lead to its demise. But sometimes a mutation may actually be beneficial to an individual, so it survives and reproduces, passing that variation on to the next generation. This often occurs when the environment is changing in such a way that it supports the mutation. We see this with pesticide-resistant insects. All it takes is one individual to have a mutation in their genome that makes them resistant to pesticides, since they can survive and then pass this mutation on to their offspring. While most of the population may die after a pesticide application, the ones that have the resistance mutation will eventually become common, essentially creating a new population that is resistant to the poison. Evolution occurs within a population because of variation among individuals. Genetic variation is when individuals in a population have variations of the same trait, such as short or long-necked giraffes. The genetic variability of that population tells us how much the trait will vary. This also tells us how well that population will adapt to environmental changes. High variability means better adaptability, but low variability means a higher likelihood of extinction. While many variations are hereditary, like a long-necked giraffe having long-necked babies, sometimes a variation comes about from a random mutation. This mutation occurs because of changes in an individual's DNA. Often, mutations are lethal because they are not beneficial to the individual. However, sometimes a mutation is beneficial, and this allows that individual to survive and create more offspring, making it more fit for its environment. Wouldn't it be great to be a chameleon? You could just blend in with whatever was around you. No need to run from a hungry predator, you just disappear from sight. Unfortunately for other animals, they don't have this ability, so they have other ways of camouflaging themselves to blend in with their surroundings. Usually this works pretty well, because if you don't blend in, you get eaten. But imagine that all of a sudden, the environment around you is drastically altered. Your camouflage is no longer beneficial if it doesn't help you hide. Worse yet, it may even make you stand out like a sore thumb, advertising your whereabouts like a big neon sign. This is exactly what happened to the peppered moths in England during the Industrial Revolution. Peppered moths got their name because they are a light gray color and look like they've been sprinkled with pepper. This coloration pattern helped the moths blend in with light-colored trees and lichen, protecting them from predators. This was a very good camouflage because the moths looked very much like the trees and lichen they rested on, sort of like a permanent chameleon color. This all changed with the Industrial Revolution in England, which began in the late 1700s and ended about 80 years later. The Industrial Revolution was a time of great change. Machines replaced much of the work that was done by manual labor, chemical manufacturing was increased, and biofuels such as wood were traded in for new fuels like coal. Life was greatly improved for many people as power became more efficient and affordable. People were earning more money, traveling farther, and in general just able to get more done. However, the pollution created by the Industrial Revolution had serious consequences, both for humans and the environment. Remember the peppered moth? Well, the lichens they rested on began dying, and the trees became covered with soot. The soot turned the trees black, making the light-colored moths stand out in stark contrast. This made them easy targets for predators, and their numbers began to dwindle. At the same time, though, the peppered moth was also thriving. How is this possible? Well, just like with any species, there are variations among individuals. Among the peppered moths, there were individuals that were much darker than the normal light color with black spots. These darker individuals would have stood out against the light-colored lichen in trees, but once the trees became darker in color, the darker moths suddenly became the ones blending in. This means they were less likely to be eaten by predators, which inevitably means they lived longer and could reproduce more successfully. So, while the light-colored moth was disappearing, the dark-colored moth was thriving. Natural selection is a process in which organisms best suited to their environment are more likely to survive and reproduce. Natural selection is the mechanism for evolution because over long periods of time, populations become better adapted to their surrounding environment. Keep in mind, evolution does not select for the best organisms. Neither the light or dark moth was a better moth. They were just different variations within a species. Instead, it's the demands of the environment that either support or work against specific traits in a population, and rarely do those changes happen as quickly as they did with the peppered moths. Normally, evolution of a species occurs over thousands, if not millions of years. But sometimes circumstances can be so extreme that changes come about much sooner, as they did during the Industrial Revolution. Because the population of peppered moths shifted from one extreme variation to the other, from the very light color to the very dark color, we call this directional selection. Instead of a blended version of the coloration, or an increase in the dark variation without a decrease in the light variation, the selection was for the moths on the other end of the color spectrum within that population. The darker trees provided more camouflage, so the adaptation was directed toward the darker moths that blended in better. Since the Industrial Revolution, many environmental regulations have been put into place that reduce pollution from factories and other sources. This has given the light-colored moth a chance for a much-needed comeback, and today they are common once again. We have learned a lot about natural selection and evolution from studying the peppered moths in England. During the Industrial Revolution, the once common light-colored moth became the minority in the population. Originally, individuals with this color variation thrived because they blended in with their surroundings very well. But once pollution became rampant, the trees became black with soot and the light-colored moths stood out like a sore thumb. At the same time that predation became more common for the light-colored moths, the dark-colored peppered moths were thriving. Instead of being the ones that stood out on the trees, they now blended into their surroundings and were better able to survive. You've likely heard of natural selection, which is often described as the survival of the fittest. This is a bit of a misnomer because in evolution, individual organisms don't willfully adapt to their environment. Rather, environmental stressors lead to the selection of specific traits among individuals within a population, pushing those traits forward through time. So, in natural selection, what is considered fit actually depends on the environment and its demands. Artificial selection is a bit different. This is when specific traits are deliberately selected for or against by humans. You may not have been aware of the term artificial selection, but you are in fact quite familiar with the concept already. Modern dog breeds have all been selected for by humans, as have most food crops that you buy at the grocery store. Again, the main difference is that natural selection is a natural process, while artificial selection is a forced selection by humans. Because of the selective process of artificial selection, it's often called selective breeding. This type of breeding has been used by humans in both plants and animals for thousands of years. The modern sweet corn you eat today is quite different from its ancestor, a plant called teosinte. Both plants are grains, but teosinte looks much more like the grass family that it belongs to, unlike our modern corn that has large, juicy kernels. Wild mustard has also been extensively selected and bred for certain traits. Broccoli, cauliflower, kale, and cabbage are all related to the wild mustard plant. For example, we get broccoli from flower suppression of the wild mustard, and kale from its leaf enlargement. What this means is that wild mustard plants with larger leaves were selected for and eventually developed into a plant that now has very large, tasty leaves. Animals have also been selected for in many domestic realms. A Great Dane looks nothing like a Chihuahua, right? And this is because in both dog breeds, the specific traits that led to their differences were selected for by humans, and animals that had these traits were bred so that the traits would continue down family lineages. The cows, pigs, and chickens that are eaten in the U.S. also have traits that are selectively bred. Larger animals that grow quickly and produce more meat are desirable because they are more cost efficient for farmers. There is no better way to create your perfect organism than through artificial selection. With selective breeding, you can breed plants that grow faster and larger and are more resistant to parasites and pests. You could breed horses that run faster, sheep that grow thicker wool, and cows that produce more meat. However, in selecting this way, you're playing a risky game. Selective breeding certainly keeps the traits you want, but it also reduces the size of the gene pool for those organisms. This brings forth mutations that would normally be suppressed within the population. After some time, the species that is produced through artificial breeding has a much lower genetic diversity than its wild ancestors. This means individuals are more susceptible to disease, and the species as a whole may not be able to adapt to changes in the environment. For example, let's look at purebred dogs, which are well known for their breed-specific health problems. The artificial selection process used for dog lineages is called inbreeding, which is the mating of organisms that are closely related. The inbreeding process creates dogs that look alike within a breed, such as Dalmatians that look like other Dalmatians, or Labrador Retrievers that look like other Labs. However, breeding close relatives also severely limits the genetic variation within the breed, so the mutations within that lineage become more common. Dalmatians are commonly deaf, and hip dysplasia is quite common with Labs. Artificial selection is a process in which humans deliberately select for or against certain traits in other organisms. This process is known as selective breeding because of the selection of traits through breeding. Artificial selection has been used by humans for thousands of years. We have created crops that are much different from their ancestors, as well as animal breeds that are each unique and specific to their traits. Artificial selection is certainly an effective method of breeding for desired traits, such as larger, faster growing crops than farm animals, but it can also create problems. Genetic diversity is greatly decreased through the inbreeding process, which is the mating of organisms that are closely related. Reducing genetic diversity means that mutations that would normally be selected against in nature may in fact be inadvertently selected for through artificial selection. This is easily seen in purebred dogs, which quite often have breed-specific health issues.
May the force be with you, or in this case, may the four forces be with you. It's a life or death, high-stakes game of evolution, and every creature, every plant, is going to need all the help it can get. In an ever-changing world, species are trying to adapt, compete, and sometimes just get lucky in an effort to continue their existence. So what are these mysterious four forces of evolution? They include mutation, gene flow, genetic drift, and natural selection. Mutations happen just by chance. Somewhere along the line, a change in a gene or a chromosome shows up as a physical or a behavioral difference in an individual. Because it affects a gene or chromosome, it can then be passed to the next generation. Maybe you think of a mutation as a creepy-looking new feature, but not always. A cat that has 6 toes on each foot has a mutation that has been passed on from a cat far in the past. Okay, that might be a little creepy, but here's one that's not. It's believed that red hair in people was a mutation. Redheads are only 1 to 2% of the population. It may be rare, but not too creepy. In gene flow, 2 populations mix genes. Over time, there's less distinction between those populations. So let's say there are 2 populations of polar bears that have not met in the recent past. Each population was existing on their own, separate chunks of land for a very long time. One population lived to the south, and one population lived in the north. There was no reason, or perhaps no way, to have met before. Then, as the climate started to change and more of the sea ice started to melt, the polar bears in the south were not finding the habitats they needed to survive. They started to shift their population towards the north and met the other population of polar bears. Once they started interbreeding, the genetic distinctions decreased. True story. Scientists are seeing this example of gene flow right now. Like mutations, genetic drift is also random. Genetic drift is the chance change in the frequency of an allele or variant form in a gene from one generation to the next generation. It shows up more in small populations. Let's say there's a population of rabbits near where you live. Most of the rabbits have typical short fur, but some of them are extra fluffy. Maybe the proportion of each kind has been the same for decades, but for whatever reason, one year there are even more short-haired rabbits born and fewer fluffy ones. Next season, more short-haired rabbits mate and even fewer fluffy ones are born. Just by chance, the genetic composition of that little rabbit population is drifting away from having fluffy rabbits. Natural selection is probably the most famous force of evolution. In natural selection, differences between individuals can be a real help or a real problem. Individuals that are better adapted to their environment have a better chance of surviving, and that also means a better chance of reproducing and passing those traits to their offspring. If a trait isn't so great for survival, well, then the chances of passing that along are not too great either. A good example of how natural selection works is with pesticide-resistant mosquitoes. Every time we humans think we've beat them with a new pesticide, a few pesky individuals have a genetic makeup that allows them to survive and pass those adaptive genes along. There are 4 forces of evolution: mutation, gene flow, genetic drift, and natural selection. A mutation is a random change in a gene or chromosome that can make for a physical or behavioral difference in an individual. Because it starts with a change in a gene or chromosome, it can be passed on to future generations. In gene flow, 2 populations mix genes. Over time, there's less distinction between those populations. Genetic drift happens by chance. Some alleles get passed along from one generation to the next, and over time they become more frequent. In natural selection, some individuals are better adapted to their environment. They outsurvive other individuals, have a better chance of reproducing, and pass along those helpful traits to the next generation.
Evolutionary fitness is how well a species is able to reproduce in its environment. If they're no longer reproducing, then they are no longer evolutionarily fit. For example, you did not ride a Tyrannosaurus Rex to the store. Why not? They're all gone. In their environment, they were extremely fit as they ate, reproduced, and continued their species on. But what most often stops evolutionary fitness, and your pet T-Rex, is a shift in the environment. Your pet T-Rex died out because the environment could no longer support it. The sun was blotted out, resulting in a massive decrease in the amount of plants, causing the oxygen level to drop, so the environment could no longer support large creatures. The plants died because there was less sun, resulting in the bottom of the food chain shrinking. With fewer plants, the large herbivores starved to death, and with no herbivores to eat, the predators starved to death. The well-known term, survival of the fittest, was not actually said by Charles Darwin. The phrase was actually coined by Herbert Spencer, who used it for social Darwinism, a concept we will not be exploring here. Survival of the fittest is actually impossible because fittest implies an end. If the fittest critter existed, it would be a massive predator that feared nothing and could not possibly mate, because by mixing its perfect genes, or internal blueprints, with anything, it would then become less fit. Charles Darwin is best known for his work on evolutionary theory. He was not, however, the first to propose an evolutionary theory. The first would be Anaximander of 600 BCE, Ionia, who noticed human limbs and fish limbs bear a striking similarity, leading him to believe that land-based animals must have originated in the sea. Darwin described the 2 mechanisms of evolution in his book On the Origin of Species. Another researcher by the name of Alfred Wallace came to similar conclusions as Darwin. However, Darwin's ideas and writings were more thorough and published, resulting in the scientific community attributing the evolutionary mechanisms to him. Darwin and Wallace's evolutionary mechanisms actually support the idea of survival of the fit. Those who are fit enough to reproduce will pass on their genes, and those that do not work will die out. Natural selection is the process by which the critter with the best adaptations survives long enough to reproduce. Let us take 200 people and divide them into 2 groups. Each group of 100 is going to live on a different island for the next 100 years. Island A is near the equator, which means it will get a lot of sunshine and will be very warm and very humid. This type of climate is very similar to what is seen in the Philippines. Because it is very warm, the vegetation tends to produce very little edible plant matter, and the animals tend to be very small. If there is very little food, the larger people will be hungry while the smaller people will be able to survive off the smaller food. In addition, the bright sun will give light-skinned people some nasty sunburn, while those with darker pigmentation in their skin will be better off. After 100 years, the 100 people we put on Island A are now smaller and darker skinned. This is seen in a person with Filipino heritage. Island B will be nearer the Arctic Circle, with heavy cloud cover resulting in tougher vegetation but larger sea animals that stay warm because of their blubber. To hunt big sea beasts like walruses, you need big people who can hunt. In addition, because this island receives less light, people on this island will need lighter skin so they can more efficiently process sunlight into vitamin D. After 100 years, you will have large, pale-skinned people who look very Scandinavian. This is the process of natural selection. What works best in the environment will endure. Natural selection works by having traits that are useful continue on to the next generation, and what does not work will be bred out. Sexual selection works by selecting what is passed on to the next generation by having those with desirable traits breed more. This works regardless of how useful the trait is. If you think of a peacock, you think of the large fan of very colorful feathers. Why do peacocks have those feathers? They do not ward off predators, they do not have any defensive properties to help protect them from predators, and they do not help them find food. They actually do just the opposite, since the male peacock must carry around this very large bundle of feathers, making it difficult to run away from predators and move through plants. The reason the male peacock has such a large feather bundle is because the females mate with the males with the largest and brightest feathers. If within each generation only the largest feather-bundled peacocks get to make babies, then those babies will only have the genes for large feather bundles. This repetition at each level adds up, and the feather bundles are now a burden because they are so large. Sexual selection works by having females or males determine what is desirable and passing whatever that may be on to the next generation. Does Homo sapiens' greatest tool, the brain, evolve as well? Can a mind and brain show evolutionary fitness? A study by Sharps, Villegas, Nunes, and Barber in 2002 looked at how well humans memorized new information, but they added an evolutionary twist. What do average college students with no outdoor experience memorize faster, animal tracks or military equipment and vehicles? Which option will allow humans in a non-modern society to survive? If you had 2 hunter-gatherers and one was able to remember and track animals, a vital component of hunting, then he will survive and be able to have more children. Generation after generation allows the hunter-gatherers who can track and hunt better survive and have more children. This study demonstrates that evolutionary fitness is not just in claws, skin, or feathers, but in the brain and in how a species thinks. Evolutionary fitness is how well a species is able to survive and reproduce in its environment. Charles Darwin outlined the mechanisms of how species change by natural selection and sexual selection. Natural selection is when the traits of a creature, such as size, skin color, or other features, make it better able to survive. Sexual selection is when a species has a trait that it finds desirable, resulting in individuals with that trait reproducing more. Change is connected to environment. So if the environment changes, then the species must also change or go extinct. Lastly, evolution affects not merely skin, claws, or feathers, but also part of the brain and thought processes of a species. Charles Darwin published On the Origin of Species with the oft misquoted line, "Survival of the fit." Fit, not fittest, was originally written. The idea here was that one merely needed to be fit enough to reproduce to pass on its genetics in the form of a new generation. Herbert Spencer was a 19th century philosopher and social idealist. He was known to be a strong supporter of Darwin's methods of evolution and a supporter of the evolutionary process by way of his writings and critiques of others. As mentioned already, Darwin published the oft misquoted line of survival of the fit. Herbert Spencer would take this concept and begin to apply it in other ways, using evolutionary theory as a means to understand the world in which he lived. Herbert Spencer's works modified the line, making it survival of the fittest. This idea and quote were then used as the foundation for what would become known as social Darwinism. Survival of the fittest implies that the strong will succeed and the weak shall perish. The fittest will be successful, and they shall rule the weaker because they are the most fit to do so. In a brutal world without social hierarchy, class, and social customs, this would mean the person who is the strongest and has the longest spear rules over everyone. But this was high society. We cannot have men in suits attacking each other with clubs. They have rules against that. So the idea of the fittest was applied to what the highest society valued: capitalism and political power. The leaders of the capitalistic world, the Rockefellers, the Carnegies, and many more, took hold of this idea and applied it to themselves. Social Darwinism is the application of what was interpreted as the law of evolution. Those who succeed in society were superior to those who did not succeed. In the minds of the radical social Darwinist, to be successful and ruthless at business was no different than the tiger eating for food. It was the way of the world. Those who failed to succeed in business were considered unfit and, it was reasoned, should be treated as such. The most noteworthy users of the social Darwinist motto, survival of the fittest, were the captains of industry, the masters of capitalism, also known as the ultra-rich. We are not talking Steve Jobs wealthy. Rockefeller was worth close to $341 billion in today's currency. How did one accrue this kind of wealth? By exploitation of the workers and using ideology that states if you are succeeding, you have moral authority. This is what it means to be the fittest. Another use of social Darwinism is to look at those who are considered failing in society. In a capitalist society, those failing are the people who occupy the lowest social strata, the poor. A connection was made between the immigrant and racial minority populations, who usually occupy the lowest socioeconomic class, and their failure to rise. Social Darwinists interpreted the immigrants' lower status as evidence of them being evolutionary failures, and allowing them to continue would cause unnecessary pain in the future. In the mildest forms of social Darwinism, you see the Rockefellers, who exploited the worker and paid low wages to discourage the working class from being able to support large families. In its harshest form, you see eugenics, or the practice of removing unwanted genetic material from the gene pool. This might mean forcibly sterilizing people who were considered unfit to have children. In extreme cases, it might mean actively killing the supposedly unfit. Herbert Spencer was a social Darwinist who compared social success to the biological success of a creature. He was not, however, a proponent of those who took his ideas to the extreme. Spencer was not as heartless as those who used his ideas. Herbert Spencer's focus was on utilitarianism, or selecting the course that maximizes the rewards or utility of a situation. His writings often centered on justice. Justice, in his mind, was equated to liberty for all people. So if everyone had equal liberty, then those who succeeded would be naturally superior to those who do not. This does not give them permission to treat those who are inferior as such. Darwin wrote survival of the fit to imply that those who were fit would live long enough to pass on their genes. Spencer wrote survival of the fittest, implying those who were most fit would survive the social world due to some biological mechanism that made them superior. This idea was taken by leaders of the capitalist industries and made into an excuse to exploit others in its mildest forms, or as a reason to remove their genetic material due to their failure to thrive. This was most likely not Spencer's original intention, since he tended to write on the topics of utilitarianism and justice. Systematics is the part of science that deals with grouping organisms and determining how they are related. It can be divided into two main branches. Taxonomy focuses on classifying, naming, and grouping organisms. A group, or taxon, can be a population, a species, a genus, or a higher-level grouping, such as family, order, class, phylum, kingdom, or domain. The plural of taxon is taxa. Phylogenetics is the study of determining evolutionary relationships or patterns of descent of organisms. All of the species of organisms that are alive today have descended from ancestral species. This is due to evolution, or simply change over time. The evolutionary relationships of ancestral species and their descendants can be diagrammed using branching evolutionary trees. Just like your family tree, an evolutionary tree indicates which ancestors gave rise to which descendants. An evolutionary tree can also be called a phylogenetic tree or just a phylogeny. There are many different ways to draw phylogenies, but they do all have certain parts that you must understand before you try to interpret them. The root of a phylogeny represents the common ancestor of all the descendants in the tree. The descendant taxa are labeled at the tips of the tree. A node splits into two branches and indicates a divergence or speciation event. The node itself represents the common ancestor of any descendants that branch off of it. The two taxa that branch off at a node are called sister taxa. They share an immediate common ancestor. In this phylogeny, taxa B and C are sister taxa. They are both equally related to taxon A. The branches of a phylogeny can be rotated around a node without changing evolutionary relationships. If you want to determine how two or more taxa are related, it is important to look at the nodes and branches in a tree and not just the ordering of descendant taxa. Sometimes the evolutionary relationships between taxa cannot be determined. This results in a phylogeny with a polytomy, or a node from which more than two groups split. A clade is a group that includes an ancestor and all of its descendants. Clades, also called monophyletic groups, can be nested in larger clades. For example, mammals are a monophyletic group because they all descended from a common ancestor. Within the mammals, there are also many smaller clades, such as primates or bats. Not all groupings of organisms qualify as monophyletic. A paraphyletic group consists of an ancestor and only some of its descendants. Reptiles are animals like crocodiles, lizards, and snakes. This is actually a paraphyletic grouping because the ancestor that gave rise to all reptiles also gave rise to birds. If birds are added to the definition of reptiles, then it could be considered a monophyletic group. A polyphyletic group is made up of various descendants with no recent common ancestor. Marine mammals are polyphyletic. Whales and seals are both marine mammals, but they are not closely related at all. Seals are more closely related to bears than they are to whales. Whales share a more recent common ancestor with deer than they do to seals. Now that you know how to read an evolutionary tree, let's look at a simple example using humans and their living relatives. From this tree, you can see that humans and chimpanzees are sister taxa. Remember, this means that they share a recent common ancestor with each other. Humans did not evolve from chimpanzees. Gorillas share a common ancestor with both humans and chimpanzees, but it is not as recent. Gorillas are just as closely related to chimpanzees as they are to humans. Of the species shown, orangutans are the most distantly related to humans. Let's review. The evolutionary relationships of ancestral species and their descendants can be diagrammed using branching evolutionary trees. An evolutionary tree can also be called a phylogenetic tree or just a phylogeny, and phylogenetics is the study of determining evolutionary relationships, or patterns of descent of organisms. Just like your family tree, an evolutionary tree indicates which ancestors gave rise to which descendants. The root of a phylogeny represents the common ancestor of all the descendants in the tree. The descendant taxa, or groups, appear at the tips of the tree. A node splits into two branches and indicates a divergence or speciation event. The node itself represents the common ancestor of any descendants that branch off of it. The two taxa that branch off at a node are called sister taxa. They did not evolve from each other, but rather share an immediate common ancestor. The branches of a phylogeny can be rotated around a node without changing evolutionary relationships. Thus, the order of descendant taxa at the tips of trees isn't important. Taxa that branch off first in the phylogeny aren't lower or less evolved than those taxa that branch off later. There are three main ways to group organisms. A clade, also called a monophyletic group, is a group that includes an ancestor and all of its descendants. Mammals are an example of a monophyletic group. A paraphyletic group, such as reptiles, consists of an ancestor and only some of its descendants. Reptiles would be a monophyletic group if you included birds. Finally, a polyphyletic group, such as marine mammals, is made up of various descendants with no recent common ancestor.
You may have learned about the water cycle or the carbon cycle before. These cycles move substances, water or carbon, throughout the environment and organisms living there. The process is fairly simple. However, the movement of nitrogen through living and non-living things is a bit more complex. The nitrogen cycle is more complex than other biogeochemical cycles because most organisms cannot use the nitrogen found in the atmosphere, even though almost 80% of the air is nitrogen. In the atmosphere, nitrogen is found as N2. This N2 is not usable by most plants and animals. In order for plants and humans to get the nitrogen they need, the nitrogen must be converted into ammonia, NH3. The conversion of this atmospheric nitrogen to the usable form of ammonia is known as nitrogen fixation. Nitrogen fixation can be defined as the process of creating ammonia from atmospheric nitrogen. Without this process, most plants and animals would not have the nitrogen needed to live. Most commonly, nitrogen fixation is carried out by bacteria. Now, this isn't all bacteria, but some specific forms. You may think of the bacteria that get you sick, but the bacteria that make ammonia from atmospheric nitrogen are found near plant roots and won't make you sick. This nitrogen-fixing bacteria is most commonly found near the roots of legumes. Legumes are plants such as soybeans and garden peas. The bacteria and the plants have a relationship that is beneficial for both. You may remember from ecology that when two species live together, it's known as symbiosis. There are several types of this close, long-term interaction between two different species. In the case of nitrogen-fixing bacteria and legumes, both species benefit from the interaction, meaning that it is mutualism. The nitrogen-fixing bacteria benefit because they have a safe place to live amongst the roots of the plants, and the legumes benefit because they basically have an unlimited source of usable nitrogen. The plant does use some energy in order to provide a good home for the bacteria, but this expense is well worth it for the plant because of the nitrogen produced by the bacteria. The specific name of bacteria found in most legumes is Rhizobium bacteria. The bacteria contain a specific enzyme, nitrogenase, that breaks the bonds between the atmospheric nitrogen. Once the bonds are broken, the bacteria can then fix the nitrogen into ammonia. The plants and then animals can then use this fixed form of nitrogen. Now that we know that nitrogen-fixing bacteria convert atmospheric nitrogen into ammonia, we need to look at why this matters. So what if plants can't use the nitrogen in the air? What do they really need nitrogen for anyway? We know that plants need elements such as carbon, hydrogen, and oxygen for photosynthesis, but nitrogen is actually also needed for this process as well. Chlorophyll, the green pigment found in the chloroplast of plant cells, is responsible for photosynthesis. One of the elements that help make chlorophyll is nitrogen. Without nitrogen, plants would not be able to perform photosynthesis. Plants also need nitrogen because it is found in all living cells. Having the proper amount of nitrogen helps with plant growth. Plants that have the right amount of nitrogen will have more seed and fruit production than those plants that do not have enough nitrogen. This nutrient also improves the quality of the leaves. Lastly, plants need nitrogen specifically for proteins. Proteins are molecules that are responsible for many life processes. In plants, proteins can be used as enzymes, which help control the rate of reactions and control metabolic activities. If plants do not have enough nitrogen, they will not be able to function properly. Because of this, nitrogen is found in many fertilizers. You may have seen ads for fertilizers that show plants that were given the fertilizer and plants that were not given the fertilizer. Generally, the plants that receive the fertilizer, and therefore plenty of nitrogen, have much bigger and more vibrant flowers than the ones that did not get this nutrient boost. Plants are not the only living things that need nitrogen in order to survive. Animals do as well. We will look at why humans need nitrogen. We know that humans get nitrogen by consuming food, such as plants that contain nitrogen. When we eat meat, we are getting nitrogen from the plants that were eaten by that animal. We already know that all cells contain nitrogen, but why else do humans need this element? Humans need nitrogen in order to make proteins. Proteins are made of smaller units called amino acids. Nitrogen is an essential part of amino acids and therefore proteins. Examples of proteins in humans can be found in muscles, hair, skin, blood, and nails. Nitrogen is also an important component of DNA, which contains our genetic information. Much like in plants, proteins can help control metabolic activity in humans. Enzymes are also found in humans and help regulate the rate of reactions. Proteins are also needed for growth, immune function, tissue repair, and even making hormones. Even though almost 80% of our atmosphere is nitrogen, most plants and animals cannot use this atmospheric nitrogen. In order for us to be able to use this nitrogen, it must first be fixed. Nitrogen fixation is the process of creating ammonia from atmospheric nitrogen. This process is carried out by specific bacteria that are found near legumes like soybeans and garden peas. The relationship between the bacteria and the legumes is mutualistic, meaning that both organisms benefit. Plants and humans need nitrogen for countless reasons. Specifically, plants need nitrogen because it is a component of chlorophyll, which is the green pigment responsible for photosynthesis. Plants also require nitrogen for proper growth and development. Humans need nitrogen for proteins and DNA. Without nitrogen-fixing bacteria, humans and plants would not be able to access the nitrogen around us.
Primary succession is the colonization of new sites by communities of organisms. It often occurs after a devastating event has wiped out the organisms that lived in the area, or with the creation of a new habitat. Let's look at a couple examples to see how primary succession occurs. Imagine a volcano erupting. Lava flows down the side of the volcano, destroying everything in its path. But when it cools, it forms new land. Initially, the new land will be barren with nothing living in the area. But after some time, simple organisms will begin to colonize the new land. The organisms that engage in primary succession are known as pioneer species. Typical pioneer species include simple lichens, algae, and fungi. These simple organisms begin breaking down the resources in the environment and make it suitable for the later introduction of more complex species, such as vascular plants. As these organisms carry out their life processes, they produce waste and some die. This leads to the formation of the organic material that will become soil. Once a small layer of soil is in place on top of the lava flow, the variety of life will begin to increase much more quickly. The pioneer plants will be crowded out by more complex plants, such as grasses and small shrubs that are able to live in the thin layer of newly formed soil. These small plants will continue to increase the conditions of the newly formed habitat and make way for larger species of shrubs and small to medium-sized trees. Avalanches are common in mountains that receive large amounts of snowfall. Suppose an avalanche occurs on a mountain and leaves a large ledge of rock exposed that had been previously covered with snow and ice. The first organisms to appear on the rocks would most likely be algae and lichens. These organisms are known to secrete acidic solutions that can cause small cracks in the surface of the rocks. As water flows into the crack and freezes and thaws with the weather, the rock will be broken into much smaller pieces on the surface. This will be the foundation for a new layer of soil. As cracks in the rocks get larger and more soil is formed through decomposition, it is easier for grass seeds that have been blown in by the wind to take hold and root into the rocky surface. The presence of these grasses will also make room for small shrubs and trees eventually. Primary succession is the initial movement of simple organisms, known as pioneer species, into new habitats that have been created. Typically, these new areas of colonization have been made available through major geological events, such as volcanoes and earthquakes. Primary succession begins with simple organisms that make way for larger and more complex species to inhabit the area. Plant life on Earth is nothing if not resilient. Have you ever taken a moment to think about the wide variety of differing habitats that exist on the planet? It's incredible, and yet from outer space, the Earth appears just blue and green. How can plants colonize every corner of the globe, especially in those extremely dry climates? Well, there is a specialized group of plants called xerophytes that survive in very dry regions. Xerophytes can live in these environments because they contain specialized features that help them prevent water loss. It's also worth noting that dry environments are not always hot. People tend to think of deserts being very dry, and they are. But so is Antarctica, and it's bitterly cold. So remember that dry areas can be hot or cold depending on geographic location. The types of adaptations possessed by xerophytes are extensive. We'll focus here on broad adaptations shared by several different species. The first adaptation has to do with their stomata and limiting water loss. Stomata are the microscopic openings in leaves that permit gas exchange. Think of them as pores for plants, and just like human pores release sweat, plant stomata release water in the form of water vapor. Certain xerophytes have a waxy covering over their stomata, thus curbing water loss. Others contain very few stomata or stomata that only open at night when it's cooler. Each of these adaptations limits water loss and allows the plant to survive in dry environments. An example of this type of plant is Adam's needle, Yucca filamentosa. This plant can survive in harsh, dry desert environments because of its waxy covering. The second type of adaptation is focused on storing water instead of just limiting water loss. To do this, plants have developed succulent leaves, plant stems, or tubers that can store water when it cannot be obtained directly from the environment. The third type of specialized adaptation used by xerophytes is focused on water intake. Some xerophytic plants have the ability to absorb surface moisture, such as dew, by using leaf hairs, while having extremely deep root systems that penetrate well below the water table. This ensures that even during drought conditions, the plants can still reach usable water. Acacia trees are xerophytic plants with deep root systems, with some roots having been found hundreds of feet underground. Specialized adaptations allow plants to survive in all types of extreme environments. Xerophytes are a group of plants that live in very dry regions. They can survive in these areas because they possess adaptations that keep them from drying out. The first adaptation are the presence of stomata, which are microscopic openings in plant leaves that release water vapor. Think of them as tiny pores. They serve to limit water loss. Plants can have waxy stomata, few stomata, or stomata that open only at night. Succulent leaves, stems, or tubers help in water storage, which helps when the surrounding environment isn't providing water. The third type of adaptation is directed at obtaining water via tiny hairs that obtain surface moisture, dew, and or extremely deep root systems.
The process of classifying living things into groups is called taxonomy, but sometimes it isn't clear how each organism should be classified. The cryptogams are an example of a group that's impossible to squish into a single taxonomic category. As a group, cryptogams are so-called lower plants that use spores to reproduce. They don't have the structures we normally associate with plants like true stems, roots, leaves, flowers, or seeds, and their reproductive parts are hidden, so we call them plant-like organisms. They share some characteristics with plants, yet they're still different enough to not fall into the same group as plants. Okay, let's take a step back for a moment and remember that all living organisms fall into 3 categories based upon their cell type, which we call domains. And within the domains are kingdoms of organisms. So we have the domain bacteria, which has one kingdom called eubacteria. Then we have the domain archaea, also with one kingdom called archaea. And last but not least, we have the domain eukarya, which has 4 kingdoms: protista, fungi, plantae, and animalia. We can further break these groups down into smaller and smaller categories, but domains and kingdoms are all you need to worry about right now. Despite sharing some characteristics with plants, only 2 groups of cryptogams are found within the kingdom Plantae. In fact, members of the cryptogam group span 4 different biological kingdoms. Now you understand why cryptogams are a hard group to classify and describe. Its members are pretty diverse. Okay, let's now take a look at the different types of organisms classified as cryptogams and where in our 4 kingdoms we may find them. In the kingdom Eubacteria, we have cyanobacteria, which was previously called blue-green algae. Then there's the kingdom Protista, where we find slime molds, green algae, and other algal groups. Next is the kingdom Fungi, which includes Basidiomycota mushrooms and their close relatives, Ascomycota cup fungi and their close relatives, and lichen. Finally, there's the kingdom Plantae, with its bryophytes, including mosses, liverworts, and hornworts, as well as the vascular cryptogams, which include ferns, horsetails, and club mosses. Phew, that's a lot of life forms with some strange names. Let's now talk about 3 common characteristics of cryptogams. Okay, so we've already learned that cryptogams don't have the same structures as most plants and that their reproductive parts are hidden. Well, some cryptogams also only reproduce asexually, meaning they don't need another organism to reproduce through the use of spores. Still, other types of cryptogams have generations that alternate between asexual reproduction and sexual reproduction, where male and female gametes from different organisms join together. Cryptogams can live in aquatic environments like algae or on land. However, those that live on land do best in shaded or humid environments. Most cryptogams need a moist environment to survive. The ferns are the only cryptogams that contain a vascular system for transporting fluids within the organism, so the other groups need to have an external water source to function. Many cryptogams are also microscopic in size. In fact, some of the organisms in the kingdom Protista are only composed of a single cell. Some are also capable of photosynthesis, meaning they can make their own food. Organisms that can make their own food or energy are called autotrophs. Still, other members of the cryptogams rely on outside sources for food, just like we do. Some of these types directly absorb nutrients from other organisms, while some gain nutrients from dead organic matter. As you can see, cryptogams are a diverse group of organisms, so it's difficult to come up with a set of traits that applies to all members. Now that we've covered the classifications and main characteristics of cryptogams, let's talk about the importance of cryptogams by introducing some fun facts about them. You might be surprised to learn that about 84% of all plant or plant-like organisms on Earth are cryptogams. In addition, algae alone are responsible for about half of all the photosynthesis that takes place, so don't forget to thank the cryptogams for your oxygen. And, as if that weren't enough, cryptogams help build the Earth's soil. They can create a layer that helps prevent erosion and traps moisture. The life cycle of cryptogams also provides nutrients, like nitrogen, to the soil, which enables other plants to grow. Okay, let's do some review to make sure everything we've learned sticks. Scientists try to classify living organisms into similar groups in a process called taxonomy, but some groups don't have a clear place. Cryptogams are so-called plant-like organisms that share some traits with plants, but most aren't found within the kingdom Plantae. Instead, they are found in the domain Eubacteria and the domain Eukarya, and in the kingdoms Plantae, fungi, Eubacteria, and Protista, and they usually live in moist environments. Some cryptogams are autotrophic, meaning they make their own food through photosynthesis, while others are heterotrophic, meaning they have to get food from other living organisms or dead organic matter. All cryptogams reproduce at least partly through the use of spores, meaning they can reproduce asexually, though some also do use sexual reproduction. Regardless of their specific characteristics, cryptogams play an important role in oxygen and soil production, making them a key part of the environment.
If you put a plant in a window, you may notice that, after a few days, it has bent toward that window. This is because the plant needs sunlight to make energy, so it grows toward the light. The growth of a plant toward a stimulus is called tropism, and the growth of a plant towards a light stimulus is called phototropism. Photo means light. Seedlings of plants grow straight upward in dark environments in order to reach the sunlight above the ground. Once they break through the surface, they start bending toward the light because the growth of cells on the dark side is faster than the cells on the light side. However, if the amount of light is the same on all sides of the plant, then it will continue to grow straight upward instead of bending. Early experiments on phototropism were based on the idea that plants were bending toward the warmth of the light, not necessarily the light itself. Charles Darwin performed many experiments in the late 1800s, and his results showed that photoreceptors, which are cells that detect light, in the tip of the plant, communicated to the lower part of the plant that curves. From this, he concluded that there must be some substance produced in the tip of the plant that is sent to the lower part of the plant, signaling it to bend. Darwin's work eventually led to the discovery of a plant hormone called auxin. Auxin is the chemical signal that causes plants to elongate and grow cells faster on the side of the plant farthest from the light. When auxin collects in the cells on the side of the stem that is away from the light source, the cells on this side are stimulated to grow. This is what causes the curvature in the stem towards the light source. There are two types of phototropism, positive and negative. Positive phototropism is growth toward a light source, and negative phototropism is growth away from a light source. Plant roots tend to exhibit negative phototropism because they need to grow deeper into the ground for plant stability, water, and nutrients. Plant roots also exhibit gravitropism, which is growth in response to gravity. Sunflowers are a great example of positive phototropism, because not only do their stems curve toward the light, but their flowers turn to face the sunlight as well. As mentioned above, your houseplants also likely exhibit phototropism. If you notice them bending toward the window, you can simply turn them around to help them grow straight again. Let's review. Most plants exhibit phototropism because it directs them towards sunlight, which they need to survive. A plant can do this because of photoreceptors in the tip of the plant that signal hormone production on the dark side of the stem. This creates an unequal amount of growth on one side of the plant, which then bends the plant towards the light, its ultimate food source.
If you were to put a plant on its side, you'd notice that after a while, the roots would start growing downward while the stem would grow upward. This response to gravity is called geotropism or gravitropism. The roots are exhibiting positive geotropism or growth with gravity, while the stem is exhibiting negative geotropism or growth against gravity. Different parts of a plant grow in opposite directions because they have different functions. The roots need to grow into the ground to provide stability for the plant, as well as to access water and nutrients. The stem needs to grow upward toward the sunlight so the plant can photosynthesize or make food from sunlight. So how do different parts of a plant know which way to grow? Plants have organelles called statuses, which settle at the bottom part of their cells and allow plants to sense gravity. They also have a hormone called auxin, which stimulates elongation and growth in plant cells. For root cells, the statuses and auxin trigger downward growth, while cells above ground are signaled to grow upward. Charles Darwin was the first to document both positive and negative geotropism in plants. He was also instrumental in correctly describing phototropism, which is plant growth toward a light source. Darwin's work eventually led to the discovery of auxin and its relationship to cell growth in plants. Scientists are now discovering that there might be other factors that trigger geotropism in plants. Some mutant varieties of plants exhibit geotropism even without statuses, though their growth is much slower. There also might be other organelles besides statuses in plant cells that contribute to geotropism. As mentioned above, if you take a houseplant and turn it on its side, it will eventually exhibit positive geotropism in the roots and negative geotropism in the stem. Likewise, if you plant a seed upside down, the roots will still grow downward, while the shoot will still grow upward. Plants that grow on steep hillsides are great examples of geotropism. They might start out growing horizontally, but they'll eventually begin to grow away from gravity and toward the sunlight. Most plants exhibit geotropism, which directs the roots down into the ground and the stem toward sunlight. This provides stability for the plant underground and a mechanism for photosynthesis above ground. Statuses and auxin are mainly responsible for both positive and negative geotropism in plants, though scientists are now finding that there might be other factors at play.
Through seed germination, a seed begins to grow up to the surface to become a seedling, and then to hopefully flourish into a plant. A seed needs proper materials to come alive from a dormant state and kickstart the process of growth. In this lesson, we'll learn what nutrients seeds need to wake up and start to grow, and we'll look at the structures that emerge in germination. In order for seeds to germinate, they need water, oxygen, correct temperatures, and sometimes light. Remember, seeds do not have leaves that are able to absorb light and carry out photosynthesis. Due to this, most of the energy is utilized via aerobic respiration, much like in an animal cell, which requires oxygen and water in order to work correctly. The seed will already have stored sugars, proteins, and other nutrients, so it will need oxygen and water to help break these apart. In our own bodies, we use oxygen and water to break chemical bonds in order to harness that energy. Seeds are very similar to us in that sense because they lack the ability to make their food like a fully grown plant will. Many plants need cold to begin the process of growing, and still others need heat, sometimes extreme heat, to spark growth. This will be dependent upon the particular type of seed that is growing. Our common houseplants, vegetables, and landscaping type plants usually require average temperatures anywhere from 50 to 80 degrees to germinate. While light may not affect seeds in the typical manner it affects grown plants, it can have some effect. Some seeds will not begin to germinate until the forest allows for enough light. This way, once the seed breaks the surface, it will be able to gather the light needed for proper growth. In order to understand how exactly germination happens, we need to learn more about types of seeds. There are two types of plants that produce seeds. Angiosperms produce seeds that are covered in a protective coating. These include fruits, such as an apple. Then we have gymnosperms, whose seeds are naked, such as pine trees. Among the angiosperms, there are monocots, with one cotyledon, and dicots, two cotyledons. The word cotyledon is literally translated as seed leaf. Cotyledons are the first leaves that are produced by the seed upon germination. If you were to cut open the seed, the cotyledon is one of the largest parts that you can see right away. Monocots include grains, bananas, palms, bamboo, and flowers that grow from bulbs like crocuses, irises, and lilies. Dicots include plants like oaks, magnolias, sunflowers, tomatoes, strawberries, squash, legumes, lettuce, and most of the plants we normally see. It's important to distinguish between monocots and dicots because they germinate differently. Regardless of the type of seed, monocots and dicots will sprout or grow somewhat similarly. In both seeds, the radicle, which is the primary root, the seed's first root, grows out of the seed and anchors itself into the ground. This allows for nutrients and water to be taken up into the seed. Once sufficient water is absorbed, an embryonic shoot emerges. This is where monocots and dicots diverge. In dicots, you can have two different types of germination, epigeal and hypogeal. The embryonic shoot has three parts: the cotyledons, the hypocotyl, the part below the cotyledons, and the epicotyl, the part above the cotyledons. In epigeal germination, the hypocotyl portion of the shoot will push through the soil and pull the rest of the shoot, the cotyledons and epicotyl, out of the soil and allow it to open up. In hypogeal germination, the epicotyl will just push through the surface of the soil, leaving everything else under the soil. You can see the differences in this side-by-side comparison. Monocot germination is different from these dicot germinations. In monocots, the radicle that pushes out of the seed has a covering called the coleoryza. The embryonic shoot that emerges also has a coating called the coleoptile. This coleoptile pushes up through the soil where the first leaves will then grow off of the shoot. Seeds need water, oxygen, proper temperatures, and sometimes proper light in order to start growing. Seeds can be either naked, such as those from gymnosperms, or covered with a fruit, like those from angiosperms. Angiosperm seeds can have different numbers of seed leaves, also known as cotyledons. Monocots have one seed leaf, dicots have two seed leaves. The radicle or primary root grows first, absorbing water, In monocots, the coleoptile pushes through the soil, sprouting leaves once it hits the surface. In dicot epigeal germination, the hypocotyl, the part below the cotyledons, pushes through the surface and pulls the rest of the shoot, sprouting leaves at the surface. In dicot hypogeal germination, the epicotyl, which is the part above the cotyledons, pushes through the surface, leaving the cotyledons below the surface.
Plant fertilization is the union of male and female gametes, reproductive cells, to produce a zygote, a fertilized egg. So how does fertilization occur? It's a pretty straightforward process that's similar for both flowering plants, angiosperms, and seed-bearing plants, gymnosperms. Let's break the fertilization process down into 4 generalized steps. Step 1. Pollination. In general, male gametes are contained in pollen, which is carried by wind, water, or wildlife, both insects and animals, to reach female gametes. The pollen is deposited on a plant's stigma, which is part of the pistil, the elongated part of a flower extending from the ovary. This process is called pollination. Step 2. Germination. Within a few minutes, pollen tubes begin growing or germinating toward the egg cell. These tubes will provide a path for the sperm carried in the pollen to reach the egg. Step 3. Penetration of the ovule. The pollen tubes penetrate the ovule, which contains the female gametes. Step 4. Fertilization. Sperm travels down the pollen tubes and fertilizes an egg. Most angiosperms undergo double fertilization, where both an egg and the polar nuclei in the embryonic sac are fertilized. After fertilization, the cells of the zygote begin dividing, and the zygote develops into an embryo or developing plant. The embryo is stored in a seed capsule, where it remains dormant until environmental conditions support its development into a new plant. There are 3 primary types of plant fertilization: porogamy, calozogamy, and mesogamy. Porogamy occurs when a pollen tube enters the ovule through a micropyle, or minute opening in the ovule. This is the most common form of plant fertilization. In calozogamy, pollen tubes enter the ovule through the chalaza, located opposite the micropyle. And in mesogamy, these tubes enter the ovule through its integument, outermost layer or layers. So let's review. Plant fertilization is the joining of male and female gametes resulting in a zygote. The most generalized form of this process requires 4 steps: pollination, germination, penetration of the ovule, and fertilization. After fertilization, the zygote divides to form an embryo. Double fertilization is common in angiosperms. The 3 main types of plant fertilization, porogamy, calozogamy, and mesogamy, are distinguished by how the pollen tube enters the ovule. Of these 3, porogamy is most common.
Have you ever looked in your garden to see the changes that plants go through during the year? Plants go through complex cycles as the seasons change. Some plants live their lives to the fullest during spring, summer, and fall, and die completely when winter comes. Other plants spend spring and summer growing and getting ready for fall and winter by storing nutrients to use during the difficult winter months. A tool that plants use to store their nutrients and energy is the tuber. A tuber is a modified plant structure that is enlarged and used as a storage container for nutrients that would be difficult to obtain during dry periods and the winter months. There are basically 2 types of tubers recognized by botanists: stem tubers and root tubers. Stem tubers form as thickened rhizomes or stolons. They normally form at the side of parent plants and are located near the surface of the soil. Stems grow out from the tubers on the top side, and roots grow from the bottom side of stem tubers. When stem tuber plants reproduce, the offspring grow from a root structure known as a rhizome. These plants store their food in the tubers of their offspring. The parent plant dies in the fall, and the nutrients in the tuber provide energy for the offspring to grow in the spring. Root tubers are modified lateral roots that swell to form a storage organ. The enlarged area of the root may be one end of the root or the entire root of the plant. Plants with root tubers are perennials. This means they are able to survive off the nutrients stored in their roots during the winter or dry period and will regenerate during the spring. This is different from stem tuber plants that use their tubers to support offspring. Common examples of plants with tubers include potatoes, cassava, and dahlias. Potatoes are one of the most commonly eaten foods on Earth. Did you know that when you bite into French fries or baked potato, you are eating a stem tuber? Potatoes develop from enlarged stolons that have developed into storage organs. Potatoes have all the parts of a traditional stem, including nodes, internodes, and premature leaves. One initial cutting from a potato can be planted in the spring, and it will grow into a large plant. New potatoes grow off the stem just below the surface of the soil and store nutrients so that each one can grow into a new plant during the following year. Do you like to eat tapioca? It is derived from the cassava plant. Cassava is the third largest source of carbohydrates in the tropics. Cassava plants have root tubers. The root of the plant is modified into one large tuber that is used to store protein and carbohydrates in the form of starch. Tubers are not only found in plants that we eat. Flowers use tubers as well to store energy and survive to the following year. An example of this is the magnificent dahlia. The dahlia is the national flower of Mexico and is known to be propagated from its tubers. Each plant grows multiple root tubers that it uses to store nutrients to survive through the winter, so it can begin to grow again the following spring. Let's review. Tubers are plant parts that are enlarged to form storage containers that the plants use to hold nutrients that allow them to feed their offspring or to survive through the winter. There are two basic types of tubers, root tubers and stem tubers. Common examples of tubers include potatoes, cassava, and dahlias.
Ancient Earth had its fair share of dominant organisms. For most of the history of life, Earth has been dominated by the microscopic bacteria and archaea, floating, swimming, and thriving in the ancient oceans that covered much of the globe. Through time, organisms got larger and more complex. There was a time when organisms resembling insects were dominant, then fish and reptiles. We've all heard of the dinosaurs ruling Earth, followed by mammals evolving. Now, humans are clearly the dominant organism. But there was a time in history you probably have never heard of, the age of the fungus. Around 250 million years ago, the Earth was populated by ancient plants and animals. The Permian extinction changed all that. Over 90% of life on Earth vanished. The animals were decimated, and so were the plants. This included many millions to billions of trees, leaving massive fields of dead wood. And what thrives on dead wood and dead animal tissue? That's right, fungi. With few animals to trample on them or eat them, few plants to crowd them, and a massive dead wood buffet, the fungi after the Permian extinction were able to dominate the landscape. The little known and even less heralded golden age of the fungus had begun. Fungi, singular fungus, is a group of eukaryotic, non-phototrophic organisms with rigid cell walls that includes mushrooms, molds, and yeasts. This definition has some words in it that probably need definitions of their own. Eukaryotic simply means that fungal cells have a nucleus, like plant and animal cells, which distinguishes them from the bacteria and archaea. Non-phototrophic means that they can't use light for energy because they lack chlorophyll, distinguishing them from plants. The cell walls of fungi are unique in that they contain large amounts of chitin, a structural component only found in the cell walls of fungi. The chitin makes the cell walls rigid. So, if fungi can't perform photosynthesis, and I've never seen a mushroom hunting game on the savanna, what do they do for nutrients? Many fungi are saprophytes. A saprophyte is an organism that acquires nutrients from dead organic matter. This can be as common as seeing mushrooms growing on dead wood, or mold surviving in your refrigerator on last week's takeout food. These fungi are actually very important to the health of the ecosystem, rapidly breaking down plant and animal material and returning it to a more usable form. Other fungi are parasites, obtaining nutrients from a host species. Many common plant diseases are a result of a fungus siphoning nutrition from the plant. There are even some human diseases caused by fungi, but we'll talk more in depth about those later. Many fungi are common contaminants of foods. Most fungi reproduce by releasing spores,
which are the resistant, reproductive, resting stage of a fungus. These spores are able to survive the high temperatures and pH extremes often used for food preservation. Spores are also not killed by freezing, and some can even germinate, resulting in actively growing fungi in your freezer. There are 3 major types of fungus: mushrooms, molds, and yeasts. Let's take a quick look at each. To the average person, mushrooms are probably the best known fungi. Usually found springing up on dead wood after cool, wet weather, the mushroom is a common sight. Mushrooms generally consist of a stalk with a large cap on top. This cap produces the spores that the mushroom releases in order to reproduce and colonize new environments. Many species of mushrooms are edible and delicious, but others can be very toxic. Since knowing the difference can take a very experienced eye, eating wild mushrooms is usually not a fantastic idea for the average person. Like mushrooms, molds are very recognizable. Forget about that brick of cheddar cheese for too long, and suddenly it's covered with green fuzz. That fuzz is why molds are often called filamentous fungi, and the filaments give it that fuzzy or fluffy appearance. Hyphae is a technical term for the long, branching mold filaments. Specialized hyphae, called conidia, are often pigmented and reach up above the surface of the food source. These conidia are responsible for producing and releasing the mold spores for reproduction and dispersal. The common and ubiquitous bread mold is in the genus Rhizopus. To a microbiologist, yeasts are the most important fungi, so I'll spend a little more time here. Yeast is the common term for any unicellular fungus. The mushrooms and molds are all multicellular organisms, but yeasts usually exist as only a single cell. The cell is larger than a bacterial cell and contains a nucleus and other organelles. Yeast reproduce by budding. The new offspring cell begins as a small outgrowth on the membrane of the parent cell, which gets larger before eventually pinching off. This process is asexual, resulting in 2 clones. Some yeast can reproduce sexually by simply fusing and creating a genetically recombined zygote cell. The zygote contains DNA from both parents. Yeasts are unicellular, yet eukaryotic, making them an important group of organisms for scientists. Humans are also eukaryotic, so performing experiments on yeast, like investigating cellular processes or testing new drugs, can provide data that can be applied to humans more accurately than work done on the more distantly related bacteria. Saccharomyces cerevisiae is the most common yeast used in research as a model organism for higher eukaryotes. In 1997, it gained the distinction of being the first eukaryote to have its genome sequenced. Saccharomyces cerevisiae might sound familiar to you for another reason. Bakers and brewers have been using this yeast for many years to make bread and alcohol. S. cerevisiae thrives by fermenting sugars, releasing carbon dioxide or ethanol in the process. The bakers and brewers began experimenting on Saccharomyces in an attempt to optimize growth and production, which expanded the knowledge base and may have led the way to its use as a model organism in research. We briefly mentioned that some fungi can cause disease in humans. Mycosis is a term given to any illness caused by the growth of a fungus either on or inside the body. There are several important human diseases caused by fungi, but they usually infect the body's surfaces or respiratory tract and are rarely serious for healthy individuals. It's a good thing, too, because fungal infections can be difficult to eliminate. As a fellow eukaryote, most of the medications that will kill a fungus are also toxic to human cells, often producing nasty side effects. Ringworm is a common fungal disease passed between infected individuals or picked up from contaminated surfaces. Trichophyton and Microsporum are the 2 most common genera of fungi that causes the characteristic ring-shaped scaly lesions. Candida albicans is another pathogen that causes several diseases in humans, depending on where it is located. Candida in the vaginal canal can cause yeast infections, characterized by itchiness and discharge. Candida in the mouth is called thrush, characterized by white spots that grow together, forming a mass of crumbly, white, milk-like curds. Let's review. The fungi are a group of eukaryotic, non-phototrophic organisms with rigid cell walls. This includes mushrooms, molds, and yeasts. Many fungi are saprophytes, getting nutrients by absorbing them from dead, decaying plant material. Still other fungi are parasitic, stealing resources from a host organism. Many fungi reproduce by releasing spores. These resistant reproductive structures are often food contaminants due to their ability to survive most common food preservation methods. Mushrooms are multicellular fungi with stalks and caps that are most often found growing on decaying plant material. Molds are multicellular filamentous fungi that are commonly found on foods. Yeasts are any unicellular fungus. Being single-celled and eukaryotic, yeasts make excellent research organisms for studying processes that can be applied to humans as well. Saccharomyces cerevisiae is a research model organism that is also used to make bread rise and beverages alcoholic. A mycosis is a disease caused by the growth of a fungus on or inside a human body. Most mycotic diseases infect the body's surfaces or respiratory tract and are not considered serious diseases. Treating fungal infections can be difficult, as most drugs that target fungi will also harm human cells.
When you hear the word fungus, you probably think of a mushroom. But fungi are so much more than that. In fact, when you think of mushrooms, which make up only part of the fungi kingdom, you can probably think of several different types. Button, chanterelle, shiitake, portobello, and so many more. Still not a believer in the diversity of fungi? Think about this. Scientists have already described over 100,000 species of fungi, though there may be many more, possibly as many as 1.5 million. The fungi species that have been described are classified into 5 groups. The names are tricky, but you may notice that each group ends in the suffix mycetes, so whether you can remember the tongue-twisting names or not, if you see this suffix, you'll know it's a fungus. Our first group is Chytridiomycetes, thought to be the oldest group of fungi. We often find these fungi in lakes, ponds, estuaries, and soil. Many species in this group are decomposers, meaning that they break down dead organic material. But other species are parasites of plants and animals and are responsible for many infections in amphibians. Next, we have the group Zygomycetes, which you have probably interacted with at some point. Some fungi in this group are fast-growing molds, such as black bread mold and molds that rot the fruit you left sitting out on the counter too long. So, should you eat that rotting fruit? Zy wouldn't go near it. Our third group is Glomeromycetes, which are very important fungi. This is because about 90% of plants form symbiotic relationships with Glomeromycetes, which help deliver nutrients to the plant while receiving other necessary nutrients in return. Like a conglomerate is multiple parts combined together, Glomeromycetes combine with plants to form a strong team. Fourth, we have the group Ascomycetes, also known as sac fungi. These fungi get their name from their sac-like structures called asci. Asci comes from the Greek asko, which means pouch, so literally this group name means pouch fungus. These live in a wide variety of habitats, marine, freshwater, and terrestrial, and can range in size from unicellular yeast to fancy morel mushrooms. Yum! Not always the good guy though, Ascomycetes are also some of the most devastating plant pathogens. Finally, we have the group Basidiomycetes, also known as club fungus. This is what most people imagine when they hear the word fungus because these are many of the mushrooms, puffballs, and shelf fungi. They get their name from their club-like reproductive structure called a basidium, which literally means little pedestal in Latin. Now imagine these types of fungi again, and you can see how they got their name. Many of these fungi are decomposers, like the shelf fungi that break down wood in forests, but they also include many devastating plant parasites as well. You just got some big terms that will take some time to digest, so let's talk about a few that you are probably somewhat familiar with already. I mentioned mold earlier, and this you have likely seen in many forms on your food or on your shower walls. Mold is simply any rapidly growing fungus that produces spores asexually. I'm sure you've seen how quickly mold can grow if you went away for the weekend and left some fruit sitting out on the counter. I also mentioned yeast, which are single-celled fungi. Yeast also reproduce asexually and really like liquid or moist habitats. And to show you that not all fungi are bad, yeast are responsible for delicious things like beer and bread before you let it get moldy. Lichen are really special because they are a symbiotic partnership of fungi and photosynthetic organisms. In this case, the photosynthetic organisms are photosynthetic algae and cyanobacteria, and you've seen this as different forms of moss. The partners are so closely woven together that you'd be hard-pressed to see them as separate organisms. In fact, the partnership is so important that these fungi, which partner to form lichens, are rarely able to survive on their own. The two organisms help each other in the symbiosis, providing habitat, nutrients, and water. Similarly, mycorrhiza is a symbiotic relationship between plant roots and fungi. The term mycorrhiza literally means fungus root. The fungi in this relationship surround the plant root but don't penetrate the cells. The fungi absorb nutrients from the soil and share them with the plant. In return, the plant provides sugars from photosynthesis to the fungi, making them both happy campers. And, as mentioned before, about 90% of plants have this type of symbiosis with glomeromycetes, since they form a conglomerate of plant and fungi. The diversity of fungi is astounding. Though there are already over 100,000 described species of fungi, there are likely hundreds of thousands more that have not been described. Fungi are classified into 5 groups: Chytridiomycetes, Zygomycetes, Glomeromycetes, Ascomycetes, and Basidiomycetes. The Chytridiomycetes are the oldest group of fungi. The Zygomycetes are responsible for food molds. Glomeromycetes helps form the conglomerate group of fungi and plant roots. Ascomycetes are the sac fungi, from the Greek asco for pouch, and Basidiomycetes are what most people think of when they think of fungi, the little pedestal fungi. Fungi are both beneficial and harmful. Molds are rapidly growing fungi that take over our foods when we leave them sitting out too long, while yeasts are single-celled fungi that make delicious bread and beer. Lichens and mycorrhizae are both symbiotic relationships with photosynthetic organisms, but lichens are symbioses with algae and cyanobacteria, while mycorrhizae are symbioses with plant roots. Both relationships help both the photosynthesizer and the fungi as they provide habitat and nutrients to each other while they share their space. The fungi kingdom is a unique and diverse group of organisms. Fungi not only provide a tasty addition to your stir-fry, they also have many ecological benefits as well. In another lesson, we learned about lichens and mycorrhizae, which are both symbiotic relationships involving fungi. Lichens are symbioses with photosynthetic organisms, specifically algae and cyanobacteria. Mycorrhizae are symbioses with plant roots. Both types of symbiotic relationships provide nutritional and habitat benefits for the fungi and other organisms involved. Fungi are also essential recyclers in the environment. As decomposers, they break down organic material and restock the environment with usable nutrients. If we didn't have fungi breaking things down, we'd just have a large pile of dead stuff all over the place, and that certainly wouldn't be good. Not only do we not want to see dead stuff piling up, but the nutrients in those dead things need to get returned to the earth so that they can be used by new, growing plants and animals. Your stir-fry is just one way that fungi are used in food. Ever had blue cheese? What makes it blue, and so tasty, is the fungi used to ripen the cheese. Truffles are highly prized fungi that people eat, and yeasts are fungi that make beer and bread. Fungi are also beneficial in the medical and pharmaceutical industry. Some fungi produce antibiotics that are used to treat bacterial diseases like penicillin. Some yeasts are also used in biomedical research to produce human proteins. While you may enjoy some tasty blue cheese, you probably don't want to eat just any moldy cheese because it will likely make you sick. This is because there are many fungi that are harmful instead of beneficial. In fact, about 1/3 of the known species of fungi are parasites or pathogens, mostly occurring in or on plants. Have you heard of Dutch elm disease? This was a harmful fungus that was accidentally introduced into the U.S. in the early 1920s, and it eventually destroyed about 70% of the elm trees in the eastern part of the country. Fungi are also serious pests in agriculture. Many crop fields these days are made of genetically identical individuals, which makes them the perfect target for disease. About 80% of plant diseases are caused by fungi, and a large variety of fungi are common on grain crops. Many of these plant pathogens can be harmful to humans and other animals. Eating infected crops can cause us to become very sick or even die. But the good news is that most animals are not as susceptible to parasitic fungi directly. Compared to 1/3 of fungi species being harmful to plants, only about 50 fungi species are known parasites to animals. You have probably heard of some of these too. Athlete's foot, yeast infections, and some lung diseases are all caused by fungi. Even ringworm is a fungus. That's right, it's not a parasitic worm, it just looks like one, which is how it got its name. With over 100,000 known species, fungi are quite diverse and abundant on Earth. The bad news is that many of them can be quite harmful, especially to plants. About 1/3 of the known fungi species are plant parasites or pathogens. Fungi are responsible for many crop diseases, as well as wiping out larger species like elms in the eastern U.S. The good news is that only about 50 species are known animal parasites, so as long as we don't ingest the infected plants, we're fairly safe. Fungi also have many benefits. In the environment, they serve as decomposers, which break down dead organic material. This is an important job because otherwise that material would just build up everywhere. This process also serves to recycle nutrients back into the ground so that they are usable by new plants and animals. Fungi also provide us with many interesting foods such as mushrooms, cheeses, and breads. Additionally, they have been very helpful to the medical industry because they can help treat things like bacterial diseases.
Have you ever seen a fungal truffle? Have you eaten one? Fungal truffles are part of a group of fungi called Ascomycota. Ascomycota literally translates to sac fungi. However, these fungi come in many forms or shapes, such as spongy, long, skinny cylinders, cup-shaped, coral-like, and others. Fungi is one of 6 kingdoms used to classify organisms. Within each kingdom are subcategories that further organize organisms. Ascomycota is one of these subgroups categorized as a phylum. A phylum simply organizes organisms from a kingdom into subcategories based on similar characteristics. Ascomycota form the largest phylum within fungi and live all over the earth. In the alternative medicine world, a prime example of Ascomycota fungi is the Cordyceps ascomycota, which is used to help support the immune system. These organisms grow on other creatures, like insects and worms, and can be red, yellow, brown, or orange. If you are a foodie, you might have had the pleasure of eating morel mushrooms. These Ascomycota are gathered after forests burn and are often available at specialty gourmet shops. Other examples of Ascomycota include those that grow with lichen in the alpine and tundra regions of the world. These have close relationships with algae, where the fungi and algae swap nutrients for survival without hindering the other, which is known as a symbiotic relationship. There are also Ascomycota that are carnivorous and eat other organisms that don't photosynthesize, such as ringworms, amoeba, protists, and other creatures that live in the soil. They trap these creatures by forming loop structures around them, like handcuffs. There are also parasitic Ascomycota, such as the blue stain fungi of pine trees. These Ascomycota create a wonderful blue pattern in the wood of pine trees, but ultimately cause the trees to die. Have you ever found a blue mold growing on bread? This mold, which is also known as penicillium, is used in the production of both food and medicine. If you have ever made bread, the yeast that's used to help make the bread rise is part of the Ascomycota phylum too. Additionally, any fermented food or beverage has also come into contact with Ascomycota. Many plant pathogens that cause crop disease are Ascomycota fungi too, such as ergot and aspergillus in peanuts. Ascomycota is the largest group of fungi on the planet that lives virtually everywhere. This fungi comes in many shapes and sizes, can be edible, can eat other organisms, and can survive in the unfavorable conditions of the alpine and tundra regions of the world. Common examples of Ascomycota are cordyceps, morel mushrooms, blue-stained tree fungi, ergot, and those associated with lichen that form symbiotic relationships with algae.
So, you may recall that the somatic nervous system is the part of the nervous system that voluntarily responds to external stimuli, and that the autonomic nervous system is the part of the nervous system that involuntarily regulates internal body functions. Additionally, the autonomic nervous system can be further subdivided into two divisions: the sympathetic nervous system and the parasympathetic nervous system. Both of these systems control the same group of body functions, but they have opposite effects on the functions that they regulate. The sympathetic nervous system prepares the body for intense physical activity and is often referred to as the fight or flight response. The parasympathetic nervous system has almost the exact opposite effect and relaxes the body and inhibits or slows many high-energy functions. The effects of the parasympathetic nervous system can be summarized by the phrase rest and digest. Let's look at an example of how these two systems function in response to changes in the environment. This is Phil. Phil is hiking in the wilderness all by himself and enjoying the tranquility of the great outdoors when he turns a corner and finds himself only feet away from a full-grown bear. In a fraction of a second, the brain recognizes the animal in front of him as a bear and classifies it as a very big threat. Immediately, the sympathetic nervous system is activated, and without any conscious control by Phil at all, several body functions are affected all at once. Number 1. Heart rate is increased. Increased heart rate results in an increase in oxygen and nutrients that reach the brain and muscles, preparing them to deal with whatever Phil will have to face. Number 2. The liver is stimulated to release glucose into the bloodstream, providing more energy that will be ready to power the muscles in case it is needed. Number 3. The bronchioles in the lungs are dilated to allow more air into the lungs, which will increase oxygenation of the blood and keep up with the increased flow of blood through the lungs due to the increased heart rate. Number 4. The pupils of the eyes are dilated. Because the sympathetic nervous system is often activated when people are surprised, pupil dilation is a visual cue that we use to read surprise on people's faces. Number 5. The adrenal glands are stimulated to secrete epinephrine and norepinephrine. The adrenal glands are a pair of hormone-producing glands located on top of the kidneys that respond to stress. Together, the epinephrine and norepinephrine secreted by the adrenal glands have the same basic effects as the nerves of the sympathetic nervous system by increasing heart rate, increasing bronchial dilation, and increasing glucose release from the liver. In addition, norepinephrine is also known to increase alertness. It may seem redundant that these hormones have the same actions as the sympathetic neurons, but hormones have longer lasting effects than nerve impulses, so while the initial fight or flight response is mediated by neurons, these hormones serve to reinforce and help to sustain the response. Number 6. Digestive activity is inhibited. In the moment of truth, a person may need every last ounce of energy they can muster. If Phil makes it through this ordeal, then he'll have plenty of time to complete digestion of whatever he ate for breakfast. Number 7. The bladder is relaxed. After all, this isn't the time for a person to relieve themselves. There are more pressing matters at hand. Even so, I'll bet some of you out there are thinking, hold on, I thought that people wet themselves when they got scared. So it turns out that the sympathetic nervous system is only activated in situations where the brain decides there is a reasonable chance of survival. In very extreme cases of danger, where the chances of survival seem remote, crippling fear often takes over and people can lose bladder and bowel control. They may also go limp and play possum as last-ditch efforts to survive. This is not a complete list of all the functions regulated by the sympathetic nervous system, but it does include most of the major functions that would come into play in Phil's situation with the bear. As it turns out, Phil opted for flight over fight and ran away. He ran for almost a whole half-mile before he dared to stop and turn around, but he could have easily run further if the need was there, because his sympathetic nervous system had prepared him almost instantly to run for his life. When Phil turns around and sees that the bear is nowhere to be found, his parasympathetic nervous system begins the process of relaxing and calming the body so that it can recover from the heightened physical and metabolic activity. Some of the effects of the parasympathetic nervous system are: number 1, heart rate is decreased. With the danger behind him, Phil no longer needs increased blood supply to his muscles, so the heart can slow down and conserve energy. Number 2, the liver stops releasing glucose into the blood and instead focuses its activity on producing bile and detoxifying dangerous substances. Number 3, the bronchioles in the lung are constricted to allow less air into the lungs. Now that heart rate is decreased and oxygen consumption in the muscles will be lower, the body doesn't need as much oxygen. Number 4, the pupils of the eyes constrict back to a more normal size. Number 5, the adrenal glands stop secreting epinephrine and norepinephrine. Now that the danger is past, it's time for the body to relax and recover, and these two hormones are counterproductive to resting and relaxation. Number 6, digestive activity is stimulated. Phil survived his encounter with the bear, and his body can now continue digesting his breakfast. He'll need the energy to replace some of the reserves he used to run away from the bear. Number 7, the bladder contracts. Without an immediate threat around, Phil may now feel the need to relieve himself if he has a full bladder. Again, this is not a complete list of all the functions regulated by the parasympathetic nervous system, but it does include most of the major functions that would be affected in this situation. In summary, there are two main divisions of the autonomic nervous system. The sympathetic nervous system prepares the body for intense physical activity and is often referred to as the fight or flight response. The parasympathetic nervous system has almost the exact opposite effect and relaxes the body and inhibits or slows many high-energy functions. It's pretty much a rest and digest response. When an immediate threat is present, the functions of the sympathetic nervous system prepare the body for extreme physical activity and include increased heart rate, glucose release from the liver, bronchial dilation, pupil dilation, secretion of epinephrine and norepinephrine from the adrenal glands, relaxation of the bladder, and inhibition of digestive activity. Once the threat is gone, the functions of the parasympathetic nervous system promote relaxation and recovery of the body and include decreased heart rate, bile production in the liver, bronchial constriction, pupil constriction, inhibition of the adrenal glands, contraction of the bladder, and stimulation of digestive activity. This is Nur. He is a biologist who has come to Earth to study human neurobiology. This is Phil. He is Nur's research subject. One night, Nur and his research assistant Blur brought Phil on board their research ship while he was sleeping and implanted a brain monitoring device. They then released the subject back into his native habitat, his house, without him ever knowing what happened. Now Nur and Blair can monitor Phil's every thought and emotion and map the parts of his brain that are stimulated in any given situation. They can even see and hear everything that Phil's eyes and ears can. After 3 weeks of monitoring Phil's brain activity, his different body functions, his thoughts, and even tracing some of the neural signals sent to and from the brain, all of the data has now been processed by the research ship's powerful central computer. Now the results are in, and Nur is not disappointed. In front of him is a diagram of the human brain with a list of biological functions associated with each part. Let's take a look at that diagram and see what Nur discovered about the human brain. This diagram shows basically what the human brain looks like if it was cut in half, just about in the middle between the right and left sides, so that Nur can see what the structures in the middle of the brain look like. We'll start with the brainstem, which is the part of the brain that connects to the spinal cord. Nur discovered that the brainstem regulated many vital but basic processes such as heart rate, breathing, blood pressure, swallowing, and digestion. This wasn't too big a surprise, because these are mostly autonomic functions and the brainstem controls them without Phil having to think about them. It makes sense that these functions are controlled by the part of the brain closest to the spinal cord, because there is no reason for the signal to go any further into the conscious part of Phil's brain to make a decision. For the most part, the brainstem can handle the relatively simple processing required to regulate these autonomic functions on its own. Located just above the brainstem is the hippocampus. The hippocampus is critical for the formation of new memories and spatial orientation. NERS studies indicated that this part of the brain might be important for memory formation and spatial orientation. However, we know that these are critical roles of the hippocampus because several studies of people with extensive hippocampal damage have shown that they cannot form new long-term memories after the damage and find themselves easily disoriented. In addition, in Alzheimer's disease, the hippocampus is one of the first parts of the brain that sustains damage, and memory loss and disorientation are among the first symptoms of the disease. Above the forward part of the hippocampus is a brain structure called the hypothalamus. The hypothalamus is the part of the brain that tells the body what it needs to do to survive and reproduce. It controls the internal thermostat of the body, hunger, thirst, the fight or flight response, the rest and digest response, and mating behavior. About a week ago, Blur observed a major stimulation of the hypothalamus when Phil came across a bear on a hike, and the fight or flight response was activated by the hypothalamus. After Phil was a safe distance away from the bear, the fight or flight response was shut down, and the hypothalamus activated the rest and digest response to allow Phil to recover from his close call with the bear. A little bit above and behind the hypothalamus is the thalamus, which is the relay station for sensory information to the cerebral cortex. The thalamus receives sensory signals from all the sensory systems except for the olfactory system. It then processes the signals and sends them along to their associated area of the cerebral cortex, where they are integrated with the conscious mind. The thalamus also controls Phil's conscious state, or basically if he is asleep or awake, and how alert he is. Sleep is basically a state where no sensory signals are sent to the cerebral cortex, and therefore the person is not aware of any of their senses. The sensory organs are still functioning and sending signals to the thalamus, but during sleep the thalamus doesn't send the signals to the cerebral cortex. The olfactory bulbs are the structures in the brain that process signals from the olfactory sensory neurons. Even though Ner doesn't know the name of these structures, he had been fairly certain that they were involved in processing the sense of smell, because they are located right next to the chemoreceptors in the nasal cavity that humans use to sense odors, and the chemoreceptors send signals directly to the olfactory bulbs. But it was good to get confirmation from the computer analysis anyway. The cerebellum is located in the back of the skull behind the brainstem and coordinates movement, balance, and hand-eye coordination. The cerebellum does not initiate body movements. Instead, the cerebellum receives sensory inputs about the states of the muscles and the positioning of all the parts of the body, as well as visual input. It then fine-tunes the motor outputs to adjust the positions of the body parts to maintain balance and coordinate body movements to achieve the desired action. So let's review. The brainstem is the part of the brain that connects to the spinal cord. The brainstem regulates many vital but basic processes such as heart rate, breathing, blood pressure, swallowing, and digestion. Located just above the brainstem is the hippocampus. The hippocampus is a brain structure which is critical for the formation of new memories and spatial orientation. Above the forward part of the hippocampus is a brain structure called the hypothalamus. The hypothalamus is the part of the brain that tells the body what it needs to do to survive and reproduce. It controls the internal thermostat of the body, hunger, thirst, the fight or flight response, the rest and digest response, and mating behavior. A little bit above and behind the hypothalamus is the thalamus, which is the relay station for sensory information to the cerebral cortex. The thalamus receives sensory signals from all the sensory organs except for the olfactory system. It then processes the signals and sends them along to their associated area of the cerebral cortex, where they are integrated with the conscious mind. The olfactory bulbs are the structures in the brain that process signals from the olfactory sensory neurons. And finally, the cerebellum is located in the back of the skull behind the brainstem and coordinates movement, balance, and hand-eye coordination. When we last saw Nur and Blur, they had just gotten the results from the first 3 weeks' worth of data from monitoring their research subject Phil. Now it's the day after, and they're about to start their weekly team meeting to discuss how their research is going. Blur is especially excited about the data and the new technology they're using to gather it.
I just love the new Brainulator 8000. It's such a relief not to have to use the old Probe King 4000.
We got some great results yesterday, but some of the data, especially the data about the cerebral cortex, was inconclusive. I think we should monitor the subject around the clock and enable the thought scan. Then maybe we can make sense of the cerebral cortex. There were nods around the table. The cerebral cortex is the largest part of the human brain that surrounds most of the other brain structures. From the data analysis, it appeared that the cerebral cortex was responsible for quite a variety of functions and many complex thinking processes, which made the data hard to sort out and get conclusive answers from. Nur and his group decided to divide the cerebral cortex up into 4 different parts, based on what appeared to be naturally occurring dividing lines in the brain. They called these parts the temporal lobe, the frontal lobe, the parietal lobe, and the occipital lobe. Hopefully, continuous thought monitoring and dividing the cortex up into 4 parts would help make some sense of its functions. So not more than a couple of hours later, Blur was in the control room, monitoring what Phil saw, heard, felt, and thought. It was a Thursday night, and Phil was searching around his house for something. Blur could see from his thoughts that Phil was trying to remember where he put something, a piece of paper that he had written a number on. Blur could also see that the temporal lobe of the cerebral cortex, or the lobe in the lower central part of the cerebral cortex, was showing lots of activity. Then Phil remembered that he had left the piece of paper in his car. He went to the car and retrieved the paper, and the activity in the temporal lobe came back down to normal levels. Blur noted the time and then entered an observation into the computer. Temporal lobe activity associated with memory. Now Phil had picked up the phone and dialed a number. When he started talking on the phone, Blur noticed another increase in brain activity in the temporal lobe, as well as an increase in brain activity in the frontal lobe, or the forward region of the cerebral cortex. In fact, it actually looked like the frontal lobe had more activity when Phil was talking, and the temporal lobe had more activity when he was listening. Blur made some more notes in the computer. Temporal lobe activity associated with hearing and language comprehension. Frontal lobe activity associated with speech. Phil spent most of the night on the phone and then went to bed. But Blur wasn't tired. In fact, he had an idea about how to test complex human brain functions. He beamed himself into Phil's garage and went to work on his car. First he slashed Phil's tire. Then he disconnected the battery. Satisfied that he had created a good test, Blur beamed back to the ship and waited for Phil to wake up and discover his handiwork. The next morning, Phil got in his car to drive to work without noticing the slashed tire, but the car wouldn't start. He tried again and again, but still nothing. Frustrated, Phil slammed his fist against the steering wheel, got out of the car, and slammed the door. He went to kick the tire and realized that it was flat, which just upset him more. Meanwhile, Blur couldn't have been happier. He had generated a major emotional response in Phil, and that response was corresponding with an increase in brain activity in the frontal lobe. He made another note in the computer. Frontal lobe activity associated with emotional outburst. Phil began to calm down and started thinking about what he should do next. Blur noticed something interesting. As Phil calmed down and he started planning his next move, a different part of the frontal lobe started showing more activity. Phil popped the hood and looked around. He noticed that the battery was disconnected and hooked it back up. Then he went to work changing the tire. Again, Blur noticed a new area of the frontal lobe was being stimulated, as well as a portion of the parietal lobe, or the top central lobe of the cerebral cortex. He made more notes in the computer. Frontal lobe activity associated with planning and motor function. Parietal lobe activity associated with motor function. The rest of Phil's day was uneventful. filled with lots of everyday functions and stimulation of multiple areas of the brain that the computer was better suited to sorting out than Blur was. But that evening was a different story because Phil had a date. Like a gentleman, Phil opened the door for his date, and after she got inside, he closed the door, trying to look cool as he did it. But this was just not his day. Poor Phil shut the door on his own hand. Blur noticed something really interesting now. There was a sudden burst of activity in a localized area of Phil's parietal lobe, but only on the right side, even though it was Phil's left hand that had been slammed in the door. Blur double-checked to make sure, and yes, Phil's left hand was in pain, but it was the right side of the parietal lobe that was stimulated. So Blur made a note: parietal lobe activity associated with pain, but on opposite side of the body from the stimulus. Phil assured his date that he was fine. He took out the bottle of wine and put his still aching hand into the bucket. The ice water was very cold, but it dulled the pain. His date laughed, but Phil figured it could be worse. At least she seemed to be having a good time. Meanwhile, Blur noticed that the same exact area of the parietal lobe that had been stimulated when Phil slammed his hand in the door was now being stimulated by his hands sensing the cold water. And again, the stimulation was on the opposite side of the brain from the hand feeling the sensation. He made another note: parietal lobe activity associated with temperature, but on opposite side of the body from the stimulus. The rest of the dinner passed without incident, and then it was time for the check. Phil started figuring out the tip. He wanted to make sure that he didn't leave too little for the waiter and look like he was cheap, but he also didn't want to spend more than he had to, so it was time to do a quick calculation. Blur noticed that as Phil did his calculation, yet another region of the parietal lobe was being stimulated, so again he made a note: parietal lobe activity associated with mathematics. After dinner, Phil took his date to the Modern Art Museum. He figured taking his date to a museum would make him look cultured, and taking her to a modern art museum would make him also seem cool and knowledgeable about current trends. Neither of these was actually the case. He just liked looking at paintings. After all, that's what they were there for, right? Blur noticed that while in the museum, Phil's occipital lobe, or the lobe located at the very back of the cerebral cortex, was consistently being stimulated far more than any of the other lobes of the cortex. He also noticed that Phil wasn't really thinking about much at all. He was just taking in all of the visual cues around him and enjoying the scenery. So Blur made another note: occipital lobe activity associated with processing visual information. The museum was closing, so Blur decided to call it a night. He thought that he had enough now to assign at least 1 or 2 functions to each lobe of the cortex, and besides, everything was closing, so there really wasn't much more that could happen on this date. At the next team meeting, Blur presented his results to Nur and the rest of the research team.
Through careful observation and by presenting a challenge for the research subject to overcome, I have been able to map several functions to the 4 lobes of the cerebral cortex. The temporal lobe is the lobe in the lower central part of the cerebral cortex, located here, and it appears to be important for memory, hearing, and language comprehension. The frontal lobe is the forward region of the cerebral cortex, located here. It appears to be important for speech, emotions, planning, and motor function. The parietal lobe is the top central lobe of the cerebral cortex, located here. It appears to be important for motor function and integrating senses like pain, touch, and temperature. And finally, the occipital lobe is the lobe located at the very back of the cerebral cortex right here. It appears to be important for processing visual information.
As you think, see, breathe, and do everything throughout the day, you are using your central nervous system. It's the system of the body that receives and processes all information from all parts of the body. It consists of the brain, the spinal cord, and neurons. It's arguably the most important system of the body. Neurons are the basic units that make up the nervous system. All cells of the nervous system are made of neurons. Neurons are able to transmit electrical impulses throughout the body as signals that cause a response. There are 3 types of neurons, known as motor, sensory, or interneurons. Motor neurons transmit information between organs, glands, and muscles. Sensory neurons send information to the brain and spinal cord from internal organs or external stimuli encountered by the parts of the body. Interneurons act as messengers that relate signals between motor and sensory neurons. The brain is the control center of the body. The brain is typically considered to have three main parts: the forebrain, the hindbrain, and the brainstem. The forebrain receives and processes sensory information, such as thinking, understanding and producing language, and motor functions. The forebrain consists of the thalamus, hypothalamus, cerebrum, and cerebral cortex. The hindbrain has parts known as the pons, cerebellum, and medulla oblongata. The hindbrain functions in maintaining balance and equilibrium, movement coordination, and processing some sensory information. The brainstem extends down to the spinal cord and is responsible for auditory and visual responses as well as motor function. The spinal cord is a bundle of nerve fibers that connects to the base of the brain and runs to the lower back. It functions to transmit information from points all over the body to the brain for processing. It also sends messages from the brain to many parts of the body. Let's review. We could not function without our central nervous system. It controls all parts of the body. It receives and interprets messages from all parts of the body and sends out instructions. The three main components of the central nervous system are the brain, spinal cord, and neurons.
Have you ever heard the expression that someone is a special snowflake? This expression stems from the very nature of snowflakes, that each one is entirely unique. But snowflakes aren't the only individually unique things in the world. We might just as easily refer to some people as special irises. That's because the iris in the human eye is just as unique as snowflakes. In the entire human population, not a single iris will be similar from one person to the next. The iris of the eye is the colored ring just behind the clear outer layer of the eye's cornea. Behind the iris is the crystalline lens, which helps to refract light. In the center of the iris is the pupil, the black hole that changes size. The iris is made of connective tissue, smooth muscle fibers, and pigments that give the iris its color. The pigments in the iris are made of melanin, or the same pigment that gives skin its color, and lipochrome. The amount of pigment in the eye creates eye color. Before you were born, genetics determined your eye color, and the unique structure of your iris was laid out. On one end of the spectrum are eyes with no pigment, which are the pink eyes of a person with albinism. A little pigment makes the eye blue. Then, in increasing amounts of pigment, are green, hazel, and finally brown eyes, which have the most pigment. Occasionally, someone has an eye that is one color and another eye that is a completely different color. And just to set the record straight, once you are an adult, your eyes don't change color. They can look different depending on what you are wearing or the light that surrounds you, but they aren't changing color. Because light eyes have less pigment, they need a little extra protection from the ultraviolet rays of the sun. A lot of sun exposure has been connected to a higher chance of cataracts and macular degeneration. Now, there is a good reason to wear cool shades, especially if you are light-eyed. Besides giving you a unique look, the iris does serve a functional purpose. The muscles of the iris contract and dilate the pupil of the eye to let in more or less light. The iris dilator muscle pulls away from the center of the iris to dilate or open the pupil, and the iris sphincter muscle pulls towards the center of the iris to constrict or close the pupil. When light is low, the iris muscles dilate the pupil to let in more light, and when the light is bright, the pupil constricts to limit the amount of light. When the pupil has to work extra hard when you are in a dark room watching the constantly changing light of your TV, then it can cause headaches or achy eyes. For that reason, it's better to keep some lights on in the room when you are watching TV. You have certainly heard of using fingerprints to distinguish between people, but relatively recently, ophthalmologists realized that each person's iris can also be used to differentiate between people. In 1994, the first patent for technology that can be used to recognize the iris of an individual was issued to a scientist named John Daugman. Of course, the technology of the 1990s has evolved since then, But iris recognition is still a very popular way to positively establish identity. In some settings, rather than carry a badge or remembering a password, your iris can be scanned to determine if it's really you. The iris of a human eye is the colored ring between the clear cornea on the outside and the crystalline lens on the inside. In the middle of the iris is the opening called the pupil. Each iris is unique and is genetically determined by the amount of pigment, melanin, and lipochrome, and the structure inside the iris. Light eyes have less pigment, and dark eyes have more. Smooth muscles of the iris constrict and dilate the pupil to regulate the amount of light that gets into the back portions of the eye. The uniqueness of each person's iris is able to be used in technology called iris recognition that can identify the uniqueness of a person's iris and distinguish their eyes from everyone else's.
The earliest stages of mammalian development following fertilization are very difficult to study. First of all, the gestational period of most mammals is long, which slows down the pace of research dramatically. In addition, most mammals produce only a few offspring at a time, and many, like humans, only produce one or two at a time, so there are fewer embryos to study. Almost all mammals develop inside the mother, which makes observation and manipulation difficult. In addition, mammalian zygotes are much smaller than other vertebrate zygotes. For instance, Xenopus laevis is a frog species that is often used to study development, and the volume of one of its eggs is 1,000 times larger than a human zygote. Now let's say that you want to study very early human development. You already can't see the single zygote or embryo because it's inside a person, and it's too small to study with ultrasound or other non-invasive imaging techniques. On top of that, you have many more ethical limitations when studying humans. You can't, for example, go out and breed people to produce embryos for scientific study. For these reasons, most developmental biology is done in non-mammalian species. Mice are most commonly used when studying mammalian developmental biology because they have one of the shortest mammalian gestational periods, they are easy to house and breed in large numbers, and they can have more than 10 pups in a single litter. Soon after fertilization, the mammalian zygote begins the process of cleavage, which is the process by which the zygote rapidly divides without growing to become multicellular. Cleavage uses the process of mitosis to replicate the genome and then divide the cells in half. However, unlike normal mitosis, there is no growth phase between divisions during cleavage. The first division occurs about a day after fertilization, and subsequent divisions occur every 12 to 24 hours after that. The zygote divides several times to form a mass of cells called a morula, which is an embryonic stage consisting of a solid, compact mass of 16 or more cells. The morula is the first embryonic stage where mammalian cells can be categorized as being either internal or external. The cells continue to divide, and when the mammalian morula reaches the 64-cell stage, the internal and external cells become separate lineages. The internal cells are called the inner cell mass, or ICM for short. The ICM will eventually become the embryo itself and its surrounding membranes. The external cells are called trophoblast cells. The trophoblast cells will play a key role in the process of implantation in the uterine wall and will eventually become the chorion, which is the embryonic portion of the placenta. Soon after the cells of the mammalian morula differentiate, the trophoblast cells secrete fluid into the center of the morula to create a fluid-filled space inside called the blastocele. Now that the embryo has taken the form of a hollow ball of cells, it is called a blastula. The trophoblast cells remain in a single outer layer surrounding the blastocele, and the cells of the ICM form a mass of cells on the inner surface on one side of the blastula. All vertebrates form a blastula during their development. However, in non-mammalian species, there is no inner cell mass. Instead, the blastula consists of just one cell layer. Unlike mammals, Other vertebrates do not attach to the uterine wall and do not form a placenta, so there is no need for the differentiation of trophoblast cells and the ICM. The specialized mammalian blastula that contains the two different cell types is called a blastocyst. The blastocyst is the last stage of mammalian development that occurs before implantation in the uterus. Let's review. After fertilization, the zygote begins the process of cleavage, which is the process by which the zygote rapidly divides without growing to become multicellular. Cleavage uses the process of mitosis without the growth phase to divide the zygote several times and form a mass of cells called the morula. The morula is an embryonic stage consisting of a solid, compact mass of 16 or more cells. The cells continue to divide, and when the mammalian morula reaches the 64-cell stage, the internal and external cells become separate lineages. The internal cells are called the inner cell mass, or ICM for short. The ICM will eventually become the embryo itself and its surrounding membranes. The external cells are called trophoblast cells. The trophoblast cells will play a key role in the process of implantation in the uterine wall and will eventually become the chorion, which is the embryonic portion of the placenta. In the next stage of vertebrate development, the embryo takes the form of a hollow ball called a blastula. In non-mammalian species, there is no inner cell mass. Instead, the blastula consists of just one cell layer. However, because mammalian embryos have to attach to the uterine wall and form a placenta, they form a specialized type of blastula called a blastocyst. A blastocyst is a mammalian blastula that contains two distinct cell lineages. In the mammalian blastocyst, the trophoblast cells remain in a single outer layer surrounding the blastocele, and the cells of the ICM form a mass of cells on the inner surface on one side of the blastocyst. While the early mammalian embryo develops on its way down the fallopian tube, it is still surrounded by the zona pellucida, which is a layer of extracellular matrix surrounding the mature oocyte, fertilized zygote, and pre-implantation blastocyst. The zona pellucida surrounds the developing embryo during its entire journey down the fallopian tube and prevents the embryo from attaching prematurely to the wall of the fallopian tube. If the embryo implants in the fallopian tube, it will grow larger and cause an ectopic pregnancy outside of the uterus, which is the mammal-specific organ where prenatal development occurs. The fallopian tube isn't big enough to handle a developing embryo, so a prematurely implanted embryo will outgrow the tube. Without intervention, the tube will burst, which can be fatal to the mother. But the blastocyst does eventually need to implant in the uterus. So how does it do this if it's surrounded by the zona pellucida? Once the blastocyst reaches the uterus, it uses a protein-digesting enzyme, or protease, to make a hole in the surrounding zona pellucida. It then squeezes through the hole to hatch out of the zona pellucida. Proteins on the outer surface of the blastocyst bind to the extracellular matrix of the uterine wall and allow the blastocyst to attach. Once attached, the blastocyst secretes a number of different proteases that digest the extracellular matrix of the uterus and allow the blastocyst to bury itself in the uterine wall. This process by which the blastocyst binds to and then buries itself within the uterine wall is known as implantation. As the blastocyst implants, the trophoblasts that make up the outer layer of cells multiply in number and invade deeper into the uterine wall. They maintain a protective barrier between the maternal cells and the developing embryo. As you might remember, the trophoblasts are the cells that will eventually form the chorion, or the embryonic-derived portion of the placenta. As the trophoblasts multiply in number and extend further into the wall, they encounter and surround maternal blood vessels that already exist within the maternal tissue of the uterus. These maternal blood vessels then fuse with spaces between the trophoblasts to form a network of sinuses that are filled with maternal blood. Even though the blood comes from the mother's circulatory system, flows through the sinuses, and then re-enters the mother's blood vessels, it's important to note that the sinuses are not blood vessels because they do not have a lining of endothelial cells to contain the blood. Instead, the blood comes into direct contact with the embryonic trophoblast cells. These trophoblasts, interspersed with the blood-filled sinuses, surround the entire developing embryo. Eventually, the blood-filled sinuses expand and the trophoblasts form finger-like structures called chorionic villi. These finger-like structures of the placenta, composed of embryo-derived trophoblasts, become surrounded by maternal blood in what is now called the intervillous space. In the meantime, the embryo itself continues to grow and develop. As it does, one of the very first organ systems to develop is the circulatory system. At only 3 weeks after implantation, the embryo already has a basic circulatory system, which in addition to growing within the embryo itself, also grows and extends into the chorionic villi as well. These embryonic blood vessels grow very close to the intervillous space, which allows nutrients to diffuse from the maternal blood through the trophoblasts and into the embryonic bloodstream. While nutrients are entering the embryonic bloodstream, waste products diffuse out of the embryonic blood vessels into the trophoblasts and finally into the maternal blood, which carries it away from the embryo. Once the embryonic circulatory system has been established, the continuous supply of nutrients allows the embryo to begin to actually grow in size and mass. As it grows and expands, it pushes the chorionic villi furthest away from the maternal blood supply into the uterine cavity. These villi that end up in the cavity eventually degenerate, and the maternal blood supply retreats from these areas as well. This leaves functioning chorionic villi and maternal blood on only one side of the embryo. This composite structure of embryonic and maternal tissues that supplies nutrients to the developing embryo is called the placenta. Let's review. While the early mammalian embryo develops on its way down the fallopian tube, it is still surrounded by the zona pellucida, which is a layer of extracellular matrix surrounding the mature oocyte, fertilized zygote, and pre-implantation blastocyst. The zona pellucida surrounds the developing embryo during its entire journey through the fallopian tube and prevents the embryo from attaching prematurely to the wall of the fallopian tube. Once the blastocyst reaches the uterus, it uses a protein-digesting enzyme, or protease, to make a hole in the surrounding zona pellucida. It then squeezes through the hole to hatch out of the zona pellucida. Proteins on the outer surface of the blastocyst bind to the extracellular matrix of the uterine wall and allow the blastocyst to attach. Once attached, the blastocyst secretes a number of different proteases that digest the extracellular matrix of the uterus and allow the blastocyst to bury itself in the uterine wall. This process, by which the blastocyst binds to and then buries itself within the uterine wall, is known as implantation. As the blastocyst implants, the trophoblasts that make up the outer layer of cells multiply in number and invade deeper into the uterine wall. As they do this, they encounter and surround maternal blood vessels that already exist within the maternal tissue of the uterus. These maternal blood vessels then fuse with spaces between the trophoblasts to form a network of sinuses that are filled with maternal blood. Eventually, the blood-filled sinuses expand and the trophoblasts form finger-like structures called chorionic villi. These finger-like structures of the placenta, composed of embryo-derived trophoblasts, become surrounded by maternal blood in what is now called the intervillous space. Embryonic blood vessels grow into the chorionic villi, very close to the intervillous space, which allows nutrients to diffuse from the maternal blood through the trophoblasts and into the embryonic bloodstream. This composite structure of the embryonic and maternal tissues that supplies nutrients to the developing embryo is called the placenta. It is the placenta that supplies the nutrients to the developing embryo and allows it to grow larger. So you may remember that the placenta is the composite structure of embryonic and maternal tissues that supplies nutrients to the developing embryo. The placenta is a unique organ found only in mammals that allows the mother to provide a very large amount of nutrients to her offspring for an extended period of time before they are even born. The placenta serves as an interface between the mother and the developing fetus and has three main jobs: attach the fetus to the uterine wall, provide nutrients to the fetus, and allow the fetus to transfer waste products to the mother's blood. The placenta is composed of both maternal tissue and tissue derived from the embryo. The chorion is the embryonic-derived portion of the placenta. It is composed of trophoblasts, which you may remember are the cells that made up the outer cell layer of the blastocyst. You may also remember that during implantation, the trophoblasts multiply in number and extend into the uterine wall. They eventually form finger-like structures called chorionic villi, which are finger-like structures of the placenta composed of embryo-derived trophoblasts. The chorionic villi are surrounded by maternal blood, which comes into direct contact with the embryonic trophoblast cells. The intervillous space is the part of the placenta that surrounds the chorionic villi and contains maternal blood. Maternal blood flows from the mother's circulatory system through the intervillous space and then re-enters the mother's blood vessels. Fetal blood flows from the fetus into two main arteries in the umbilical cord, through the capillary network of the chorionic villi, and is returned to the fetus by the umbilical vein. The maternal blood that enters the placenta is nutrient and oxygen rich. A growing fetus obviously requires lots of nutrients and oxygen that it cannot provide for itself, so the fetal blood that flows through the capillaries of the chorionic villi is nutrient and oxygen poor. Because of this disparity in the nutrient and oxygen concentration between the fetal blood in the villi and the maternal blood in the intervillous space, nutrients and oxygen diffuse from the maternal blood into the villi and into the fetal blood. Conversely, the fetal blood contains high concentrations of carbon dioxide and other waste products, while the maternal blood in the intervillous space contains low concentrations of these waste products. As a result, carbon dioxide and other waste products diffuse out of the fetal blood, across the trophoblasts of the chorionic villi, and into the maternal blood that surrounds the villi. The mother's lungs, kidneys, and liver will easily remove these waste products from her blood. Notice that in the placenta, the maternal and fetal blood does not mix. The two blood supplies remain separated by the fetal trophoblasts and endothelial cells. However, nutrients and waste products, including oxygen and carbon dioxide, are free to diffuse from one blood supply to the other down concentration gradients. Embryonic trophoblasts are not incorporated into the tissues of the growing fetus, but they are still very important and versatile cells. After the chorionic villi are established, many of these trophoblasts fuse together to form very large syncytiotrophoblasts, which are large multinucleated trophoblasts that are formed by the fusion of several smaller cells. These syncytiotrophoblasts are a main component of the chorionic villi and are the cells that are in direct contact with the maternal blood. Syncytiotrophoblasts are also the major source of most placental hormones, including human chorionic gonadotropin, which is also known as HCG. Yet other trophoblasts form columns of cells which serve to anchor the placenta to the uterus. These junctional trophoblasts secrete extracellular matrix proteins that help them anchor the placenta to the uterus. Let's review. The placenta is the composite structure of embryonic and maternal tissues that supplies nutrients to the developing embryo. The placenta serves three main functions: attach the fetus to the uterine wall, provide nutrients to the fetus, and allow the fetus to transfer waste products to the mother's blood. The placenta is composed of both maternal tissue and tissue derived from the embryo. The chorion is the embryonic derived portion of the placenta. It is composed of fetal blood vessels and trophoblasts, which are organized into finger-like structures called chorionic villi. The chorionic villi are surrounded by the intervillous space, which is the part of the placenta surrounding the chorionic villi that contains maternal blood. Maternal blood flows from the mother's circulatory system through the intervillous space and then re-enters the mother's blood vessels. Nutrients and oxygen diffuse from the maternal blood into the villi and into the fetal blood. At the same time, carbon dioxide and other waste products diffuse out of the fetal blood across the trophoblasts of the chorionic villi and into the maternal blood surrounding the villi. Embryonic trophoblasts are very versatile cells. Some trophoblasts fuse together to form very large syncytiotrophoblasts, which are large, multinucleated trophoblasts that are formed by the fusion of several smaller cells. These syncytiotrophoblasts are a main component of the chorionic villi and are the cells that are in direct contact with the maternal blood. They are also the major source of most placental hormones. Yet other trophoblasts form columns of cells which serve to anchor the placenta to the uterus. These junctional trophoblasts secrete extracellular matrix proteins that help them anchor the placenta to the uterus. The placenta is clearly the most important extraembryonic structure for placental mammals. However, there are other extraembryonic structures that also contribute to the development of mammals and other vertebrates as well. First, let's talk about the functions of these structures during the development of a vertebrate that lays eggs, like a bird, since egg-laying mammals evolved before placental mammals. Birds and other egg-laying vertebrates have four different extraembryonic membranes: the chorion, the allantois, the yolk sac, and the amnion. In vertebrate eggs, the chorion is the outermost membrane and lines the inside of the eggshell. You may remember, though, that in mammals, the chorion forms the embryonic portion of the placenta. The allantois is the sac-like extraembryonic membrane that removes waste from the embryo. As the embryo grows, so does the allantois, eventually pressing against the chorion. This allows the allantois to exchange oxygen and carbon dioxide with the air outside the eggshell. In addition, the allantois also serves as a disposal site for uric acid. The yolk sac is the extraembryonic membrane that surrounds the egg yolk. The yolk sac has a well-developed vascular system that transports nutrients from the egg yolk to the developing embryo. And finally, the amnion is the extraembryonic membrane that surrounds the developing embryo. The amnion is filled with fluid, and its main job is to serve as a shock absorber to protect the embryo against any jarring impacts or movements. As you can see, the four different extraembryonic membranes each has a specific function in vertebrates that lay eggs. However, when mammals began using the placenta to nourish their unborn offspring, the needs of the embryos changed, and some of the extraembryonic membranes were adapted to serve other purposes. Please note that for the purposes of this lesson, when I talk about mammals, I am referring to placental mammals. The development of egg-laying mammals like the duck-billed platypus and the echidna more closely resemble the development of other egg-laying vertebrates like birds and reptiles. As you may remember, in mammals, the chorion forms the embryonic portion of the placenta. Before implantation, the mammalian and bird chorions are quite similar in structure. However, as the mammalian embryo implants and the placenta develops, it becomes far more complex and serves a very different function from the chorion of birds. The allantois also has to be greatly modified to serve the purposes of placental mammals. Instead of forming a sac-like structure to dispose of wastes, the allantois of mammals forms part of the umbilical cord. It is interesting to note that in this case, evolution conserved the waste disposal function of the allantois, but the structure of the allantois was greatly modified to form an umbilical cord that carries the waste away from the embryo and towards the mother's blood vessels. Placental mammals have no yolk, so the yolk sac contains only fluid and does not play a major role in nourishing the embryo. However, the mammalian yolk sac does maintain its well-developed vascular structure and contributes to the vascular system of the developing embryo. Of all four extraembryonic membranes, the only one that maintains the same form and function in birds and mammals is the amnion. Just like in birds, the amnion of mammals forms a fluid-filled sac around the embryo and serves as a shock absorber. One could make the argument that since mammalian embryos go wherever their mothers do, the function of the amnion is more crucial in mammals than it is in egg-laying vertebrates, whose eggs may not move between the time that they are laid and when the offspring hatch. In any case, the fluid contained inside the amnion is called amniotic fluid and is also necessary for normal development of the lungs and limbs. The amniotic fluid not only surrounds the fetus, but it also fills its lungs, mouth, and nasal cavity. In cases where amniotic fluid volumes are very low, the lungs or the limbs of the fetus may not fully develop. After the baby is born and needs to start breathing air, underdeveloped lungs can be fatal. As a result, amniotic fluid volume is often used as one of the indicators of healthy development during pregnancy. Let's review. Vertebrates have four different extraembryonic membranes: the chorion, the allantois, the yolk sac, and the amnion. In vertebrates that lay eggs, the chorion is the outermost membrane and lines the inside of the eggshell. The allantois is a sac-like extraembryonic membrane that removes waste from the embryo. The allantois exchanges oxygen and carbon dioxide with the air outside the eggshell and serves as a disposal site for uric acid. The yolk sac is the extraembryonic membrane that surrounds the egg yolk. The yolk sac has a well-developed vascular system that transports nutrients from the egg yolk to the developing embryo. And finally, the amnion is the extraembryonic membrane that surrounds the developing embryo. When mammals began using the placenta to nourish their unborn offspring, the needs of the embryos changed, and some of the extraembryonic membranes were adapted to serve other purposes. The chorion was adapted to form the embryonic portion of the placenta, and the allantois of mammals evolved to form part of the umbilical cord. The mammalian yolk sac doesn't contain any yolk, but it does maintain its well-developed vascular structure and contributes to the vascular system of the developing embryo. Of all four extraembryonic membranes, the only one that maintains the same form and function in birds and mammals is the amnion. Just like in birds, the amnion of mammals forms a fluid-filled sac around the embryo and serves as a shock absorber. The fluid contained inside the amnion is called amniotic fluid. In addition to serving as a liquid cushion, amniotic fluid is also necessary for normal development of the lungs and limbs. Today, we're going to talk about how the early vertebrate embryo transforms itself from a very simple ball of cells into a more complex embryo with multiple layers of different cell types. We're going to talk about gastrulation, which is the process that results in the formation of three distinct germ layers in the early embryo. Gastrulation is a necessary process that occurs in all multicellular organisms except for sponges. Sponges are unique among multicellular animals in that they do not contain multiple tissue types. Their entire body is composed of basically one type of cell. Because they do not need to differentiate their cells, they also do not need to go through the process of gastrulation. However, all other animals do need to differentiate their cells, and the first step in this process is gastrulation, which is accomplished in different ways in different species. In some animals that lay eggs, such as frogs, gastrulation is accomplished when the hollow blastula invaginates to form an inner layer of cells called the endoderm, and an outer layer of cells called the ectoderm. A third middle layer of cells, called the mesoderm, is formed as cells begin to populate the space between the endoderm and the ectoderm. These three layers, the endoderm, the ectoderm, and the mesoderm, are called the primary germ layers. So here's a frog blastula. If you imagine that someone is poking the blastula and pushing one side of it in, that's what the process of invagination looks like. The hollow ball now looks like a cup with an inner layer of endoderm and an outer layer of ectoderm. This cup-like structure is called a gastrula. The mesoderm is formed when cells begin to form a middle layer between the endoderm and the ectoderm. Eventually, the endoderm will push all the way through the bottom of the cup to form a tube with an opening on each end. The two openings will eventually become the mouth and anus of the animal, and the inner cavity of the tube will become the gut. In mammals, gastrulation is achieved in a very different way. This is because the outer layer of the mammalian blastocyst, which is roughly equivalent to the blastula, becomes the mammalian placenta and is not available to create the primary germ layers of the embryo. Instead, the inner cell mass, or ICM, has to form the three primary germ layers of the mammalian embryo after the blastocyst has implanted in the uterine wall. Instead of forming a hollow ball and invaginating to create the gut tube and the three cell layers, the ICM of the mammalian embryo forms a single layer of cells called the epiblast. As these epiblast cells proliferate, some of them migrate out of the epiblast to form the endoderm, which is this layer here. And then after that, more cells migrate out to form the mesoderm, which is this cell layer here. The cells that remain in the epiblast form the ectoderm, which is this cell layer up here. With the formation of the three primary germ layers, the embryo has begun the process of tissue differentiation. Each of the three primary germ layers will form a different set of specific organs and tissue types in the animal. The ectoderm will form the nervous system, the epithelial cells of the mammary glands, the lens of the eyes, hair, pigment cells, and the epidermis of the skin. The mesoderm will form skeletal muscle, smooth muscle, the heart, blood vessels, blood cells, kidney, spleen, fat cells, the skeleton, most connective tissues, and most of the urogenital system. The endoderm will form the entire epithelial lining of the gastrointestinal tract, as well as the liver, pancreas, gallbladder, thyroid, the epithelial lining of the trachea, and the respiratory surface of the lungs. Let's review. Gastrulation is the process that results in the formation of three distinct germ layers in the early embryo. In some animals that lay eggs, such as frogs, gastrulation is accomplished when the hollow blastula invaginates to form an inner layer of cells called the endoderm and an outer layer of cells called the ectoderm. When the blastula undergoes gastrulation, it's as if someone is pushing one side of the blastula in to form a cup-like structure with two cell layers. A third middle layer of cells, called the mesoderm, is formed as cells begin to populate the space between the endoderm and the ectoderm. These three layers, the endoderm, the ectoderm, and the mesoderm, are called the primary germ layers. After gastrulation, the cup-like embryonic stage that contains at least two distinct germ layers is called the gastrula. Eventually, the endoderm will push all the way through the bottom of the cup to form a tube with an opening on each end. The two openings will eventually become the mouth and anus of the animal, and the inner cavity of the tube will become the gut. In mammals, the outer layer of the blastocyst forms the placenta and is not available to create the primary germ layers of the embryo. Instead, the inner cell mass, or ICM, forms all three primary germ layers of the mammalian embryo. The ICM of the mammalian embryo forms a single layer of cells called the epiblast. As these epiblast cells proliferate, some of them migrate out of the epiblast to form the endoderm, and then the mesoderm. The cells that remain in the epiblast form the ectoderm. Each of the three primary germ layers will form a different set of specific organs and tissue types in the animal. The ectoderm will form the nervous system, the epithelial cells of the mammary glands, the lens of the eyes, hair, pigment cells, and the epidermis of the skin. The mesoderm will form skeletal muscle, smooth muscle, the heart, blood vessels, blood cells, kidney, spleen, fat cells, the skeleton, most connective tissues, and most of the urogenital system. The endoderm will form the entire epithelial lining of the gastrointestinal tract, as well as the liver, pancreas, gallbladder, thyroid, the epithelial lining of the trachea, and the respiratory surface of the lungs.